Importance of triglyceride, TAG. Triglycerol, importance of triglycerols. Triglycerol are the ester of glycerols with fatty acid structure. Triglycerol consists of 3 molecular of fatty acids ester to 1 molecular of glycerol. Properties of triglycerol: hydrolysis. Properties are hydrolysis, saponification, rancidity, lipid peroxidation. Triglycerols are hydrolyzed by lipase and release fatty acid and glycerol. The hydrolysis biochemical importance: the fat and oils that are widely distributed in both plants and animals are chemically triglycerols. The major lipids in the diet are triglycerols. TAG is the major energy storing lipids. In diabetes mellitus, hypertriglycerolemia reveals insulin deficiency. Fatty acid. Define and classify fatty acids with example. Classification of fatty acids. Fatty acids: unsaturated and saturated. Saturated. Mono, unsaturated, polyunsaturated, and eicosanoids. Unsaturated and saturated contain no double bond in their hydrocarbon. No double bond. Contain one or more double bond. Unsaturated. Saturated. Mono, unsaturated, polyunsaturated, eicosanoids. Monounsaturated, for example, oleic acid, polyunsaturated, and eicosanoids, for example, prostaglandins. Dianoic acid, linoleic acid, trianoic acid, linoleic acid, tetranoic acid, arachidonic acid, pentanoic acid, timodonic acid, hexanoic acid, carbonic acid. On the basis of number of carbon atoms, chain length, short chain fatty acids containing less than 10 carbon atoms. Less than 10 carbon atoms. For example, acetic acid and propanoic acid. Acetic acid and propanoic acid. Long chain fatty acids containing more than 10 carbon atoms. For example, palmitic acid, stearic acid. Nutritional classification: non-essential fatty acids. They are fatty acids which body can synthesize. Palmitic acid, stearic acid, etc. Essential fatty acids: these are polyunsaturated acids, PUFA, cannot be synthesized in the body by the body and therefore must be supplied by diet. And for example, linoleic acid, lanolinic acid, and arachidonic acid. Common sources of essential fatty acids: vegetable oil, fish oil, cod liver oil, egg yolk. Name the essential fatty acids with their dietary source and biochemical importance. Essential fatty acids: the polyunsaturated fatty acids that cannot be synthesized in the body and therefore must be supplied in diet are known as essential fatty acids. Essential fatty acids are regarded as essential because their presence in diet is essential, since they are not produced in the body. Essential fatty acids for human body. Linoleic acid. It is 18C omega-6 fatty acid with double bonds. Lanolinic acid. It is 18C omega-3 fatty acid for 3 double bonds. Arachidonic acid. It is 20C omega-6 fatty acid with 4 double bonds. Name, structure, and source. Name: linoleic acid, alpha linoleic acid, and arachidonic acid. Linoleic acid, alpha linoleic acid, omega-6 18, alpha linoleic acid, and arachidonic acid. Alpha linoleic acid, omega-6 18, alpha linolenic acid, omega-3 18, 18:3, and arachidonic acid omega-6 20:4. Source: corn, peanut, cottonseed, soybean, and many plant oils. Frequently found with linoleic acid but mainly in linseed oil. Found in animal fats, peanut oil, and important components of phospholipids in animals. Arachidonic acid becomes essential when precursor linoleic acid is absent on diet. Biochemical importance and functions: maintains fluidity of biological membrane. They serve as precursor of eicosanoids, decreases plasma cholesterol levels by dietary means and thus reduces the risk of coronary heart disease, increases clotting time and fibrinolytic activity, synthesis of structural lipid, concerned with optimum gonadal activity, cure skin lesions. Deficiency features: deficiency of EFA in the diet causes dermatitis, impaired glandular function, fatty liver, faulty vision, poor wound healing, and hair loss, etc. Nice to know. Numbering and distinction of carbon in fatty acids. Carbon atoms are numbered from carbon, carbonyl carbon, carbon 1. The carbon atoms adjacent to the carboxylic carbon are also known as alpha carbon and gamma carbons, respectively. And this terminal methyl carbon is known as omega or N carbon. The double bonds in a fatty acid can also be denoted relatively to the omega or methyl terminal end of the chain, starting from the methyl end. Carbon atoms in a fatty acids are numbered as omega 1, 2, 3, etc. The numbering of carbon atoms in two different ways is given below. Omega 1, 2, 3, 4, 5, 6. The number before the colon indicates the number of carbons at the chain. Delta 4 indicates the number of positions in double bonds. Delta 9 indicates a double bond between carbon 9 and 10 of the fatty acids. Omega 9 indicates a double bond on the ninth carbon containing form of omega carbon. For example, in arachidonic acid, in an omega 6 fatty acid, because the terminal double bond is 6 in found in omega end. It is 20 C long and has 4 double bonds. Draw the structure with name of fatty acids with the formation and answer. What do you mean by omega 3, omega 6 fatty acids? Give the importance. Fatty acids. These are long chain puffers with the first double bond beginning at 6th bond position when starting from the methyl and end of the fatty acid molecule. For example, linoleic acid. It is called omega 6 fatty acid because here the double bond closest to the omega end begins at 6 carbon around from the end. Arachidonic acid. It is called omega 6 fatty acid because here the double bonds closer to the omega end begins at carbon 6 carbon concentrated from this end. Source: vegetable oils, for example nuts, avocados, olive oil, soybeans, and various oils including sesame, coconut seed, and corn oil are the commonest source of fatty acids. Importance: they are they lower plasma cholesterol when substituted for saturated fats. Plasma LDL are lowered, but HDL, which protect against coronary heart disease, are also lowered. The powerful benefit of lowering LDL are always partially offset because the decreased HDL. The linoleic acid along the alpha linoleic acid and omega 3 fatty acids are required for fluidity of membrane structure and synthesis of eicosanoids. Omega 3 fatty acids. These are long chain PUFS with the first double bond beginning at the third bond position from methyl end. For example, linoleic acid. This is called omega 3 fatty acid because here the double bond is closest to the omega and it begins at the third carbon containing from this end. Source: omega 3 polyunsaturated fats. Principal alpha linoleic acids are found in plants, fish oil, For example, Ilsa salmon also contains it. Importance: dietary. Omega-3 polysaturated fats suppress cardiac arrhythmias, reduces serum triglycerides, decreases the tendency for thrombosis, lower blood pressure, and subsequently reduces the risk of cardiovascular mortality. But they have little effect on low density lipoprotein and elder cholesterol level. The omega-3 polysaturated fatty acids are included in infant formulas to promote brain development. Note well, carbon atoms of unsaturated fatty acids UFA are numbered, beginning at the omega carbon end, methyl terminal of the chain. In these norms, PUFA are often classified as omega-3 fatty acids, omega-6 fatty acids, etc., according to the position of the first double bond from the W carbon. Mention the source and the nutritional significance of unsaturated fatty acids. Fatty acids that contain one or more double bonds are called unsaturated fatty acids. They represent about 50% of the body's fatty acid pool. Type 2 unsaturated fatty acids. Monostructured fatty acids, MUFA. They contain one double bond. Source: found in olive oil, canola oil, etc. Mention the source and nutritional significance of unsaturated fatty acids. Unsaturated fatty acids. Fatty acids contain one or more double bonds are called unsaturated fatty acids. They represent about 50% of the body fat pool. Monosaturated fatty acids. Polysaturated fatty acids. Classify the polyunsaturated fatty acids according to the number of double bond. Dianoic acid, tetroic acid, tetraionic acid, pentionic acid, hexanic acid. Linoleic acid, linolenic acid, arachidonic acid, thymodonic acid, and carbonic acid. Biochemical importance: polyunsaturated fatty acids decrease plasma cholesterol level by dietary means and thus reduces the risk of the coronary heart disease. 20 carbon polyunsaturated fatty acids give rise to eicosanoids. Write down the sources and importance of polyunsaturated fatty acids. Source and polyunsaturated fatty acids: cotton oil, soybean, sunflower oil, corn, fish, etc. Ghee and coconut oil are poor sources of PUFA. Clinical importance of PUFA: dietary intake of polyunsaturated fatty acids reduces the plasma cholesterol level. PUFA will help in transport of cholesterol by LCTA mechanism and excretion from the body. Give the importance of arachidonic acid. Importance of arachidonic acid. Arachidonic acid 5, 8, 11, 14. Eocosa tetranoic acid is the precursor for most of the prostaglandin in humans, including prostacyclins and thromboxanes. Arachidonic acid is one of the essential fatty acids which are the structural components of biological membrane, prevents fat accumulation in the liver, maintains proper growth and reproduction of the organisms. Eicosanoids. There are specialized fatty acids derived from Eicosa. 20 carbon polyunsaturated fatty acids are included: prostaglandins, thromboxanes, and prostacyclins, leukotrienes, and lipoxygens. Biosynthesis: arachidonic acid is an Eocosa polyunsaturated fatty acids that is derived from essential fatty acid, linoleic acid. It does not occur freely in the cell, remain as stratified normally in membrane phospholipids. After breakdown of membrane phospholipid, arachidonic acid is released into list and produces eicosanoid by lipoxygenase and cyclooxygenase pathways. Cell membrane phospholipid: arachidonic acid, leukotrienes, lipoxygenase, prostaglandins, prostacyclin and thromboxinase alpha 1. Functions of eosinoids. Prostaglandin, cause of vasodilation. Proteinic edema, pain and inflammation. Prostacyclin, cause of vasodilation. Inhibits platelet aggregation. Thromboxane A, cause of vasoconstriction. Promotes platelet aggregation. Leukotrienes, causes vasoconstriction. Promotes platelet aggregation, causes bronchospasm. Lipoxins cause vasodilation, inhibit neutrophil chemotoxins and adhesion, stimulate monocyte adhesion. What are the prostaglandins? Prostaglandins are one type of eosinoids derived from eocosa, polyunsaturated fatty acids, through cyclogenase pathways. Subtypes. They are derived from the basis of number of double bonds like PG1, PG2, PG3, substitute group of proteins like PGD2, PGE2, PGF2 alpha. Production of prostaglandins. Membrane phospholipids, arganoid acid and prostaglandin. Function of prostaglandin. They act as local hormone. They are mediators of inflammation and homeostasis. Causes vasodilation. Proteinic edema, involved in the pathogenic pain and fever inflammation. Therapeutic use includes prevention of conception. Induction on labor in term. Induction of labor at term. Termination of pregnancy. Prevention of gastric ulcer. Control of inflammation and blood pressure. The diaphragm. It is a dome-shaped musculo-amniotic portion which innervates between the thorax and the abdomen. The diaphragm forms the root of the abdomen. The thoracic surface is convex on the left and the right sides, and it is depressed in the middle. The summits of the convexity are known as cupolae. The right cupola is slightly higher than the left due to the presence of liver. The peripheral part of the diaphragm is muscular strength, and the central part is tendinous, which is occupied by the central tendon origins. It arises from the inner surface of the thoracic outlet, and the origins are arranged in three groups: sternal, costal, and vertebral. Sternal by two fleshy slits from the back of the xiphoid process. Costal from the inner surface of the lower six ribs and their costal cartilages interdigiting with the transverse abdominis. Vertebral. On either side, it arises from the three sources: a pair of crura right and the left, vertebral pair of right and the left, from a pair of medial arcuate ligaments, from a pair of lateral arcuate ligaments, from a pair of crura right and the left. The right crura is longer and more muscular than the left. It arises from the front of the bodies of intervertebral discs, the upper three lumbar vertebrae. The fleshy right crura exerts more work to displace a heavy organ like the liver during inspiration close to the esophageal opening. The right crura gives attachment to the suspensory muscle to the duodenum, which is attached below to the duodenal flexure, which is attached below the duodenal flexure. Left crus arises from the bodies of the disc of upper two lumbar vertebrae only. Both crura are connected across the midline by a abdominal arcuate ligament, by a medial arcuate ligament. The left cruise is connected to the bare area of the posterior surface of the cardiac and stomach by the gastrophrenic ligament. Connected to the bare area of the posterior surface of the cardiac and stomach by the gastrophrenic ligament. From the pair of a medial arcuate ligament, medial thombrocostal arch. Each ligament bridges across the front of the psoas major and is formed by the thickening of the psoas fascia. It is attached medially to the body of the first and the second lumbar vertebra, laterally to tip the transverse process of the first lumbar vertebra. Each ligament a pair of lateral articuate ligament. Each ligament arches in front of the quadratus lumborum and is formed by the thickening of anterior layer of thoracolumbar fossa. It is attached medially to the tip of the transverse process of the first lumbar vertebra and the lateral to the lower border of the 12th vertebra. Insertions. All fibers are inserted into the center tendon, which is situated in the middle in the median depressed part of the diaphragm. Insertion. All fibers are inserted into the central tendon, which is situated in the middle depressed part of the diaphragm, close to the sternum. All fibers are inserted into the central tendon, which is situated in the median depressed part of the diaphragm, close to the sternum. The central tendon is shaped like a trefoil leaf, presenting median right and left leaflets. At the junction of the median and right leaflets, it presents the vena cava opening, and to the left of the opening is present a central point of the decussion from which four diagonal bands radiate. Sternal fibers are the shortest and those who arise from the ninth costal cartilage are the longest. Sternal fibers are the shortest and those who arise from ninth costal cartilage are the longest. Opening and the diaphragm. The openings of the diaphragm are major and minor. Major openings. These are vena cava, esophageal and aortic. Vena cava. It is situated on the central tendon at the junction of the median of the right leaflet, opposite the disc between the 8th and the 9th thoracic vertebrae, about 2.5 centimeters to the right and the left of the midline. Situated when about 2.5 centimeters to the right of the medial line, opposite to the 8th on the 9th thoracic vertebra. Shape. It is quadrilateral structure passing inferior vena cava, the wall of which is adherent to the fibrous ring of the opening. Inferior vena cava. A few branches of the right phrenic nerve. A few lymph vessels from the liver. Few on the right phrenic nerve. Effects of contraction. Which of the diaphragm contracts in inspiration, the vena cava opening dilates and more blood enters the right atrium. Esophageal opening situation. The muscular part of the diaphragm at the level of the 10 thoracic vertebra, that is 8 এবং 10, about 1.25 centimeters on the left side, on the left middle. Shape and formation. It is elliptical in shape and formed by the splitting of the fibers of the right growth. Structures passing. Esophagus, anterior posterior vagal trunks, formed respectively by the left and the right vagus nerves. Esophageal branches of the left gastric artery and the corresponding tributaries of the left gastric vein. Lymphates from the liver. Phrenoesophageal ligament extends upwards from the subdiaphragmatic fascia and fibrous coat of the esophagus. Effects of concentration. It prevents the irregular blood. It prevents the regurgitation of food from the stomach to the esophagus during inspiration from opening and constraint. From opening is during this inspiration the opening is constricted, hence inspiration and deglutition cannot take place simultaneously. Opening, aortic opening. Situation. It is situated behind the diaphragm, esopneumortic, at the level of the lower border of the thoracic vertebra, slightly to the left of the medial plane. Slightly to the left of the medial plane. Left. Shape. It is rounded. Boundaries. Medial arcuate ligament. Body of the true vertebra. Corresponding crus. Structures passing: abdominal aorta, thoracic duct, and sometimes azygous vein. When the artery is as lumbar azygous vein, sometimes azygous vein when it arises at lumbar azygous vein. Effect of construction: no change. Minor openings: a triangular gap known as the space of Larry is present between the sternal origin and that from the seventh costal cartilage. It transmits superior epigastric vessels and a few lymphatics. Space of Larry between the sternal origin or from the seventh costal cartilage. It transmits superior epigastric vessels and a few lymphatics from the liver. When the opening is enlarged, it is known as a foramen. When the opening is large, it is known as foramen Morgagni. Between the digitation of the costal origin of the diaphragm and the transverse abdominis, lower 5 intercostal vessels and nerves. Between the digitation of the costal origin of the diaphragm between the transverse abdominis. Between the costal origin of the diaphragm from the 7th to the 8th cartilages, muscular phrenic vessels. Seventh and the 8th costal cartilage, muscular phrenic vessels. Behind the lateral articulate ligament, subcostal vessels and nerves. Behind the medial articulate ligament, sympathetic trunk, and sometimes the less splanchnic nerve, piercing each crus, great and less splanchnic nerves, and pierced by the inferior azygous vein. Left phrenic nerve pierces the left pleura in front of the central tendon. Sometimes a small opening is present in the central tendon between the medial and the left reflex. It transmits a small vein which is remnant on the suprasplenic part of the left vitelline vein. Nerve supply, motor supply. The motor supply of the entire diaphragm is derived solely from the phrenic nerve C6, C3, C4, C5. Nerve supply. The motor supply. Each pleuric nerve pierces the diaphragm, ramifies on the abdominal surface, and supplies the muscle on the abdominal surface. Each nerve divides usually into anterior medial, sternal, anterior lateral, posterior medial, and posterior lateral branches. It's into anterior medial, anterior lateral, posterior medial, posterior. Right phrenic nerve supplies the right part of the diaphragm up to the right margin of the esophageal opening, and the left phrenic nerve supplies the left part of the diaphragm up to the left margin of the esophageal opening. Therefore, the right crus is supplied by both. Therefore, the right crus is supplied by both phrenic nerves. The right phrenic nerve supplies the right part of the esophagus up to the right margin of the esophageal opening. The left phrenic nerve supplies the left part of the esophagus up to the left margin opening of the esophageal opening. Therefore, the right crus is supplied by both nerves, whereas the left crus is supported only by the left phrenic nerves. Sensory support: central part by phrenic nerves, peripheral part by lower 6 or 7 intercostal nerves. Blood supply: muscular phrenic and pericardiophonic arteries, branches of the internal thoracic artery, lower 5 to 6 posterior intercostal artery, superior phrenic artery, last branch of the descending thoracic aorta, inferior phrenic artery, first branch of the abdominal aorta, veins corresponding with the arteries, and the drain into the systemic veins. Actions of diaphragm on the thoracic cavity. It is the principal muscle of inspiration. When the diaphragm contains, lower ribs are fixed. Fixed vault of the diaphragm descends from the ventral determinants of the thorax increases. Upper abdominal viscera are displaced below the earth. When the limit of the bulging is reached, central tendon becomes fixed, and the further contraction lower ribs are elevated. Thereby, anterior, posterior, and the transverse diameter of the thorax are increased. Average range of the movement of diaphragm is required breathing is about 1.5 cm. Absolute range of movements in deep breathing varies between 6 cm to 10 cm. Total surface area of the adult diaphragm is about 120 or 270 square centimeter. Therefore, out of 500 milliliters of tidal air, about 400 milliliter of air is down into the lungs and quiet respiration by descent diaphragmatic aorta. Eventually, the diaphragm is the principal muscle of the inspiration. On the abdominal cavity, it is composed of abdominal viscera and increases the abdominal pressure. Abdominal pressure on the abdominal cavity. On vomiting, micturition, defecation, micturition, on the three major openings. During inspiration, vena cava opening dilates, esophageal opening constricts, and aortic opening undergoes no change. Effects of posterior of the movement of diaphragm. When the diaphragm is released, the height of the increases. Eventually, development of the diaphragm. The diaphragm is a mesodermal portion of composed origin, and it is developed from the following sources: anteromedial part, including the central tendon, vena cava, and esophageal openings. The part of the developed from the septum transversum, which is a mesodermal portion below the pericardial sac, extends from the ventral body wall and the ventral surface of the esophagus. During the head fold of the embryo, the septum migrates caudally from the second cervical to the lower border of the 12th thoracic vertebra, where the septum reaches the end of the second month in the fetal life. The opposite of the third, fourth, and seventh cervical segments in the septum is invaded by myotomes of those segments with their corresponding nerves, which subsequently unite in the phrenic nerve, posterior part between the esophageal and the aortic openings. This part is developed from the dorsal mesentery of the esophagus. Well, in each side, the dorsal mesentery of the side septum transverse triangular and gap exists clearly in fetal life. Diaphragm hernias. In rare occasions, foramen morgagni may present due to the failure of external origin of the diaphragm through the gap of abdominal visceral hernia of the thorax, or foramen of Morgagni. Abdominal viscera and the hernia of the thorax. Costovertebral trigone or the Baldac's triangle. Costovertebral trigone. Sometimes the diaphragm fails to arise from the lateral arcuate ligament on the both sides, forming a triangular gap known as the costovertebral trigone. A congenital diaphragmatic hernia takes place through the opening of the abdominal viscera hernia into the thorax due to the positive intra-abdominal pressure. Bogner's hernia is usually observed on the left side. Lateral arcuate ligament on the both sides. Hiatal hernia. Through the dilated esophageal sinus, it consists of two types, sliding and rolling. The sliding hernia is more common and takes place when the gastroesophageal junction of slides into two thorax through the lax hiatus, particularly in lying position. This is associated with irregulation of acid from the stomach and esophagus expressed in heartburn. In rolling hernia, the gastroesophageal junction remains normal. In rolling hernia, normal, and the hernial sac containing usually the fundus of the stomach projects into the thorax. Slide of esophagus, paraesophageal and hiatal hernia. The patient may complain of pain, nausea, and dysphagia. Morphology of the diaphragm. The complete thoracoabdominal diaphragm is found only in mammals. It is derived from the innermost muscle of the primitive body wall. This explains why the intercostal origin diaphragm integrate with the transverse abdominis. The mediastinum. The mediastinum is the space between the right and the left pleural sacs and limited on each side by the mediastinal pleura. It extends from the thoracic inlet above the diaphragm below and from the sternum in front of the 12th thoracic vertebra. The thoracic space between the 8th and the right and the left pleural sac and limited on each side by mediastinal pleura. It extends from the thoracic inlet above and diaphragm below and from the sternum in front of the 12th thoracic vertebra. For the purpose of description and the mediastinum, it is divided by an imaginary horizontal plate extending from the sternal angle to the lower border of the thoracic fork into the superior and inferior parts. Inferior mediastinum is subdivided further by the pericardium and heart into anterior, middle, and posterior mediastina. The middle mediastina is the widest subdivision. Angle to the border of thoracic fork into the superior inferior parts. The inferior mediastina is subdivided further by the pericardium and heart in the anterior, middle, and posterior mediastina. The middle mediastina is the widest subdivision. At two areas, the mediastinal contains no important structures and two pleural sacs are in close apposition behind the body of the sternal, extending from the sternal angle up to the fourth costal cartilage. Opposite thoracic 8 or thoracic 9 esophagus crosses in front of the descending aorta and in the mediastinal pleurae of both sects behind the body of the extending from the sternal angle to the fourth costal cartilage. Opposite thoracic 8 or thoracic 9 esophagus crosses in front of the descending aorta mediastinal plane of both sacs come in opposition mediastinal plane of both sacs come in opposition between them, forming a mesoesophagus. Most of the thoracic veins occupy the right part of the mediastinum. As the venous walls are often little resistance, a mediastinal growth bulges more on the right side and compression upon the right lung. pretracheal and paravertebral fossa extended from the neck into the superior mediastinum. Hence, a thyroid tumor or a retropharyngeal abscess may extend into the mediastinum. In lateral radiographs, the cardiac show intervenes between the retrosternal space for anterior mediastinum and the retrocardiac space for posterior mediastinum. In lateral radiograph, the cardiac shadow innervates between the retrosternal space for anterior mediastinal and retrocardiac space for posterior mediastinum. The cardiac shadow innervates between the retrosternal space for anterior mediastinum and the retrocardiac space for posterior mediastinum. Esophagus passes through the retrocardiac space and is visualized by Ballium swallow. The mediastinum is developed from the primitive ventral and dorsal mesoesophagus. Primitive ventral and dorsal mesoesophagus. Subdivisions and contents. Superior mediastinum boundaries. In front, mediastinum sternum. Behind, bodies of upper 4 thoracic vertebrae. Intervening disc and anterior longitudinal ligament. Above, thoracic inlet. Below, horizontal plane extending below the sternal angle and lower border of thoracic 4. On each side, mediastinal pleura. Contents. These are arranged into 3 groups: retrosternal, intermediate, and prevertebral. Retrosternal, intermediate, and prevertebral. Retrosternal structures. Origins of sternohyoid and sternothyroid muscles. Thymus. It is retrosternal structures. Origin of sternothyroid and sternohyoid muscles. Thymus gland. It is prominent after birth and rapidly diminishes after puberty. Thyroid gland prominent after birth and diminishes after puberty. It consists of two asymmetrical lateral lobes which overlap the upper part of the pericardium. Structurally, thymus consists of lobules, reticular fibers, and reticular cells. Structurally, thymus consists of lobules, reticular fibers, and reticular cells. Lymphocytes and lammated corpuscles. Hassel's corpuscles, lymphocyte, and lammated corpuscles. And right and left brachiocephalic veins, upper part of the superior vena cava and left superior intercostal vein. Intermediate structures. Arch of the aorta with its 3 large branches. Brachiocephalic, left common carotid, and left subclavian arteries. Intermediate structures. Phrenic, vagus, and cardiac nerves. Prevertebral structures. Superior mediastinum, retrosternal, intermediate, and prevertebral. Prevertebral structures. Trachea with its paratrachea and tracheobronchial lymph nodes. Esophagus, left recurrent laryngeal nerve passing upward along the tracheoesophageal groove. Thoracic duct passing upward behind and to the left of the esophagus, passing upward and left of the esophagus. Origin of the longus coeli muscle extending into the right thoracic vertebra. Anterior mediastinum. It innervates between the body of the sternum and the fibrous pericardium. Above the 4th costal cartilage, the mediastinum is narrow, but below the level it is somewhat broad and due to division on the left pleural sac. Above the 4th costal cartilage, the mediastinum is narrow, the 4th costal cartilage, but below that level it is somewhat broad due to the division of the left pleural sac. Inferior, superior, and inferior sternopericardial ligaments. Some loose areolar tissue and retroesternal lymph nodes. A few mediastinal branches and internal thoracic artery. Contents. Superior and inferior sternopericardial ligaments. Some loose areolar tissue and retroesternal lymph nodes. A few mediastinal branches of internal thoracic artery. Middle mediastinum. It is the widest subdivision, occupying mainly by the heart and the pericardium, and it is limited on each side by the mediastinal pleura. Contents. Pericardium encloses the heart. Ascending aorta, the pulmonary trunk, dividing into right and left pulmonary arteries. 4 pulmonary veins. Lower part of the superior vena cava, arch of the azygous vein. Bifurcation of trachea, right and left bronchi, deep cardiac plexus nerves and inferior trachea, bronchial lymph nodes, phrenic nerve and pericardio-phrenic vessels. Posterior mediastinum. It is the longest subdivision. Boundaries in front. From above downwards, bifurcation of trachea, pulmonary vessels, fibrous pericardium, the posterior slope of the surface of the diaphragm. Posterior mediastinum. Behind. Bodies of lower 8 thoracic vertebrae, vertebrae, intervertebral disc and interior longitudinal ligament. Above. Imaginary horizontal plane below the diaphragm on each side the mediastinal pleura. Contents. Longitudinal structure, esophagus, descending aorta, azygous and hemizygous vein, thoracic duct, vagus nerve, splanchnic nerves, posterior mediastinal lymph nodes. Transverse structure, termination of superior and inferior hemizygous veins, deviation of thoracic duct from right to left, posterior intercostal arteries, some of the posterior intercostal veins. Esophagus, trachea, superior vena cava, sternal angle, ascending aorta, pulmonary trunk, xiphoesternal joint, pericardium, the diaphragm, mesoesophagus, greater splanchnic nerve, lesser splanchnic nerve, thoracic duct, arch of the aorta, superior mediastinum, ganglion, sympathetic trunk, left lung root, posterior mediastinum, descending aorta, esophagus, least splanchnic nerve, thoracic mediastinum, left lateral viewer. Give the structure and functions of macrophages. Macrophages or histiocytes are phagocytic and are most numerous in loose connective tissue. They are distributed widely in different types of the body and belong to the mononuclear phagocytic system (MPS), which subserves an important purpose or defective mechanism in the body. Origin of macrophages. Macrophages come from specific white cells called monocytes. The monocytes are born from stem cells. Macrophages or histiocytes are phagocytes and are most numerous in loose connective tissue. Origin of macrophages. Macrophages come from specific white blood cells called monocytes. Monocytes are born from stem cells in the bone marrow and circulate throughout the blood stream. Once a monocyte leaves the blood, it matures into a wandering macrophage for a fixed macrophage. Wandering macrophages travel throughout the blood and lymph streams to perform their job. Fixed macrophages strategically concentrate in the specific areas like the lung and the intestine. Distribution of macrophages with different names. Connective tissue, macrophage, histiocyte. Lymphocytic organs, spleen, lymph nodes, bone marrow and thymus, macrophage. Skin, Langerhans cells, liver, Kupffer cells, lungs, alveolar and macrophages, kidney, mesenchymal cells, central nervous system, brain and spinal cord, microglia. Bone osteoblast. Structure and features of macrophages. Cell is relatively large, 10 to 30 micrometer in diameter. Surface is irregular with platelets, protrusions and indentations. Cytoplasm contains coarse granules or vacuoles in a light microscope. It has well-developed Golgi apparatus, many lysosomes and prominent troia. Nucleus is oval or kidney-shaped, located acentrically. Functions of macrophages. Phagocytosis. Macrophages act as a defense mechanism. They phagocyte cell decrease bacteria and abnormal extracellular matrix elements, neuroplastic cells and intern elements that penetrate the organism. Antigen presentation. particularly important for uptakes, processing and representing of antigens for lymphocyte activation, secrete enzymes, for example collagenase and actinokinase, and participate in defense and repair function. Name the cells of mononuclear phagocytotic system with their location. Mononuclear phagocytic system (MPS). It is a system which consists of phagocytic cells derived from monocyte and denote a population of antigen processing cells involved in the processing of foreign substance. Lifespan. All the long-living cells may survive for months in a tissue. Name. The macrophage-like cells have been given different names in the different organs, or Kupffer cells in the liver, macrophage cells in the central nervous system, Langerhans cells in the skin, and osteoclast in the bone tissue. Origin. All are derived from monocytes. Function. The main function of RPS are phagocytes secretion, antigen processing, and antigen presentation on other cells of immune system. All are derived from monocyte. The main functions of MPS are phagocytosis, secretions, antigen processing, and antigen presentation of other cells of immune system. Distribution and main function of cells of the mononuclear phagocytic system. Cell type, location, main function. Monocyte. Blood. Main functions: precursor of macrophages, macrophage, connective tissue, lymph organs, lungs, bone marrow, and peritoneal cavities. Main function: provides production of cytoskeleton, chemotoxin factors, and several other molecules that participate in inflammation. Antigen processing, presentation. Kupffer cell, liver, same as macrophages. Cell type: macroglia cell, central nervous system. Langerhans cell, epidermis of the skin. Dendritic cell, lymph node, spleen. Osteoclast, bone. Multinuclear giant cell, connective tissue. Mast cell. Mast cells are derived from bone marrow, precursor, and are characterized by abundant basophil cytoplasmic granules. Histologic features: shape: large, oval, and round. Diameter: 20 to 30 micrometer. Cytoplasm filled with basophilic secretory granules. Nuclear: small, peripheral, and nuclear and centrally situated. Potent of mast cell granules with their functions. Heparin, that act locally as an anticoagulant. Histamine, which promote increased vascularity, permeability, and smooth muscle contraction. And serine protease, which activate various medications and inflammations. Eosinophil, eosinophil, and neutrophilic chemotactic factors, which attract these nucleosides. Leukotrienes C, D, and E are show reacting substance of anaphylaxis, SRA, which also triggers with muscle contraction. Distribution of mast cell. Dermis of the skin. Mucosa living, digestive and respiratory tracts. Mucosal mast cells. Liver capsule along the blood vessels. Perivascular mast cell. Define concentration fibers. Name the fibers and connective tissue. Connective tissue fibers. The connective tissue fibers are formed by proteins that polymerize into elongated structures. These fibers provide strength to connective tissue. Types of connective tissue fiber. There are three main types of connective tissue fiber: collagen, reticular, and elastic fiber. Reticular and elastic fiber. Collagen and reticular fibers are both formed by protein collagens, and elastic fibers are composed of mainly the protein elastin. Name the connective tissue fibers, give the distribution and functions. Connective tissue fibers with their distribution. Type: optical properties, distribution, and main function. Type 1: thick, highly. Tendon, skin, tendon, bone. 2: loose aggregated fibers, cartilage, and various body. 3: thin, weakly birefringent, skin, muscle. 5: frequently forms facial tissue. 6: small fibers and cartilages, network forming collagen. Detected by Immunocytochemistry. All basal and external lamellae. 10. Detected by immunocytochemistry. Hypertrophic cartilage. And 7. Detected by immunocytochemistry. Epithelial basement membrane. Detected by immunocytochemistry. Cartilage. Varieties of body. And 11. Detected by immunocytochemistry. Placenta, skin, tendons. And then 14. Detected by placenta. Detected by immunocytochemistry. Placenta and bone. Distribution of elastic and reticular fibers. Plastic fibers. Walls of large arteries. Smooth muscle. Ligamentum nochae. Androneurium. Ligamentum flava. Hemopoietic organs. Trachea, bronchi and alveoli. And then vocal cord. Parenchyma organs such as liver. Write about collagen fibers. Collagen fibers. Collagen fibers are rough fibrous proteins that are thick and do not branch. They are the most abundant fibers, and they are found in most all of the connective tissue of our organs. Histological features. Each collagen fibers are rough fibrous proteins that thick and do not match. They are most abundant fibers. They are found in most of the connective tissue and organs. Histological features. Each fiber is composed of bundle of fibers collagen proteins into a number of fibrin substances known as tropocollagen. Histological features. Each tropocollagen consists of polypeptide chains called alpha chain. In electron microscope, the collagen fibers shown in transverse striations with alternated light and dark bands. Mechanical properties. Collagen fibers. Collagen fibers are inelastic and fixable and have high elastic strength. Distribution of main 4 types of collagen fibers. Type 1. Bone, tendon, ligament, dermis. Dentin of teeth, fascia and aponeurosis. Type 2. Collagen fiber. Type 3. Smooth muscle cells of cardiovascular gastrointestinal systems. Type 4. Collagen basement membrane. Enumerate the stages of collagen synthesis. The stages of collagen biosynthesis. Formation of mRNA, each type of alpha chain. Synthesis of procollagen and alpha chains. Properties at both ends. Hydroxylation and specific polymer and lystic residues. Attachment of soluble galactosides. Ensemble of procollagen molecules. Non-helical peptides. Transport of soluble protein into Golgi complex. Packing of soluble collagen in secondary vesicles. Secondary vesicles affected by macrotubules and microfilaments. Transport soluble procollagen molecules in cell substance. Exocytosis of procollagen molecules and extracellular space. Write the difference among different connective tissue fibers in a tabulated form. Traits. Appearance. Collagen fibers colorless, only demonstrated by yellow. Chemical composition. Formation, structure, distribution, consistency, flexibility, elasticity, tensile strength, excitability, transparency and boiling. Convert to gelatin. Write down the structure, location and function of loose connective tissue. Loose connective tissue. Loose connective tissue sometimes is called loose areolar tissue. Loose connective tissue is sometimes called the loose areolar tissue. Is a very common type of connective tissue that appears in those areas where considerable amount of mobility is required. It is characterized by loose, irregular management of connective tissue fibers and abundant ground substance. Characteristic features. Loose connective tissue has all the main components of connective tissue. The most numerous cells are fibroblasts and macrophages, but types of connective tissue cells are also present. Collagen, elastin, reticular fibers appear in the tissue with a moderate amount of ground substance in a flexible, well vascularized, not then stress distribution. Capillary layer of dermis, hypodermis in the lining of the peritoneal and pleural cavities, in glands, in the mucous membrane. Functions. They use support epithelial tissue. They are resistant to tension, resistance to transmitter pressure. They maintain the structure of expandable organs and give them support. They provide filtration. Short note matrix. Extracellular matrix material or matrix is a non-living material and is synthesized by connective tissue cells, particularly by fibroblast composition. It is composed of microamorphous elements or ground substance and fibrous elements. Ground substance is a highly hydrophilic viscous complex of ionic macromolecules, glycosaminoglycans and protoglycans, and multi-adhesive glycoproteins. They stabilize ECM by binding with receptors in the surface cells of the matrix components. Fibers provide strength to the tissue. They are of three types: collagen fiber, elastic fiber, reticular fiber. Ground substance. The ground substance is highly hydrophilic, viscous, semi-fluid material which occupies the internal between the cell and the fibers composition. It is formed mainly of these three types of components: glycosaminoglycans, hyaluronic acid, chondroitin sulfate, then darmatan sulfate, keratin sulfate, and hyperamic acid. Proteoglycans, multi-adhesive glycoproteins, laminar fibronectin, chromonectin, and osteorectin. Functions: provide morphological functions to framework and tissue. Short note: adipose tissue. Adipose tissue is characterized by connective tissue, which adipose tissue fat cells prominent type, white adipose tissue and brown adipose tissue. Multinuclear and single ring cell functions: provide a storehouse of fat, acts as cushion between many cells such as ischiorectal fats, helps to convert the body heat, play important role, give mechanical support. Fat is responsible to provide control of the human body distribution in subcutaneous tissue around the kidney, in the mesenteries of omentum, on bones and bone marrow, as localized in the synovial membrane. Define granular tissue. Granular tissue is a specialized type of tissue that is the hallmark of tissue repair called granules. And term granular tissue derives from the pink, soft, granular appearance of surface wounds. Characteristic histological features of granular tissue presents a new small blood vessels, proliferation of fibroblasts. The new vessels are leaky, allowing the passage of plasma proteins fluid into the vascular space. Thus, new granular tissue is edematous. What is antigenesis and vasculogenesis? Antigenesis is the physiological process which new blood vessels form pre-existing vessels. This is distinct from vasculogenesis, which is due to the novo formation of endothelial cells from mesodermal cell precursors. Regulation of erythropoiesis. The production of RBC cells is regulated to provide an adequate carrier capacity in blood. The hormone erythropoietin (EP) is the primary regulator of erythropoiesis and is released from the kidneys when O2 tension in the renal parenchyma is reduced. Committed erythroid stem cells in bone marrow and responsive to EP, which increases the rate of cell division and differentiation into mature red cells. Explain why polycythemia is produced when a person exceeds the high altitude hypoxia, stimulation of kidneys, production of erythropoietin, stimulation of bone marrow, production of RBC. Physiology: polycythemia of high altitude. Describe the morphology of RBC. Morphology of RBC: shape circular biconcave disc, diameter 2.8 micrometer, thickness 2.5 micrometer in thick point and 1 micrometer less in the center. Surface area 1.40 micrometer square. Volume 90 to 95 microcube. Non-nucleated. Each RBC contains 29 picogram of hemoglobin. What are the functions of RBC erythrocytes? Functions of RBC: respiratory functions. The hemoglobin of RBC transport O2 from the lungs to their tissues and CO2 from the tissues to the lungs. Acid-base balance. The hemoglobin of RBC acts as buffer and helps to maintain the acid-base balance. Maintenance of viscosity. RBC helps to maintain the viscosity of blood. If RBC is increased in the plasma polycythemia, the viscosity of blood will be increased. Production of pigments. Various pigments are produced from hemoglobin after the disintegration of RBC. For example, bilirubin, biliverdin, etc. Production of pigments. Various pigments are from RBC. After distinction of RBC, various pigments are produced. Determination of blood group. It contains antigen AB blood group antigen, Rh antigen, which determines the blood group. Ionic balance. RBC maintains the intracellular ionic balance by a special permeability of the membrane. Shape of RBC. Normal RBC cells are circular biconcave disc. Importance of biconcave shape. It allows considerable alteration of the cell volume without increasing the tension on the cell membrane. It can accommodate much fluid in hypotonic environment. Hemoglobin remains distributed in a very thin layer. This facilitates quick saturation or desaturation with the gases. Hemoglobin remains distributed in a very thin layer. This facilitates quick saturation or desaturation with the gases. It can easily squeeze when it passes through the capillary pores. It can easily squeeze when it passes through the capillary pores. Viva question. What is the advantage of absence of nucleus in RBC? The advantage of absence of nucleus in RBC. It gives the biconcavity of the RBC. It also gives more room for hemoglobin. Viva. Define the following terms related to abnormal forms of RBC. Abnormal RBC. Microcyte. When RBC are smaller than normal in size, they are called microcyte. When smaller than size, they are called microcyte. When RBC are larger than normal size, they are called macrocyte. Hypochromic. When the concentration of RHB per RBC is less than normal, the RBC are said to be hypochromic. When the variation in size of RBC from the normal size is when a variation in the size of RBC from the normal size is called anisocytosis. A variation in size of RBC from normal size is called anisocytosis. Poikilocytosis. A deviation from normal shape of RBC is called a deviation from normal shape of the RBC is called shape of RBC is called poikilocytosis. What are the variations in shape of red blood cell? Variation in shape of red blood cell. Shape of RBC is altered in many conditions, including different types of anemia, crenation, shrinkage as in hypertonic conditions, spherocytosis, globular form as in hypotonic conditions, shrinkage form in hypertonic conditions, crenation, spherocytosis, globular form in hypotonic conditions. Elliptocytosis, elliptical shape as in certain types of anemia. Elliptical shape as in a certain type of anemia. Sickle cell crescent shapes as in sickle cell anemia. Poikilocytosis, unusual shapes due to the deformation of cell membrane. The shape will be of flux, hammer, or other unusual shape. What is Howell-Jolly bodies? The certain types of anemia, some nuclear fragments are present in ectoplasm of the RBC. These nuclear fragments are called Howell-Jolly bodies. Define the following terms. Normocyte. RBC when normal in size is called normocyte. normochromic when the concentration of hemoglobin per RBC is normal. This is called RBC. It's called normochromic. And hematopoiesis. Normocyte, normochromic, and hematopoiesis. The process of the production of RBC under normal physiological condition is called hematopoiesis. Hematolysis. The process breakdown of hemolysis. What are the peculiarities of RBC? Erythromesis sedimentation rate. What is ESR? The factors influencing ESR. When blood is mixed with a suitable anticoagulant and is made and to stand vertically in a narrow tube, the red blood cells settle down to the bottom of the tube. Rate at the which occurs is called erythrocyte sedimentation rate. Methods of erythrocyte sedimentation rate: Western Greens method and Winthrop's method. Value: Western Greens method, male 6 millimeters in the first hour, female 12 millimeters in the first hour, 6 millimeter and 12 millimeter. And Winthrop's method 12 millimeter and 18 millimeter. Factors influencing ESR: cellular constitute of RBC, RBC count, RBC and vice versa. Protein constitute of blood, fibrinogen, globulin, Rolex formation and ESR. Albumin, low albumin equals to more ESR, more fibrinogen equals to increased ESR, more globulin equals to Rolex formation and increased ESR. So albumin এর বিপরীত। Plasma hemoglobin core, ESR increase. Specific gravity of blood, dilution of blood, increased ESR. Viscosity of blood, decreased ESR. Viscosity of blood, decreased ESR. Method of determination: ESR value is more in Western Trope method than Western Greens method. The length and the diameter of the tube, ESR, varies between Western Greens tube and PCV tube. Western Greens method is better because the length of the tube is more, so the rise of ESR is estimated. So higher rise of ESR is estimated. Western Green. Position of ESR tube: any degree of inclination of the ESR tube, decreased ESR. Duration of test: duration increases, ESR increases. Atmospheric temperature increases, ESR increases. Condition with increased or decreased ESR: conditions increase ESR, conditions with decreased ESR. Physiological, pathological. Conditions with ESR, conditions with decreased ESR, increased ESR. Physiological: menstruation, pregnancy and high atmospheric pressure. Pathological: multiple myeloma malignancy, myocardial infarction, acute rheumatic fever, collagen disease, rheumatoid arthritis, hemorrhage, chronic inflammation and tuberculosis, severe anemia. Pathological: in high altitude, in dehydration. Physiological: decreased ESR, high altitude, in dehydration, in defect in Rolex formation. Pathological: polycythemia vera. Pathological: polycythemia vera, hematochromatosis, sickle cell anemia, hepatic failure, fibrinogen formation, ESR. Congestive cardiac failure, fibrinogenemia. Cause of rise or marked sky rise ESR. Sky rise of ESR 100 millimeter or more in the first hour. Multiple myeloma, aplastic anemia, advanced malignancy, tuberculosis, TB, SLE, Kala Azar. Clinical importance of ESR: it has no diagnostic value. It is non-specific and supportive test. It is done to the progression of a disease. It is done to assess the progression of a disease. It is done for the follow-up of the disease and the treatment. Nice to know for a viewer. Normally, the red blood cells RBC remain suspended and uniformly in circulation. It is called suspension of stability of RBC. If blood is mixed with an anticoagulant and allowed to stand in a vertical tube, the red cells settle down due to gravity with a supernatant layer of clear plasma. ESR is also called sedimentation rate, SAD rate, or Bernanke reaction. It was first demonstrated by Edward Bernanke in 1897. Short note: Rolex formation definition: the process by which RBCs adhere one over another in piles is called Rolex formation. In Rolex formation, the RBC looks like a pile of coins. Cause of formation: it is cause unknown. It is believed to be some that the condition is a display of the surface tension that arises that causes them to be approximated in the one another opposite their larger surfaces while suspended in a fluid. Influencing factors: the Rolex formation increases when there is an increase of plasma fibrinogen and gamma globulin. The formation increases when there is increased plasma fibrinogen and gamma globulin. Importance: the tendency of RBC to settle down increase when they form Rolex. Thus, Rolex formation increases in ESR. Pronounced Rolex is Rolo, OS into. Hematopoiesis: synthesis of blood cells. Hematopoiesis or hematopoiesis is the process of origin, development, and maturation of blood cells. Blood forming cells may be divided into 4 groups depending on their capacity for self renewal, self division, and ability to form different cell types: pluripotent hematopoietic stem cells, multipotent hematopoietic stem cells, committed progenitor cells, maturation cells. Pluripotent hematopoietic stem cell, multipotent, pluripotent, hematopotent, committed and maturing. The process of blood cell formation therefore involves a sequence of cell division and differentiation. Erythropoiesis is the process of red cell formation. Thrombopoiesis is the process of platelet formation. And leukopoiesis is a general term described white blood cell production. Draw and diagram to show the stages of development of formed elements of red blood cell and bone marrow cell. The process of blood cell formation therefore involves sequence of cell division and differentiation. Erythropoiesis is the process of red cell formation. Draw a diagram to show stages of development of formed elements in blood from bone marrow and cell. Stages of development formed elements of blood from bone marrow and cell. Pluripotent hematopoietic stem cell. Pluripotent hematopoietic stem cell. Lymphoid stem cell. T lymphoid, B lymphoid. Colony forming unit. Colony forming unit B. Colony forming unit E, erythropoietin. Colony forming unit GM. Granulocytes, macrocytes, are megakaryocytes, platelets. Steps of genesis of neutrophil from a stem cell of red bone marrow. Pluripotent hematopoietic stem cell. Pluripotent hematopoietic stem cell. Colony forming unit S. Colony forming unit GM. Lymphoid cells, T lymphocyte, B lymphocyte. Myoblast and then myelocyte. Promyelocyte আর মনোসাইট থেকে ম্যাক্রোফেজ আর প্রোমায়েলোসাইট থেকে নিউট্রোফিল, ইউসিনোফিল, বেসোফিল, পলিমরফোনিউক্লিয়ার বেসোফিল, পলিমরফোনিউক্লিয়ার ইউসিনোফিল, পলিমরফোনিউক্লিয়ার নিউট্রোফিল. Red blood cells, erythropoiesis. Erythropoiesis is the process of the origin, development, and maturation of erythrocytes. Maturation of erythrocytes. Definition: erythropoiesis means the process of formation of erythrocytes under normal physiological conditions. It starts in the third week of intrauterine life. Sites of erythropoiesis. Intrauterine life from birth to 20 years. After 20 years, intrauterine life from third week to the third month. Mesoderm of the yolk sac. Mesoderm of the yolk sac. Mesoderm from third month to fifth month. Liver mainly. From fifth month to birth, red bone marrow. Red bone marrow. Red bone marrow, vertebrae, bones of the pelvis and upper end of the humerus and femur. Vertebra, sternum and pelvis. Relative rates of red blood cell production in bone marrow in different bones at different ages. What are the types of erythropoiesis? Types of erythropoiesis: mesoblastic erythropoiesis, hepatic erythropoiesis, and myeloid erythropoiesis. Types of erythropoiesis: mesoblastic erythropoiesis. From third week to third month of uterine life, primitive nucleated RBCs are produced intravascularly from mesoderm of the yolk sac. Mesoderm of yolk sac. This is called mesoblastic erythropoiesis. Hepatic erythropoiesis. From third to fifth month of the uterine life, nucleated RBC are produced extravascularly from the liver, spleen, and lymph nodes. This is called hepatic erythropoiesis. From third month to fifth month. Myeloid erythropoiesis. Myeloid erythropoiesis. From fifth month to the end to the month of pregnancy to delivery after birth, non-nucleated RBCs are produced extravascularly, intravascularly, and extravascularly. From the red bone marrow to the bones. From the red bone marrow of the bones. This is called myeloid erythropoiesis. What is medullary and extramedullary erythropoiesis? Medullary erythropoiesis. The erythropoiesis in the red marrow is called medullary erythropoiesis. Normal erythropoiesis in adult and later half of the intrauterine life is medullary erythropoiesis. Extramedullary erythropoiesis. The erythropoiesis in the site other than the red bone marrow, such as liver, spleen, lymph nodes, and mesoderm of the yolk sac, is called extramedullary erythropoiesis, where it occurs in fetus. It normally occurs in the mesoblastic and hepatic stages of erythroblastic. It normally occurs in mesoblastic and hepatoblastic stages of erythroblastic. In fetus, it normally occurs in the mesoblastic and hepatic stages of erythropoiesis. It normally occurs in mesoblastic and hepatic stages. Mesoblastic and hepato stages of erythropoiesis in fetus. In adults, it occurs in liver, spleen, and lymph nodes in disease in which the bone marrow becomes destroyed or fibrous. Stages are what are the stages steps of erythropoiesis? Developmental stages steps of erythropoiesis: pluripotent hematopoietic stem cell, committed stem cell, proerythroblast, basophil erythroblast, polychromatophil erythroblast, orthochromatic erythroblast, reticulocyte, and erythrocyte. Maturation characteristics of RBC classes that occur during RBC erythropoiesis. Maturation of the erythropoiesis involves a decrease in cell size, increased condensation, and finally pycnosis of the nucleus. Maturation of characteristics: decreased cell size, increased condensation, and finally pyrokinesis of the nucleus. Accumulation of hemoglobin, change in the staining reaction of the cytoplasm for basophil via polychromatophil to eosinophil. Briefly discuss the different steps of erythropoiesis. Stages: cell size, micro in diameter, nucleus, cytoplasm, hemoglobin, and mitosis. Stage 1: proerythroblast. Large, occupies 2/3 of the cell volume, nucleus 3/3 of the cell volume, contains several nuclei, contains chromosomes filled with strep reticulum. Basophilic, absent hemoglobin, presence only in the stage of the stress. Stage 2: basophil erythroblast. Nuclei disappears. Chromatin network is fine and shows very few nodes of condensation. Basophilic, begins to appear, active meiosis presence. Stage 3: further condensation of chromatin, polychromatic, increases, and active presence. Stage 4: orthochromatic erythroblast. Small pi kilo chromosome presence and cartwheel appearance. Orthochromatic erythroblast in presence in cartwheel appearance. Reticulocyte disappears and erythrocyte, nucleus disappears, nucleus absent. তো 15 থেকে 20 মাইক্রোমিটার থেকে এটা 3.5 মাইক্রোমিটার, মিন 7.3 মাইক্রোমিটার। basophilic polychromatic stage 3, and then eosinophilic. How can you differentiate between a mature and immature RBC? Difference between a mature and immature RBC: smaller, larger immature RBC. Biconcave, rounded, eosinophilic, basophilic. Present hemoglobin, present, absent. Mitosis, absent, maybe present. 120 days lifespan, immature 1 to 2 days. Eisman stain, supravital stain. Factors necessary for the development of RBC. Nutritional factors: essential amino acids, globin synthesis, iron, heme synthesis, vitamin B12, DNA synthesis. Essential globin synthesis, heme synthesis, vitamin B12, DNA synthesis, folic acid, DNA synthesis. Porphyrins, heme synthesis, pyridoxine, heme synthesis. Bile salt helps to absorb heavy metals. Trace elements, catalase reaction for Hb synthesis. Physiological stimulating factors: erythropoietin. It increases erythropoiesis by stimulating the difference of bone marrow stem cells. Chronic kidney disease: erythropoietin synthesis by kidney, erythropoiesis anemia. Erythropoiesis synthesis by kidney, erythropoiesis anemia. Hypoxia, high altitude hemorrhage. Chronic heart disease, anemia. Hypoxia, release of erythropoietin, erythropoiesis. Hormones: ACTH, TSH, GH, prolactin, adrenocortical hormones, thyroid hormones. Testosterone, EP production, erythropoietin, RBC, and HB value in man. Estrogen, erythropoietin, RBC, HB value in female. Indirect action factors: bile salt helps absorption of vitamins and trace elements. Intrinsic factor: still helps in absorption of B12. State the role of hypoxia and acute hemorrhage on erythropoiesis. How does erythropoiesis regulate erythropoiesis? Regulate erythropoiesis. Both hypoxia and acute hemorrhage stimulates erythropoiesis RBC production, but through different mechanisms. Hypoxia-induced erythropoiesis mechanism. Oxygen levels detected by kidney peritubular cells. Erythropoietin EPO secretion acts on bone marrow to stimulate RBC production. Hypoxia-inducible H factor stabilizes the AO2 activities, EPO gene transcription, effects, hemoglobin and hematocrit, high altitude polycythemia effects, 3P, 2P-BPG in RBC, run shift on O2 HB dissociation curve, better O2 delivery to tissues. Example, chronic lung disease or high altitude adaptation. Acute hemorrhage induced erythropoiesis mechanism, sudden blood loss, blood volume and O2 carrying capacity. Two-phase response, immediate minute hours, sympathetic activation, vasoconstriction and cardiac output. No immediate EPO release, takes 2 to 24 hours. Acute hemorrhage-induced erythropoiesis mechanism, sudden blood loss, blood volume and O2 carrying capacity. Two-phase response, immediate and delayed. Minute hours sympathetic action and no immune EPO release. Delayed hypoxia from blood loss EPO stimulates bone marrow and reticulocytes. Immature RBC in blood in 3 to 5 days. Effects. Restores RBC mass, takes 1 week for full recovery. Iron dependent, requires dependent iron storage. Hemorrhage depletes iron. Example, post-traumatic bleeding or surgical blood loss. High effects of high altitude on erythropoiesis. Primary response, hypoxia-driven erythropoiesis at high altitude, 2,500 meters. Hypoxia. Arterial PO2 stimulates kidney to release erythropoietin. Erythropoietin acts on bone marrow erythropoiesis RBC production. Key mechanism, hypoxia inducible factor, stabilized in low O2, activities in EPO gene transcription. What is the maturation factors for erythropoiesis? Maturation factors for erythropoiesis. The following special maturation substances necessary for the maturation of nucleated red cells. Vitamin B12, folic acid. So the important things are vitamin B12 and folic acid. Acts as enzyme on DNA synthesis, acts as catalyze on DNA synthesis. Coenzyme and catalyze. Alternative answers. Maturation factors are needed for the development of RBC for the stages subsequent to proteoblast that are iron, intrinsic factor of the Castle, vitamin B12, folic acid and vitamin C. Erythropoiesis definition. Site, stage with diagram, changes during erythropoiesis, factors affecting erythropoiesis. Discuss the erythropoiesis, mentioning definition, factors influence erythropoiesis, stages of erythropoiesis and changes following the erythropoiesis. Definition, factor affecting erythropoiesis, stages of erythropoiesis, changes following erythropoiesis. Erythropoiesis point to be included. Definition, steps of erythropoiesis, changes occur during erythropoiesis. Enumerate the factors that control it. Erythropoiesis definition. Please see above steps of erythropoiesis, changes during erythropoiesis, factors affecting erythropoiesis. Erythropoiesis definition, site of erythropoiesis and different stages of life, steps of erythropoiesis. Factors essential for erythropoiesis, changes in mature RBC. Erythropoiesis. Mention the role of erythropoietin in erythropoiesis. Definition. It is a circulatory glycoprotein hormone which is higher organism in the prime regulation of erythropoiesis is erythropoietin. It is a circulatory glycoprotein hormone which in higher organisms is the prime regulator for erythropoiesis. Glycoprotein hormone. Chemical structure. It is a glycoprotein containing protein 74%, carbohydrate 24%, sources kidney 85%, liver 15%. Regulation of regular secretion. The usual stimulus for EP secretion is hypoxia, but secretion of the hormone can also be stimulated by cobalt salts and hormone which act as androgens ACTH, TSH, GH, prolactin, adrenocortical hormones, and thyroid hormones. Role of erythropoietin. Erythropoietin increases the number of erythropoietin sensitive committed stem cells in the bone marrow that are converted to red blood cells, precursors, and subsequently to mature erythrocytes. Regulation of secretion. The usual stimulus for EP secretion is hypoxia, but secretion for hormone can also be stimulated by cobalt salts and hormones such as androgens ACTH, TSH, GH, prolactin, adrenocortical hormones, thyroid hormones. Rate in hypothyroidism. Erythropoietin increases the number of erythropoietin sensitive committed stem cells in the bone marrow that are converted to red cells, precursors, and subsequently to immature erythrocytes. Importance. When a person bleeds or becomes hypoxic, erythropoietin stimulates erythropoiesis and thus increases tissue generation. Hematopoietic stem cell, erythropoietin, proerythroblast, red blood cells. Tissue oxygenation, erythropoietin. What is Howell-Jolly bodies? Howell-Jolly is the certain type of anemia. Some nuclear fragments are present in the ectoplasm of the RBC. These nuclear fragments are called Howell-Jolly bodies. Define the following terms. RBC when normal in size is called normocyte. When the concentration of RBC per RBC is normal, the RBC is called the normochromic. Hematopoiesis, the process for processing RBC under alpha-physiological condition is called hematopoiesis. The process breakdown RBC is called hemolysis. What are the peculiarities of RBC? Absence of nucleus, absence of mitochondria and glycogen, absence of DNA, RNA, so synthesis of protein RBC is not possible. It contains hemoglobin. What are the causes of variation of RBC count? Physiological causes. Diurnal variation. The RBC count is the lowest in the morning and highest in the evening. The variation is about 4% of mean RBC content of blood per day. Lowest in the morning or highest in the evening. Height, altitude. In high altitudes, RBC count rises due to contract of spleen. Hypoxia stimulate erythropoiesis. Muscular exercise. RBC count increases during muscular exercise during the contraction of the spleen. High environmental temperature. It increases RBC count. High Andrometric pressure, it decreases RBC count. High biometric pressure, it decreases RBC count. High oxygen pressure, it decreases RBC count. Sex, the count is more in male and the female due to testosterone. Infant, during first few days the life have high RBC count. Emotional conditions, RBC count increases during the emotional conditions such as anxiety increases. It is cause of increase in sympathetic activity in case such as muscular exercise. Pathological causes, any condition leading to defective oxygen of blood raises RBC count such as emphysema, pneumothorax, pulmonary fibrosis, pulmonary tuberculosis, chronic and congenital heart diseases, chronic CO poisoning, arsenic, phosphorus and lead poisoning, polycythemia, any condition leading to hyperconcentration, burn, shock, cholera, severe diarrhea, severe vomiting. It's a increased RBC count. Conversely, hemoglobin will lead to fall RBC count, loss of blood, followed by liquid replacement other than any other fluid, leading to hemodilution. Consequently, hemodilution will lead to fall of RBC count, loss of blood, followed by liquid replacement other than any fluid may lead to blood loss, followed by liquid replacement other than any other fluid may cause hemodilution. Erythrocyte sedimentation rate, ESR. Sympathetic thoracic sympathetic trunk. The thoracic sympathetic trunk is a ganglionated chain situated either side on the vertebral column. Superiorly, it is continuous with the cervical sympathetic chain. At the thoracic inlet, and inferiorly it is with the lumbar sympathetic chain. Superiorly, cervical sympathetic chain, inferiorly lumbar sympathetic chain. After passing behind the medial arcuate ligament, after passing behind the median arcuate ligament of the diaphragm. The thoracic sympathetic chain begins at stellate ganglion. It begins at a stellate ganglion. Cervicothoracic ganglion, and descends in the thorax in the neck from the first rib head of the second to 10 ribs. Descends on the thorax in front of the neck of the first rib head of the second to the 10th ribs along the bodies of the thoracic 11 and the phylloid vertebra in front of the posterior intercostal nerve vessels along with the bodies of the nerve vessels. Thoracic 10 to thoracic 10 ribs along the bodies of the thoracic 11 and thoracic 12 vertebra. In posterior intercostal nerves and vessels. In the side of the body of the thoracic 12 vertebra, it passes behind the median arcuate ligament and continues with the lumbar sympathetic trunk. Median arcuate ligament and with the lumbar sympathetic trunk. Lumbar arcuate ligament in the median arcuate ligament and the sympathetic trunk. Ganglia. Initially, each thoracic sympathetic trunk has 12 ganglia. Laterally, each thoracic sympathetic trunk has 12 ganglia corresponding to 12 thoracic spinal nerves. Later, the first thoracic ganglion commonly fuses with the inferior cervical sympathetic ganglion. First thoracic ganglion fuses with the inferior cervical sympathetic ganglion. Inferior cervical sympathetic ganglion fuses with the inferior cervical sympathetic ganglion to form the cervicothoracic stellate ganglion. And the second ganglion also occasionally fuse with the first ganglion. The first ganglion occasionally fuses with the inferior cervical sympathetic ganglion. cervical sympathetic ganglion, inferior cervical sympathetic ganglion. The second ganglion may also occasionally be with the first ganglion. There are usually 11 ganglion in the thoracic erection, sometimes maybe only 10 ganglion. Right supra. Each ganglion lies at the level of the corresponding vertebral disc, and it's connected to the corresponding spinal nerve with the white and the gray ramus communicants. Branches. The branches of the thoracic sympathetic trunk are divided into two groups: medial branches and lateral branches. Medial branches, the branches of the sympathetic trunk. Medial branches. The medial branches supply viscera. They are as follows: consist of postganglionic fibers and distributed to the heart, great vessels, lung, esophagus through the following plexus: pulmonary plexus, cardiac plexus, aortic plexus, esophageal plexus. Cardiac plexus, esophageal plexus, pulmonary plexus, aortic plexus. Medial branch supply to the viscera. Medial branch. The medial branch from the 5th to 12th thoracic ganglion consists of preganglionic fibers, and from the 3 splanchnic nerves. The medial branches from the 5th to 12th thoracic ganglion consist of preganglionic fibers, and from the 3 splanchnic nerves: greater splanchnic nerve, lesser splanchnic nerve, least splanchnic nerve. Medial branches consist of branches from the ganglia and medial branches from the 1st to 5th, followed by consisting postganglionic fibers distributed to the heart vessels, the 1st to 5th. Heart vessels and lungs, 1st to 5th, and the 5th to 12th postganglion consist of preganglionic and 3 symmetric nerves. Greater splanchnic nerve. It is formed by the preganglionic fibers arising from the 5th to the 9th. It is formed by the preganglionic fibers arising from the 1st to 9th thoracic sympathetic ganglia. It descends obliquely on the vertebral column and pierces the corresponding tooth of the diaphragm and terminates in the celiac ganglion. Part 3 also terminates in the arterial ganglion and suprarenal gland. It provides sympathetic supply to foregut and adrenal medulla. Lesser sympathetic tract. It is formed by the preganglionic fibers from the 10 to the 11 ganglia. So let's see 5th to 10. 5th to 10 corresponding, formed by the 5th to 9th ganglia. The greater is 5th to 9th, and the lesser is 10 and 11. Mesenteric ganglion. Source of the diaphragm. Similar to the great nerve, pierces the corresponding tooth of the diaphragm and terminates in the superior mesenteric ganglion. And provides sympathetic supply to the midbrain. At least, it is also called the renal nerve. This tiny nerve arises from the 12th thoracic ganglion. May be absent. It descends obliquely at the greater and the lesser splenic nerves, pierces either the crus and or passes behind the medial arcuate ligament, passes behind the medial arcuate ligament, diaphragm, and terminates into renal plexus. It provides sympathetic supply to the kidneys. Further details about the thoracic splanchnic nerves, medial branches and splanchnic nerves. The ganglia is divided into the median branches and splanchnic nerves. The median branches are from 1st to 5th. Branches of the medial branches, they supply to the viscera, the heart, the esophagus, the pericardium, the heart, the great vessels, the nerve esophagus, the heart, the esophagus, the great vessels, and the lung. Pulmonary plexus, cardiac plexus, aortic plexus, esophageal plexus, and the greater, and then The splanchnic nerve categorized into three different parts: greater, lesser, and least. The greater is consists of the fifth, sixth, seventh, eighth, and ninth thoracic sympathetic ganglia. Thoracic sympathetic ganglia. It clears the right and the left pura of the diaphragm, and the greater lesser is the fourth level, and the least is the renal nerve. Renal nerve is tiny nerve, and is sometimes be absent. Further details about thoracic sympathetic nerves are as follows. Their nerves provide sympathetic supply to the organs of the abdomen. Their fibers originate from the thoracic spinal segment, from thoracic 5 to thoracic 10. They travel through the white rami communicants and pass through the sympathetic ganglia without synapsing, and join together to form a splanchnic nerve. Pass behind the kidney. These are further details about thoracic splanchnic nerve. These nerves provide sympathetic supply to the organs of the abdomen. Their fibers originate from the thoracic spinal segment, from thoracic 5 to thoracic 10, and originate from the thoracic spinal segment, from thoracic 5 to thoracic 10. Travel through white rami communicants and pass through sympathetic ganglia. Travel through white rami communicants and pass through white rami communicants and sympathetic ganglia without synapsing, and joined together from the splanchnic nerve. Thus, they are made out of preganglionic fibers which synapse with the cilia superior mesenteric, and thus they form of preganglionic fibers which synapse with the cilia superior mesenteric and arteriolar ganglion. From them, posterior ganglion fibers arise and supply the abdominopelvic organs. There are sets of paired of thoracic splanchnic nerves: greater splanchnic nerve, lesser splanchnic nerve, least splanchnic nerve. They are described in detail earlier. Lateral branches. So all of these were medial branches and now lateral branches. Lateral branches supply limbs of the body wall. Their supply is pilomotor, pseudomotor. Pseudomotor, and vasomotor. Pilomotor, pseudomotor, and vasomotor to the skin of these regions. The preganglionic fibers arise from the lateral horns of the spinal segment and enter into sympathetic ganglion. The preganglionic fibers arise from the lateral horn of the spinal segment and enter into sympathetic ganglion via white rami communicants of the spinal nerve. The postganglionic fibers from the ganglion enter into the spinal lump rami communicants to supply the corresponding dermoid upper limb of the body wall. So medial branches and lateral branches. Lumbar splanchnic nerves. They arise from the lumbar part of the sympathetic chain. The nerve fibers arising from the synapses in the inferior. They arise from the lumbar part of the sympathetic chain. So arise from the lumbar part of the sympathetic chain. The fibers arising from the tongue synapse in inferior mesenteric ganglion. The posterior fibers arising from the ganglion supply the smooth muscle and gland of the pelvic. Posterior fibers ganglion, the smooth muscles of the pelvic viscera. Correlation: thoracoabdominal sympathectomy. Thoracoabdominal sympathectomy is a bilateral thoracoabdominal sympathectomy done is a bilateral thoracoabdominal sympathectomy is done to relieve the severe hypertension. The surgical procedure involves the removal of the sympathetic trunk from T5 to L to T5 to thoracic 5 to lumbar 2 ganglia. Exertion of sympathetic nerves. As a result, there occurs splanchnic vasodilation and consequent fall in blood pressure. For sympathetic splanchnic vasodilation, result it causes sympathetic vasodilation. The upper limb sympathectomy, the thoracoabdominal sympathectomy, the upper limb sympathectomy is used to treat the Raynaud's disease. In this part of the thoracic sympathetic chain is excited below the level of the stellate ganglion. It is a clinical condition where the nerves for the sympathetic ganglion. It is a clinical condition where the sympathetic nerve below the stellate ganglion is excited to treat the Raynaud's disease. Raynaud's disease. Raynaud's disease. Injury in the satellite region may cause bilateral Horner syndrome. Hypertension during spinal anesthesia. Sometimes hypertension occurs due to hypotension due to anesthesia, to the paralysis of sympathetic outflow and splanchnic nerves. Referred pain of the diaphragm. The irritation of the diaphragm secondary to peritonitis causes pain due to the stimulation of the phrenic nerve. Pain from cervical 3 to cervical 5. This pain is referred to the corresponding tip of the shoulder. The irritation of the diaphragm secondary to peritonitis causes pain due to the stimulation of the phrenic nerve. Root nerve of cervical 3 to cervical 5. The pain is referred to the corresponding tip of the shoulder, being supplied by the suprasubclavian nerve. Peritonitis. Note well, the thoracic duct begins in the abdomen, courses through the thorax, and terminates in the neck. Relations. The relations of the thoracic ducts are as follows. At the aortic orifice of the diaphragm, anterior, median arcuate ligament of the diaphragm, posterior, thoracic 12 vertebra. To the right, aegis vein. To the left, aorta. In the posterior mediastinum anterior. In the posterior mediastinum anterior, diaphragm, ascending aorta, esophagus, posterior, vertebral column, anterior longitudinal ligament, terminal parts of the hemizygous and accessory hemiazygous vein, right posterior intercostal artery. To the right, aegis vein. To the left, descending thoracic aorta. In the root of the neck, carotid sheath containing the left common carotid artery, the left interjugular vein, and the left vagus nerve, posterior, vertebral artery and vein, scalenus anterior muscle, medial border, phrenic nerve, thoracocervical trunk and its branches, suprascapular, transverse clavicle, and anterior thyroid arteries. Tributaries. The tributaries of the thoracic duct are as follows. In the thorax, a pair of descending lymph trunks from the posterior intercostal lymph nodes of the lower 6 bases. Lymph vessels from the posterior mediastinal lymph nodes. Ascending lumbar lymph tracts draining upper local aortic lymph nodes. These are bilateral and pierce the corresponding crus of diaphragm and eventually join the thoracic duct. In the posterior mediastinum. In the neck, left jugular lymph tract draining lymph from the left half of the head and neck. Left subclavian lymph tract draining lymph from the left upper limb. The left bronchiomediastinal trunk. Clinical correlation. Injury of the thoracic duct. The thoracic duct is a thin walled and appears colorless during surgery. Therefore, it is sometimes injured inadvertently during surgical procedures in the posterior mediastinum. Laceration of the thoracic duct during the lung surgery leads to leakage of chyle into the pleural cavity, producing a clinical condition called chylothorax. The cervical part of the thorax duct may be damaged during the block dissection of the neck. It should be ligated immediately. If ligated, the lymph results by anastomotic channels, but if the injury is not detected at the time of operation and hence not ligated, it may cause an unpleasant chyle fistula and the leakage of lymph. Immediate ligation of the duct is required to stop the leakage. Obstructions of the thoracic duct, sometimes in filarial infection. The thoracic duct is obstructed by microfibrial apertures, which are near Bancroft, leading to whispered effects such as chylothorax, chyloperitoneum, chyluria, and even the accumulation of chyle in the tunica vaginalis of testis, chylocele. Development. There are three stages in the development of thoracic duct. Stage 1. In this stage, the network of lymph channels is seen in front of the thoracic part of the vertebral column. Stage 2. In this stage, the two longitudinal lymph channels appear in the network of lymph channels, one on the left, another on the right, within a number of cross communications. Stage 3. In this stage, the cross communication appears opposite to thoracic 5. Right longitudinal channels below these cross communications and left longitudinal channels above this cross communication persist for the thoracic duct. All other parts disappear. Azygous and hemizygous vein. The term azygous means single, and it is without a companion. A zygous system of veins consisting of azygous, hemiazygous, and accessory hemiazygous vein. These veins lie in front of the thoracic part of the vertebral column and play an important role in the venous drainage of the thorax. Azygous vein. The zygous vein is present on the right side of the thoracic vertebral column in the posterior mediastinum. It consists the inferior vena cava with the superior vena cava. It is provided with valves and cells halfway along the azygous arch between the vertebral azygous vein and the point where the azygous vein enters the SBC. This part may appear tortuous. The functions of the azygous vein are as follows. It drains the venous blood from the upper part of the posterior abdominal wall and thoracic wall. It consists of SVC and IVC. Thus, it forms an important collateral channel connecting the SVC and IVC. Formation. The formation of azygous vein is variable. It is formed in one of the following ways. Formed by the union of right subcostal and right ascending lumbar vein at the level of the thoracic dual vertebra. Arises from the posterior inferior vena cava near the renal veins as a continuation of the right subcostal vein. Occasionally, it may arise from the right renal or right first lumbar vein. Course and terminations. The azygous vein after formation ascends up and leaves the abdominal by passing through the aortic opening of the diaphragm and enters the posterior mediastinum. There it ascends vertically, lying in front of the vertebral column up to the level of the thoracic vertebra, where it arches forward above the hilum of the right lung to terminate in the superior vena cava at the lower end of the second costal cartilage. Relations: anterior, esophagus, posterior, lower 8 thoracic vertebra, lower right posterior intercostal arteries, lower 8 thoracic vertebra, right posterior intercostal arteries. The right lung and pleura, great splanchnic nerves. To the left, thoracic duct, descending thoracic aorta, esophagus, right border. The arch of the azygous vein is related below the root of the right lung. On the right side to the right lung and the pleura, and the left side to the left border of the esophagus, trachea, and right vagus nerve. Tributaries. The tributaries of the esophagus vein are as follows. Right superior intercostal vein formed by the union of the second, third, and the fourth posterior intercostal veins. 5th to 11th right posterior intercostal veins, hemizygous vein at the level of the lower part of the thoracic 8 vertebra. Accessory hemizygous vein at the level of the upper border of the thoracic 8 vertebra. Right subcostal vein. Right ascending lumbar vein. Right brachial vein near the terminal end of the zygous vein. Esophagus veins with the exception of those of its lower end. Mediastinal veins, pericardial veins. Clinical correlation. In the case of obstruction of SVC, superior vena cava, the zygous vein serves as the main collateral channel to shut the blood from the upper half of the head to IVC, inferior vena cava. Hemizygous vein. The hemizygous vein lies on the left side of the thoracic vertebral column and corresponds to the lower part of the azygous vein, mirror image of the lower part of the azygous vein. Formation. The hemizygous vein formed on the left, similar to the azygous vein, by the union of left accessory lumbar vein and the left subcostal vein. It may arise from the posterior surface of the left renal vein. Tributaries. The tributaries of the hemizygous vein are as follows. The lower 3, 8 to 9 to 11th posterior intercostal veins, left subclavian vein, left ascending lumbar vein, small esophagus and medial mediastinal veins. Accessory hemizygous vein. The accessory hemizygous vein, superior hemizygous vein, lies on the left side thoracic vertebral column and corresponds to the upper part of the zygous vein, mirror image of the upper part of the zygous vein. Courses and terminations. The accessory hemizygous vein begins at the medial end of the left, fourth, and fifth intercostal space and descends on the left side of the vertebral column. At the level of the thoracic 8 vertebra, it turns to the right, passes in front of the vertebral column, posterior to the aorta, esophagus, and the thoracic that remains the zygous vein. Note well, sometimes the terminal parts of the hemizygous and accessory hemizygous vein joins together to form a common trunk that crosses across the vertebral column to open into the azygous vein. Tributaries. The following are the tributaries of the accessory hemizygous vein. 5th to 8th left posterior intercostal vein. 5th to 8th left posterior intercostal vein, left bronchial veins. Thoracic sympathetic trunks. The thoracic sympathetic trunk is a ganglionated chain situated on either side of the vertebral column. Superiorly, it is continuous with the cervical sympathetic chain at the thoracic inlet and inferiorly with the lumbar sympathetic chain after passing behind the medial arcuate ligament of the diaphragm. Courses and relations. The thoracic sympathetic chain begins at the stele ganglion, cervicothoracic ganglion, and descends in the thorax in front of the neck of the first rib, head of the 8th to 10th rib, along the bodies of the thoracic 11 to thoracic 12 vertebra, in front of the posterior intercostal nerves and vessels. On the side of the body of the thoracic 12 vertebra, it passes behind the medial arcuate ligament and becomes continuous with the lumbar sympathetic chain. Ganglia. Initially, the thoracic sympathetic trunk has 12 ganglia corresponding to the 12 thoracic spinal nerves. Later, the first thoracic ganglion commonly fuses with the inferior cervical sympathetic ganglion to form the cervicothoracic stele ganglion. The second ganglion also may occasionally fuse with the first ganglion. Thus, there are usually 11 ganglion in the thoracic sympathetic trunk. Sometimes there may be 10 ganglion, and each ganglion lies at the end of the corresponding vertebral disc, and it's connected to the corresponding spinal nerves by the white and the gray ramus communicans branches. The branches of the thoracic sympathetic trunks are divided into two groups, medial and lateral. Median branches. The median branches supply the viscera. They are as follows. The first branches from the 1st to 5th Ganglion consist of posterior ganglionic fibers and are distributed to the heart, blood vessels, lungs, and esophagus through the following plexus: pulmonary plexus, cardiac plexus, aortic plexus, esophageal plexus. The medial branches of the 5th to 12th thoracic ganglion consist of preganglionic fibers from the three splanchnic nerves, greater splanchnic nerves. It is formed by the preganglionic fibers arising from the 5th to 9th thoracic sympathetic ganglion. It descends obliquely on the vertebral bodies, pierces the corresponding grooves of the diaphragm, and terminates mainly in the celiac region, partly into terminate into aortorenal ganglion and the suprarenal ganglion. It provides sympathetic supply to the foregut and adrenal medulla. Lesser splanchnic nerves. It is formed by the preganglionic fibers from the 10 to 11 ganglia. Its course is obliquely similar to the great splanchnic nerve, pierces the corresponding grooves of the diaphragm and terminates in the superior mesenteric ganglion. It provides sympathetic supply to the hindgut. Less sympathetic nerve. It is called the renal nerve. This tiny nerve arises from the 12th thoracic ganglia, may even be absent. It descends obliquely as a greater and lesser splanchnic nerves, pierces either on the grooves and passes behind the medial arcuate ligament of the diaphragm and terminates at the renal plexus of ganglions. It provides sympathetic supply to the kidneys. Further details about sympathetic supply nerves. These are as follows. There are nerves provided sympathetic supply to the origin of the abdomen. Their fibers originate from the thoracic abdomen of thoracic 5 to thoracic 12 and travel through the white rami communicates and pass through the sympathetic ganglion without synapsing and join together to form a sympathetic nerve. Thus, they are made out of the preganglionic fibers, which synapse with cilia, superior mesenteric, and aortic ganglia. From them, posterior ganglion fibers arise and supply abdominal pelvic organs. There are three sets of air thoracic sympathetic nerves: greater sympathetic nerves, greater splanchnic nerves, lesser splanchnic nerves, least splanchnic nerves. They are already described in the detail earlier. Later branches. The lateral branches supply limbs of the body of the wall. The supply is spinomotor, submotor, and vasomotor on the skin of the region. The preganglionic fibers arise from the lateral horns of the spinal segment and enter into the sympathetic ganglion via white rami communicants of the spinal nerves. The postganglionic fibers from the ganglion re-enter the spinal nerve via gray rami communicants and supply the corresponding dermatom of the upper limb of the body of the wall. Lumbar splanchnic nerves. They arise from the lumbar part of the sympathetic trunk. The nerve fibers arising from the tongue synapses in the inferior mesenteric ganglion. The postsynaptic fibers arising from the ganglion supplied smooth muscle and the glands to the pelvic viscera. Clinical correlation. Thoraco-abdominal sympathectomy. The bilateral thoraco-lumbar thoraco-abdominal sympathectomy is done to reveal severe hypertension. The surgical procedure involves the removal of sympathetic trunk from thoracic 5 to lumbar 2 ganglion and excision of nerves. As a result, there occurs splanchnic vasodilation and consequent fall in blood pressure. The upper limb sympathectomy is used to treat the Raynaud's disease. In this case, part of the thoracic sympathetic chain is excited below the level of the sterile ganglion. Note well, injury to sterile ganglion may cause ipsilateral horns of the syndrome. Hypertension during spinal anesthesia. Sometimes hypertension occurs due to high spinal anesthesia due to the paralysis of sympathetic outflow and sympathetic nerves. Referred pain of the diaphragm. The irritation of the diaphragm secondary to priorities causes pain due to the stimulation of the phrenic nerves, root of the cervical 6, cervical 5. The pain is referred in the corresponding tip of the shoulder, begins supplied by the supraclavicular nerve. What is chylous accumulation of chyle in the tunica vaginalis around the testis. Hospital complaining of swelling in his scrotum associated with precardiatic fever and passage of milky urine. In examination, the doctor found out that the scrotum hole was enlarged. The scrotum was thick, mainly to the bottom. The aspiration of scrotum, its milky fluid came out. It was diagnosed as a case of Child-Pillson and Curella. What causes chylocera? When the thoracic duct is obstructed by microfibria, it leads to widespread effect which chylothorax, chyloperitoneum and chyluria. What is the cause of the thickened of the scrotum skin? Lymphostasis. Largest lymphatic channel in the body, thoracic duct. Chylothorax, accumulation of chyle in the pleural cavity. Chyle, accumulation of chyle in the tunica vaginalis. Chyluria, presence of chyle in the urine. Renal nerve, least splanchnic nerve. Commonest cause of damage of thoracic duct, the root of the neck. Blocked disease of the neck. Largest collateral channel connecting superior inferior vena cava, azygous vein. Largest sympathetic nerve, great splanchnic nerve. Most posterior intercostal veins drain into azygous venous system. Diaphragm of inferior thoracic aperture. The thoracic outlet is closed by a large dome-shaped flat muscle called diaphragm. Since it separates thoracic cavity from abdominal cavity, it is also termed thoraco-abdominal diaphragm. The diaphragm is the principal muscle of respiration. It is dome-shaped and consists of peripheral mucosal part and central fibrous part called central tendon. Origin. The origin of the diaphragm is divided into three parts. They are sternal, costal and vertebral. Sternal part. It consists of two fleshy slits which arise from the posterior surface of the xiphoid process. Costal part. On each side it consists of 6 fleshy slits which arise from the inner surface of the lower 6 ribs near their costal cartilage. Vertebral part. The part arises by means of right and left crura of diaphragm. Right and left crura of diaphragm. 5 arcuate ligaments. 5 arcuate ligaments. Vertebral part arises by means of 1 right and left crural diaphragm and 2 5 arcuate ligaments. Crura, right crurals. It is a vertical fleshy bundle which arises from the right side of the anterior aspect of the upper 3 lumbar vertebrae and intervening intervertebral discs. Left crurus. It is vertebral fleshy bundle which arises from the left side of the anterior aspect of upper 2 lumbar vertebrae and the intervening intervertebral disc. The medial margin of the crura are tendinous. The middle margin of the crura are tendinous. Arcuate ligaments. Median arcuate ligament is an arched fibrous band stretching between the upper ends of the 2 crura. Median arcuate ligament is the thick end upper margin of the psoas sheath. It extends from the side of the body of lumbar 2 vertebra to the tip of the transverse process of lumbar 1 vertebra. Lateral arcuate ligament is the thickened upper margin of fascia covering the anterior surface of the quadratus lumborum. It extends from the tip of the transverse process of lumbar 1 vertebra to the 12th vertebra. Arcuate ligaments. Medial arcuate ligament. and lateral arcuate ligament. Note well, medial arcuate ligament is the arched fibrous band stretching between the upper ends of the two crura. The fibrous band is stretching between the upper ends of the two crura. Medial arcuate ligament is the thicker upper margin of the psoas sheath. Medial arcuate ligament is the thicker. It extends from the side of the body from lumbar 2 vertebra to the tip of the transverse process of lumbar 1 vertebra. Lateral arcuate ligament is the thickest upper margin of the fascia covering the anterior surface of the quadratus lumborum. It is the thicker upper margin of the fibrous covering the anterior surface of the quadratus lumborum. It extends from the tip of the transverse process, lumbar 1 vertebra and the 12th vertebral rib. Note well, the right crus is attached to the more number of vertebra because the right side of the diaphragm has to contact with the massive liver, has to contract on the massive liver. Insertion from circumferential origin, white supra. The muscle fiber converges towards the central tendon and inserts into its margins. Central tendon and inserts into its margins. The features of the central tendon are as follows. It is trefoilite in shape, having a single triangular anterior central inflet or two B and C triangle shaped posterior leaflets. It resembles an equilateral triangle. Insertion from circumferential origin. The muscle fibers converge towards the central tendon and insert into its margins. The circumferential origin. The muscle fibers converge towards the tendon and insert into the margins. The features of the central tendon are as follows. It is trifold, the insertions. The muscle fibers covering towards the central tendon inserts into the margins. The features of the central tendon are as follows. It is trifoliate in shape, singular triangular arterial central leaflet and two tongue-shaped posterior leaflets. It resembles an equilateral triangle. The right posterior leaflet is short and stout, whereas the left posterior leaflet is thin and long. It is inseparably fused with the fibrous pericardium. It is located nearer the sternum than to the vertebral column. The surfaces and relations. The superior surface of diaphragm projects on either side as dome or cupolar into the thoracic cavity. Depressed area between the two domes is called central tendon. The superior surface is covered by endothoracic fascia and is related to the bases of right and left pleura on the sides and to the fibrous pericardium on the middle. The superior surface of the diaphragm projects on either side as a dome or cupolar into the thoracic cavity. Depressed area between the two domes called central tendon. The superior surface is covered by the endothoracic fascia and is related to the bases of right and left pleura on side and the fibrous pericardium in the middle. The inferior surface of diaphragm is lined by the diaphragmatic fossa and peritoneum. On the right side is related to the right lobe of the liver, right kidney and right suprarenal gland. On the left side is related to the left lobe, left liver, fundus of the stomach, spleen, left kidney and left suprarenal gland. Opening of the diaphragm. The openings of diaphragm are classified into two types, major openings and minor openings. Major openings. There are three named major openings: vena cava opening, esophageal opening and aorta opening. The location, shape and the vertebral levels of these openings are present in table 15.2. The structure passing through three major openings of diaphragm is listed on table 15.3. Note well. Effect of contraction of diaphragm on the three major openings. Contraction of diaphragm enlarges the caval opening to enhance venous return. The contraction of the diaphragm enlarges the caval opening to enhance the venous return. Contraction of the diaphragm has a sphincteric effect on the esophageal opening. Hitchcock effect. Contraction of the diaphragm has a sphincteric effect on the esophageal opening. Contraction of diaphragm has no effect on the aortic opening because, strictly speaking, it is outside the diaphragm. Minor openings. These are unnamed structures passing through these openings are as follows. Superior epigastric vessels pass through the gap space of Larry. Superior epigastric space of Larry between the muscular slip arising from the xiphoid process and the seventh costal cartilage. Location and the shape and the vertebral levels and the major openings of the diaphragm. Openings. Vena cava opening. It is the central tendon slightly to the right of that median plane between the central and the right posterior leaflets. Shape. Quadrangular or square. Thoracic 8 vertebra. Esophageal opening slightly to the left in central tendon slightly to the left on the medial plane. The fibers of the right crural split around the opening and act like pinched cork. Oval or elliptical. Aortic opening in the middle behind the median articulate ligament. Circular or round. Thoracic 12. Lower border of the body. Structures passing through the three major openings of diaphragm. Vena cava opening. Superior vena cava, right phrenic nerve. Esophageal opening. Esophagus, right and left vagal trunks. Esophageal and esophageal branches of left gastric artery. Or aortic opening from the right to the left. We see azygous vein, thoracic duct and aorta. Muscular. So the minor opening. Superior epigastric vessel. Musculophrenic artery passes through the gap between the slip of origin of the 7th and the 8th ribs. Passes through the gap between the slips origin from the 7th to the 8th ribs. Lower 5 intercostal nerves and vessels pass through gaps between the adjoining costal slips. Subcostal nerves and vessels pass deep to the lateral articulate ligament. Lateral arcuate ligament. Sympathetic chain passes deep into the medial arcuate ligament. And greater, lesser and less sympathetic nerves pass by the piercing and the crus of diaphragm on the corresponding side. Hemizygous vein pierces the left crus of the diaphragm. Nerve supply. The diaphragm is supplied by right and left phrenic nerves. Lower from intercostal and subcostal nerves. Motor supply by right and left phrenic nerves. Sensory supply central part by right and left phrenic nerves. Peripheral part by lower 6 to 10 intercostal nerves. The phrenic nerves are formed one on either side on the neck from ventral rami of cervical 6 to cervical 5. It lies on the anterior aspect of the scalenus anterior, then ascends vertically deep to cervical vein into thorax on the side of the pericardium to provide in all important motor supply to diaphragm, that key respiratory muscle. The detailed distribution is as follows. The right phrenic nerves provides motor innervation to the right half of the diaphragm up to the right margin of the esophageal opening. The left phrenic nerve provides motor innervation to the left half of the diaphragm up to the left margin of the esophageal opening. The phrenic nerves also provide sensory innervation to the central tendon of the diaphragm and pleura and peritoneum related to it. The intercostal nerves supply the peripheral part of the diaphragm. Arterial supply. The diaphragm is supplied by the following arteries. Superior phrenic arteries, also called phrenic arteries, from thoracic aorta. Inferior phrenic arteries from abdominal aorta. Pericardiophonic arteries from the internal thoracic artery. Musculophrenic arteries, the terminal branches of the interthoracic arteries. Superior epigastric arteries, the terminal branches of the interthoracic arteries. Lower 5 intercostal and subcostal arteries from the aorta. Lymphatic drainage. The lymph from diaphragm is drained into the following groups of lymph nodes. Anterior diaphragmatic lymph node, situated behind the xiphoid process. Posterior diaphragmatic lymph node, situated near the arterial orifice. Right lateral diaphragmatic node, situated near the caval opening. Left lateral diaphragmatic node, situated near the esophageal opening. Actions of diaphragm. The diaphragm acts to subserve the following functions. Muscle of inspiration. The diaphragm is the main principal muscle of inspiration. When it contracts, it descends and increases the ventral diameter of the thoracic cavity. Muscle of abdominal straining. The contraction of diaphragm along with contraction of muscle of anterior abdominal wall arises in the intra-abdominal pressure to evacuate the pelvic contents. Voluntary expulsive force. For example, micturition, defecation, vomiting, and perturbation. Muscles of weightlifting. By taking deep breaths and closing the glottis, it is possible to raise the intra-abdominal pressure to such an extent that it will help support the vertebral column and prevents its flexion. It assists the postvertebral muscle in the lifting the heavy weights. Thoracolumbar pump. The descent of diaphragm decreases the intrathoracic pressure and at the same time increases the triabdominal pressure. This pressure changes compresses the inferior vena cava and consequently its blood is forced upward into the right atrium, sphincter of esophagus. The fibers of the right crus of diaphragm subserves a sympathetic control over the esophageal opening. Clinical correlation. Diaphragmatic paralysis. Paralysis of the diaphragm. The unilateral damage of the phrenic nerve leads to a unilateral diaphragmatic paralysis. This condition is diagnosed during fluoroscopy when an elevated hemidiaphragm is seen on the site of the lesion and showing paradoxical movements. The bilateral damage of phrenic nerves leads to complete diaphragmatic paralysis. It is a serious condition that may cause respiratory failure, hiccups. They occur due to involuntary spasmodic concentration of the diaphragm accompanied by the enclosure of glottis. Hiccups normally occur after eating or drinking as a result of gastric irritation. The pathological causes of hiccups include diaphragmatic irritation, phrenic nerve irritation, and hysteria and uremia. Development. The diaphragm develops into the region of neck from the following 4 embryonic structures: septum transversum ventrally, pleurophrenic membranes at the sides, dorsal mesenteries of the esophagus dorsally, body wall mesoderm peripherally. Adult derivatives. central tendon of diaphragm develops from the sputum transversum. Domes of diaphragm develops from the pleuroperitoneal membrane. Parts of the diaphragm around the esophageal develops from the dorsal mesentery of esophagus. Peripheral parts of the diaphragm develops from the body of the wall mesoderm. For detailed development. Descending of diaphragm. The musculature of diaphragm develops from the third to fifth cervical myotomes. Hence it receives its motor innervations from C3 to C5. The spinal segments, phrenic nerves. Later, when diaphragm descends from the neck to the definitive position, the thoraco-abdominal junction, its nerve supply is dragged down. This explains the long course of the phrenic nerve. Mediastinum. Specific learning objectives. After studying this chapter, the student should be able to define mediastinum, give its boundaries and main contents. Describe the boundaries and components of superior, inferior, middle, and posterior mediastinum. Enumerate the subdivisions of the mediastinum as seen in a chest radiograph. Give the anatomical basis of mediastinal syndrome, mediastinal shift, and extension of pus in posterior mediastinum from neck. Mediastinum. The mediastinum L middle septum. Large middle septum is the middle septum of the thoracic cavity between the two pleural cavities. It consists of all the viscera and structures of the thoracic cavity, for example, heart and its great blood vessels, esophagus, trachea, and principal bronchi, aorta, mediastinal lymph nodes, etc. Except the lungs, the mediastinal occupies the central compartment of thoracic cavity. Thus, strictly speaking, it is a broad central partition which separates the two laterally placed pleural cavities. Arteries. The two laterally placed pleural cavities. It is covered on either side by the mediastinal pleura. Boundaries. Anterior sternum, posterior vertebral column, bodies of thoracic vertebrae and intervertebral disc. Superior thoracic aperture. Inferior diaphragm and each side mediastinal pleura. Note well, the mediastinum is a rigid structure as observed by the students in the cadaver. Emblamed dead body is not a rigid structure as observed by the students in the cadaver. Embedded dead body. In a living individual, the mediastinum is highly mobile septum because it consists primarily of hollow visceral structures bound together by loose connective tissue, often infiltrated with fat. Contents. The major contents of mediastinal are thymus, heart, the great blood vessels enclosed in pericardial sac, ascending aorta, arch of aorta, and descending aorta with their branches. Tracheal bifurcation, superior vena cava, azygous vein, esophagus, thoracic duct, neural structures such as sympathetic trunk, vagus nerve, and phrenic nerve, lymphatic nodes. Lateral superfaces of mediastinum. The structures forming the right and the left lateral surfaces of the mediastinum are shown on figure 20.3. Esophagus, trachea, right vagus nerve, right brachiocephalic vein, superior vena cava, root of right lung, azygous vein, right sympathetic chain, right atrium covered with pericardium. Right phrenic nerve. Trachea, esophagus, left subclavian artery, left common carotid artery, thoracic duct, arch of aorta, left vagus nerve, left recurrent laryngeal nerve, root of the left lung, left sympathetic chain, left ventricle covered by pericardium, left phrenic nerve. Dimensions, divisions. For the purpose of description and organization of the structures, the mediastinum is artificially divided into two parts, superior mediastinum and inferior mediastinum, by an imaginary plane, transverse thoracic plane, passing through the sternal angle anteriorly and the lower border of the body of the fourth thoracic vertebra disc between thoracic 4 and thoracic 5 vertebra posteriorly. The inferior mediastinum is further subdivided into three parts by the pericardium enclosure heart. The part in front of the pericardium is called the anterior mediastinum, and the part behind the pericardium is called the posterior mediastinum. The pericardium and the contents, heart and roots of the great vessel constitute the middle mediastinum. The pericardium, it consists of heart and root of the great vessels. Middle mediastinum. The division and subdivisions of the mediastinum are shown on flowchart 20.1. Clinical correlation. Visualization of the subdivisions of the mediastinum on the chest radiograph. The subdivisions of the mediastinum are well visualized in the lateral view of the X-ray of chest. The shadow above the sternal plane represents the superior mediastinum. The subdivisions from the inferior mediastinum are detailed as follows. Cardiac shadow above the interior part of the diaphragm represents the middle mediastinum. Retrosternal shadow in front of the cardiac shadow represents the anterior mediastinum. Retrocardiac shadow between the cardiac shadow and the shadow of the vertebral column represents the posterior mediastinum. Superior mediastinum boundaries. Anterior and manubrium sterni. Boundary superior manubrium sterni. Bodies of the upper four thoracic vertebrae. Bodies of the upper four thoracic vertebrae. Plane of the superior thoracic aperture. An imaginary line passing through the sternal angle of the right and the lower border of the body of the fourth thoracic vertebra behind the transverse thoracic plane on each side lateral mediastinal pleura. Superior mediastinum boundaries. Anterior manubrium sterni. Posterior bodies of the upper four thoracic vertebrae. Anterior manubrium sterni. Posterior boundaries of the upper four thoracic vertebrae. Superior plane of the superior thoracic aperture. Inferior the imaginary line passing through the sternal angle in front and lower border of the body of the fourth thoracic vertebra behind transverse thoracic plane on each side mediastinal pleura. Contents. Arteries. Arch of the aorta, brachiocephalic artery, left common carotid artery and left subclavian artery. Veins. Right and left brachiocephalic veins. Upper half of the SVC. Superior vena cava. Left superior intercostal vein. Arch of the aegis vein. Nerves. Phrenic nerves. Vagus nerves. Right and left. Superior sympathetic trunk of the cardiac nerves. Right and left. Left recurrent laryngeal nerve. Lymphoid organs and lymphatics. Thymus, thoracic duct and lymph nodes. Tubes. Trachea, esophagus. Muscles. Sternohyoid, sternothyroid, longus colli. Note well, for the purpose of orientation during surgery, the major structures of the superior mediastinum are arranged in the following order from anterior to posterior: thymus, large veins, large arteries, trachea, esophagus and thoracic duct, sympathetic chain. Left common carotid artery, left recurrent laryngeal nerve, left recurrent laryngeal nerve, left subclavian artery, left superior intercostal vein, left vagus nerve, arch of the aorta, pericardium, superior vena cava, left brachiocephalic vein, right brachiocephalic vein, right vagus nerve, brachiocephalic artery, trachea. There are left sympathetic chain, right sympathetic chain, esophagus, thoracic duct, trachea, right vagus nerve, left recurrent laryngeal nerve, left subclavian artery, left vagus nerve, left common carotid artery, brachiocephalic artery, right phrenic nerve, right brachiocephalic vein, left brachiocephalic vein, right brachiocephalic vein, left brachiocephalic vein, thymus, sternothyroid, sternothyroid. Clinical correlation: the superior mediastinum, all large veins, as we see right and left brachiocephalic veins are on the right side, and all large arteries, arch of the aorta and three branches are on the left side. Consequently, during increased blood flow, the large veins expand enormously, while the large arteries do not expand at all. This is because there is sufficient dead space in the right side. It is into this space that tumors of the mediastinum tend to project. Anterior mediastinum boundaries: anterior, body of the sternum; posterior, cardiopulmonary enclosing heart; superior, transverse thoracic plane separating the superior and inferior mediastinum; inferior, diaphragm on the each side; mediastinal pleura. Contents: loose areolar tissue; superior and inferior sternopericardial ligaments. Contents: loose areolar tissue; superior inferior sternopericardial ligaments. Sternopericardial, stretching between the sternum and pericardium. 3 or 4 lymph nodes and lymphatics. Lymph nodes or lymphatics. Mediastinal branch of the internal thoracic mammary arteries. Mediastinal branches of internal thoracic mammary arteries. Lower point of the thymus in children. Content: clinical correlation: the anterior mediastinal is very narrow space. It continues through the superior mediastinum with the pretracheal space of the neck. Pretracheal. Therefore, neck infection in pretracheal space may spread into the anterior mediastinum. Middle mediastinum boundaries: anterior, mediastinum; posterior, mediastinum; superior, mediastinum; inferior. Diagram. Contents: heart, pericardium and interior. Ascending aorta, middle mediastinum. Ascending aorta, pulmonary trunk divided into the two pulmonary arteries: pericardiophrenic arteries. Veins: as we see, lower half, pulmonary veins, right and left. Nerves: phrenic nerves, deep cardiac plexus nerves. Lymph nodes: tracheobronchial lymph nodes, tubes. Bifurcation of trachea: left, right and left principal bronchi. The main contents of the middle mediastinum are pericardium and its contents, i.e. the heart and the root of the great vessels. Posterior mediastinum boundaries: anterior, pericardium and its contents. Bifurcation of trachea and pulmonary vessels. Pericardium and its contents bifurcation of trachea and its pulmonary vessels. Posterior bodies of the lower 8 thoracic vertebra. Posterior and intervening vertebral disc. Superior transverse thoracic plane separating the superior mediastinum from the inferior mediastinum. Diaphragm on the each side mediastinal pleura. Contents esophagus, thoracic duct, descending thoracic aorta and its branches, azygous, hemizygous and accessory hemizygous veins, vagus nerves and anterior and posterior esophageal plexus, sympathetic trunks and splanchnic nerves, posterior mediastinal lymph nodes. Clinical correlation mediastinitis. It is the inflammation of the loose connective tissue of the mediastinum. It is the inflammation of loose connective tissue of the mediastinum. The facial space of the neck and the pretrachea and the retropharyngeal space extends below into the mediastinum with the thoracic cavity. Therefore, deep infections of the neck may readily spread into the thoracic cavity, causing mediastinitis. Subcutaneous emphysema and the root of the neck. The structures that make up the mediastinum are embedded in the loose connective tissue that is continuous with the loose connective tissue of the root of the neck. Therefore, esophageal perforations caused by penetrating wounds, air escapes into the connective tissue space of the mediastinum and may ascend beneath the fascia to the root of the neck, producing subcutaneous emphysema. Mediastinal syndrome. The compression of mediastinal structure by any growth such as tumor or cyst gives rise to a group of signs and symptoms, producing an clinical condition called mediastinal syndrome. The common causes of mediastinal syndrome are bronchogenic carcinoma, aneurysm of aorta, enlargement of mediastinal lymph nodes, and Hodgkin's disease. The clinical features of mediastinal syndrome are as follows: engorgement of veins in the upper half of the body due to the obstruction of SBC, dyspnea, difficulty in breathing due to the compression of trachea, dysphagia due to the compression of the esophagus, difficulty in swallowing, dysphonia, hoarseness in voice due to the compression of the left recurrent laryngeal nerve, and erosion of the bodies of the thoracic vertebra due to the presence of vertebral column. Erosion of the bodies of the thoracic vertebra due to the presence of vertebral column. Erosion of the bodies of the thoracic vertebra. Vertebral pressure on the vertebral column. Widening of the mediastinum. Widening of the mediastinum. The widening of the mediastinum is often observed in chest radiographs. It can occur due to a number of reasons, such as hemorrhage into the mediastinum for lacerated great vessels, aorta SVC following trauma, for example head-on collision, massive enlargement of the mediastinal lymph nodes due to the cancer of lymphoid tissue, namely malignant lymphoid, and enlargement of hypertrophy of the heart due to congestive heart failure. It is the common cause of widening of the inferior mediastinum. Mediastinal shift. The mediastinal is a common shift in the language and pleural pathology. The mediastinum shifts to the affected diseased side due to an appreciable reduction of the lung volume and disease of intrapleural pressure as in the collapse of lung and artery and atelectasis. Atelectasis, the mediastinal shifts to the healthy side when intrapleural pressure is appreciably high on the affected side, as in the pneumothorax and hydrothorax. Mediastinal shift indicates the lung pathology. The mediastinal shift can be detected by palpating the trachea in the suprasternal notch. Extension of the pulse into the posterior mediastinum from the neck. The posterior mediastinal is continuous through superior mediastinum with spaces in the neck between the petracheal and prevertebral layers of the deep cervical fascia, such as retropharyngeal space, therefore pass from the neck, extends into the posterior mediastinum. Extension of pus into the thighs of the posterior mediastinum. The source sheath continues with the posterior mediastinum by a funnel-shaped uriface, therefore pus from the posterior mediastinum can easily enter into the source sheath of the tract down into the thighs of the region on the femoral triangle triangle. Mediastinum contains all the thoracic viscera except lungs. Large structure of the mediastinum. Heart. Common causes of the widening of the inferior mediastinum. Enlargement. Hypertrophy of the heart. Enlargement. Hypertrophy of the heart. Mediastinum is dynamic because and palleable and movable septum because structures forming are hollow, fluid, air-filled and bound together by the loose connective tissue. Compartment of the thoracic cavity occupied by the mediastinum. Central compartment. Common causes of mediastinal shift in the diseased side. Collapse of lung and atelectasis. Common causes of mediastinal shift into the healthy side. Common causes of mediastinal shift into the diseased side. Collapse of lung and atelectasis. Common cause of mediastinal shift to the healthy side. Pneumothorax and hydrothorax. A 60-year-old male visited the hospital and complained about that he has noticed an alternation of his voice, has lost 15 kilograms of weight, has persisting cough with blood-stained sputum. On asking, he told that he is a chronic smoker and smokes 20 to 30 cigarettes per day. The X-ray chest reveals a large, well-circumscribed radio pack shown inhaler regression of the right lung and widening of the mediastinum. A diagnosis of bronchogenic carcinoma was made. Bronchogenic carcinoma is common in male or females. Mention the incidence of death. It is common in males and account for about 1/3 of all cancer deaths in man. Occurrence of the bronchocarcinoma is more common in the right lung compared to the left lung. What is the anatomical basis? Because the right bronchial tree is more exposed to the carcinogens, for example, cigarettes, smokes, tarry particles from roads are as right bronchus in the wider and lies in line with the trachea. What is the cause of hoarseness in the voice? The bronchogenic carcinoma spreads rapidly to tracheobronchial and bronchiomediastinal lymph nodes. Involves in recurrent laryngeal nerve leading to hoarseness in voice. Through blood vessels, i.e. hematogema spread. For details. Mention the anatomical basis of metastasis of bronchogenic carcinoma to brain and bones through blood vessels. The arterial supply of the heart. The heart is supplied mostly by two coronary arteries, right and left. Only the inner 75 to 100 micrometer of endothelium surface get nutrition directly from the blood and in the cardiac chambers. Anatomically, coronary arteries are end arteries because they anastomote with each other in their trunks. Branches and sub-branches mostly in precapillary level. Functionally, however, they behave like end arteries, since most of the anastomosis remain impervious. Each coronary is a vasoram of the ascending aorta. Because of the heat is developed because the heart is developed from the fusion of two primitive endothelial tubes which represent the ventral aorta, right coronary artery. It arises from the anterior aortic sinus, right coronary sinus, in the clinical usage of the ascending aorta, slightly below the supravalvular ridge. The first arteria passes forward and to the right between the pulmonary trunk and the right auricle. Passes forward between the pulmonary trunk and the at first the artery passes through the right between the pulmonary trunk and the right auricle. Then it passes downward and to the right along the right part of the arterioventricular groove. It winds around the inferior border of the heart, passes upward and left along the posterior part of the anteroventricular groove and reaches the crux of the heart, which is the meeting point of internal posterior meeting point of intraarterial, posterior intraventricular and posterior part of the arterioventricular groove. In 60% of subjects, the terminal part of the right coronary artery anastomoses with the circumflex branch of the left coronary artery. Slightly to the left side of the crux. In 10% cases, the right coronary end at the inferior border of the heart. In some subjects, the right coronary transverses the entire posterior part of the atrioventricular groove. The right coronary artery transverses the entire posterior part of the atrioventricular groove. Anterioventricular branch. Posterior intraventricular branch. Right ventricle, left ventricle. Branches. For the purpose of description, branches are divided into two groups. For arising from the first segment of the aorta into the inferior border, and those arising from the second segment up to the crux. The branches are from the first segment. Ventral rami, right conus artery. Ventral rami, right conus artery. It is usually the first branch of right coronary artery and supplies the infundibulum of the right ventricle. The right conus artery branches of the vertebral rami. It is usually at the first branch of the right coronary artery and supplies the infundibulum of the right ventricle. Sometimes it arises directly from the anterior arterial sinus. Supplies the infundibulum of the right ventricle. Right conus artery. Sometimes it arises directly from the anterior coronary sinus. In such cases, it is called the third coronary artery. The left conus artery. In such cases, called a third coronary artery. The right conus artery anastomoses with the left conus artery derived from the circumflex branch of the left coronary artery. The anastomotic necklace thus formed around the infundibulum is known as the annulus of Vieuxnes. right anterior ventricular rami, right ventricular rami, and right conus rami. These are 3 or 4 in number. Right anterior ventricular arise from the right angle from the trunk of the coronary artery and supply the sternocostal wall of the right ventricle. The right margin artery is said to be the longest ventricle ramus. Longest ventricle ramus. It follows the inferior border of the heart towards the apex and supplies adjoining surface of the right ventricle. Arterial rami, ventral rami, and arterial rami. These are arranged in the anterior, lateral, and posterior groups. They are supplied myocardium of the right atrium. The sinoatrial artery. The sinoatrial artery belongs to the arterial rami. It arises in 65% cases from the internal segment of the right coronary artery, and in 35% of cases from the circumflex of the branch of the right coronary artery. Typically, the nodal artery passes around and backwards from the right coronary between the right auricle and the root of the ascending aorta. Typically, the nodal artery passes upward and backward from the right coronary artery and right auricle and the root of the ascending aorta. Then arches laterally in front of the orifice of superior vena cava, and finally runs down towards the central axis of the sinoatrial node. Branches from secondary segments: right posterior ventral rami, posterior interventricular branch, right posterior ventral rami. A few branches arise from the trunk of the right coronary artery in the posterior part of the artery. Ventricular group supply the diaphragmatic surface of the right ventricle. Posterior ventral rami, posterior interventricular branch. It arises from the right coronary up near the crux of the heart. The single branch in 70% of subjects and passes along the posterior interventral groove close to the apex in anastomosis with the anterior interventricular branch of the coronary artery. Sometimes 2 or 3 posterior interventricular branches. Sometimes 2 or 3 posterior interventricular branches run parallel to another. In 10% individual, the posterior interventricular branch is derived as a continuous left coronary artery. On the basis of the continuity of the posterior interventricular branch from the right or the left artery, the right coronary predominance or left coronary predominance is described. The posterior interventricular artery provides a few branches to the diaphragmatic surface of the right and the left ventricle, but the most significant branch are the septal rami, which supply the posterior inferior 1/3 of the ventral septum. First, the septum ramus, the posterior interventricle, undergoes an inverted loop that supplies the atrioventricular node. In 80%, 90% of subjects, the AV nodal artery is derived from the right coronary artery, and 10 to 20% cases, the left coronary artery. Right posterior atrial rami supply the majority of the individuals' posterior surface of the both right and the left atria. Left coronary artery. The left coronary artery is wider than the right coronary artery and supplies the larger volume of the myocardium. The left coronary artery, it arises from the left posterior arterial sinus, left coronary sinus, and the clinical usage, the ascending aorta. The trunk of the artery passes initially behind the pulmonary trunk. And then appears forward and to the left between the pulmonary trunk and left auricle, where it divides usually into 2 branches: anterior interventricular and circumflex. On rare occasions, 2% of the aforesaid branches are separate from the left pulmonary sinus. The trunk of the artery does not provide, as a rule, any significant branches. ventricular artery, anterior interventricular artery. It is continuous of the left coronary artery ascending from the corresponding group of along anterior interventricular artery. It is the continuation of the left coronary artery descends in the surrounding groove along the sternocostal surface of the apex. After winding around the incisura apicis cardis ends of the inferior surface by anastomosing with the posterior interventricular branch of the coronary artery, the anastomosis usually takes place at the junction of anterior one third of the posterior two third of the posterior interventricular groove. Branches: anterior ventricular rami. Anterior ventral rami from both right and left ventricles pass obliquely outwards. Left ventricle rami are prominent, 3 or 4 number, and wind around the left surface of the heart and are known as diagonal arteries, which supply thicker sternocostal wall of the left ventricle. On the right ventricle rami forms the left crus artery, which supplies the infundibule of the right ventricle. Septal rami passes deeply and supply anterosuperior two-thirds of the ventral septum. Circumflex artery. It passes at first along the right part of the arterioventricular groove. Winds around the left border of the heart and occupies the posterior part of the atrioventricular groove, where it anastomoses with the right coronary artery, usually on the left side of the crus. The circumflex artery lies close to the mitral annulus, and may be damaged in mitral valve replacement. Branches. The circumflex artery gives rise to the following branches. Arterial and ventricular rami supplies the adjoining surface of the right atrium and left ventricle. The atrial ventricular rami supports the adjoining surface of the right atrium and left ventricle. Sinoatrial nodal artery arises from the circumflex branch. Sinoatrial nodal artery. Left margin of the artery. It is a prominent branch which follows the left border of the heart towards the apex. In coronary artery bypass surgery, a vascular autograft is usually anastomosed in the left margin of the artery distal to the site of the obstruction. Posterior intraventricular artery extends as a continuation of the circumflex branch in 10 to 20% individuals. In such condition, AV node is supplied by the septal ramus of the left coronary artery. AV node is supplied by the septal ramus of the left coronary artery. An arterial branch of circumflex branch communicates with the similar branch of the right coronary artery along the anterior arterial wall. Such anastomotic branches known as Kugel's artery. Summary of the coronary distribution. Right coronary artery supplies the whole right atrium, most of the right ventricle except a strip along the anterior interventricular groove, posterior inferior one third of the ventricular septum, and as a node and as B node in the majority of subjects. Left coronary artery supplies most of the left atrium, left ventricle except a single strip along the posterior and inferior surface of the heart, and also supplies the anterosuperior two-thirds of the ventricular septum. Peculiarity: sometimes left coronary artery arises from the pulmonary trunk. Producing cyanotic symptoms following by the left ventricular failure. On the occasions, left coronary artery arises from the anterior aortic sinus and undergoes a longer course behind the pulmonary trunk before dividing. In this anomaly, this trunk of the artery may be compressed between the aorta and the pulmonary trunk. After severe exercise and culminate in sudden cardiac death, SCD. Parasympathetic stimulation has negligible direct effect on the coronary vessels. Sympathetic alpha receptors, constrictor receptors are predominant in epicardial coronary vessels. Beta receptors are dominant in intramuscular arteries. Therefore, sympathetic stimulation contracts the epithelial arteries and dilates the intramuscular arteries. Coronary arteries are the only vessels where the blood flows in diastole. Coronary predominance in men. Majority of people possesses right coronary predominance, where the posterior interventricular artery is derived from the right coronary. Minority of the coronary population has left coronary predominance when the posterior interventricular artery extends from the continuation of the left coronary artery. These people are likely to be affected by coronary disease because the entire right ventricle and the ventricular septum are under the nutritional control of right coronary artery, and the obstruction of the latter may produce output failure of systemic circulation. On rare occasions, the posterior interventricular branches are derived from both coronary arteries. Individuals with such balanced type of coronary distribution are less affected by coronary diseases. Observations. RCA. First segment of right coronary artery shows a conus branch. Right coronary artery. First segment of the right coronary artery shows a conus branch which makes an upward loop in front of the ascending aorta. Another elongated branch from the central axis of ascent node. RCA is visible to follow the right and inferior bottom of the right ventricle upwards the apex of the heart and supplies adjoining surface of the right ventricle. Marginal branches of the left coronary artery shows LAD, left anterior branch, descending branch along the anterior interventricular group. The latter provides two or more prominent diagonal arteries. LAX, circumflex branch of the left coronary artery, occupying the left and the posterior AV groove and joining with the second segment of the right coronary artery shows LMA, left marginal artery. Apex, and an elongated branch of the supplies diaphragmatic surface of the left ventricle. LMA is preferred site for vascular autography in coronary ablations. Obstructions. Observations. RCA, right coronary artery. LCA, left coronary circumflex, circumflex of the left coronary circumflex. Close contact of left coronary circumflex with the fibrous ring of mitral annulus should not be disturbed in prothesis operation of heart valves. Myocardial circulations, interarterial anastomosis. Potential anastomosis exists between the branches of the coronary arteries. 85% of the anastomosis takes place in precapillary level and 15% of the capillary level. Therefore, coronary arteries are not typically end arteries, though functionally they behave like end arteries. Enterocoronary anastomosis up to 100 to 200 micrometer in calibre takes place at all levels. Subepithelial, myocardial, and epithelial and subendocardial. Frequent sites of anastomosis are annulus of Viescence, interventricular septum, and Krugel's arteries. Anastomosis of the coronary arteries multiply with age. Hence, prognosis of the coronary disease is better in an old age than in young. Anastomotic vessels are relatively straight in normal heart but much solid in hearts affected by coronary occlusions. Tributaries of the coronary sinus. Sinusoidal. epicardium, myocardium, cardiac sinuses. In normal heart, but much coiled heart affected by coronary occlusions. Arterioventricular anastomosis. These help to supply the nutrition of the myocardium if a minute branch of the artery is obstructed by flow of the regurgitated blood of the coronary sinus. Regurgitated. Arteriosinusoidal anastomosis. Arteriovenous anastomosis, arteriosinusoidal anastomosis. Most of the branches of the coronary arteries break up into a number of myocardial sinusoids. Sinusoidal and sinulaminar from the myocardial sinuses. Most of the veins drain into the coronary sinus. Some blood from the sinusoids drain directly into the lumen of the heart via vena cordis. Arterioluminar. Some branches of the coronary artery directly open into the lumen of the heart. Luminosinusoidal. Some blood from the lumen of the heart irrigates the myocardium via vena cordis and through lamina minimorum. This system of circulation is vestigial in man. Collateral circulations. The subepithelial fat. Potential communications exist between the branches of the coronary arteries and the branches of the internal thoracic artery and descending aorta. Pericardial branch of the phrenic and esophageal. In slow obstruction of the coronary artery, these collateral channels dilate and maintain nutrition of the heart. Applied anatomy. In most common cases of death in affluent society across the world is ischemia heart disease (IHD), in which results from the occlusion of the coronary artery or its major branches. Primary causes of the disease is the granular formation of atheromatous plaque beneath the endothelial lining of the heart. Subsequently, a crack or ulcer develops in the plaque on the right plate are aggregated to form a thrombus. This results in narrowing of the vessel, which may be granular of sudden and incomplete or complete. Sudden and complete obstruction of a major branch is the chief cause of sudden death by myocardial infarctions. If the complete occlusions of a granular onset, the patient may survive due to development of efficient collateral anastomosis, but such patients have to undergo a restricted and careful life due to limited cardiac reserves. Clinically, the occlusions of the arteries at the major branches may be located within the first 2 cm of the left anterior descending and circumflex branches of the proximal and distal right and the left coronary artery. Average frequencies of crucial narrowing of the 3 major artery trunks are as follows: anterior interventricular artery of the left coronary artery, anterior interventricular branch of the left coronary artery, circumflex branch of the left coronary artery, other coronary diagonal branches of the anterior ventricular artery, left marginal branch of the circumflex artery, trunk of the left coronary artery. Incomplete and spasmodic obstruction of the coronary arteries by vascular spasm with or without atherosclerosis of vessels is expected as an angina pectoris, in which the subject complains of intense compressive type of precordial pain of effect, which is occasionally referred along the left upper arm side of the neck. This type of pain usually subsides after taking rest, hence is called effort angina. Stress ECG or treadmill test for exercise tolerance often provides definite clues for the effort angina associated with myocardial ischemia on exertion. Some patients get prompt relief from pain by taking an appropriate coronary vasodilator drug like sorbitol table sublingually along with the other supportive drugs. In unstable angina, the symptoms of cardiac pain occur even at rest. Such cases require more elaborate investigation, including coronary angiography. In cases with signs of coronary arterial block, some specific measures like coronary angioplasty or coronary bypass surgery may be deemed necessary depending on the nature of extent of coronary blocks as revealed in angiography. Cardiac pain occurs even at rest. Such cases require more elaborate investigation, including coronary angiography. In cases with signs of coronary arterial block, some specific measures like coronary angiography or coronary bypass surgery may be deemed necessary depending on the nature of the extent of the coronary blockage as revealed in the angiography. Coronary angiography involves passing a sterile cardiac catheter through the femoral artery in the thigh right into the coronary arteries. Following the introduction, a radioplaque dye is injected in the observation of the X-ray film. Such a procedure is to be undertaken in cath lab unit of the cardiography department. This procedure helps the detection and actual localization of any block in the coronary arterial channels. In coronary angioplasty procedure, the site of coronary block, usually by arteriomus plaque, is provided by the so-called balloon angioplasty method via the cardiac catheter under X-ray control. Atherosclerotic plaques are removed, and this is followed by placing a suitable drug platelets under the site. The coronary bypass surgery, which involves open heart surgery, the blocked portions of the coronary artery is bypassed and replaced by sections of the stable blood vessels taken from elsewhere. Conventionally, such an autograft is taken from tributaries of the sphenous veins of the thigh. Saphenous veins of the thigh. However, a graft may be also taken from suitable internal mammary artery of the interior chest wall. The latter is considered more ideal. Halter monitor means semi-permanent attached on the person's chest of the ECG electrodes with continuous automatic recording arrangements for 24 hours around the clock. Thus, a more complete picture can be observed regarding arrhythmic attacks, particularly the sick sinus syndrome. The complete block of spell of ectopic beats along with the ischemia. Venous drainage of the heart. About 60% of venous blood of the heart drains into the right atrium via coronary sinus. Where 60% of venous blood drains into the right atrium via coronary sinus. Remaining 40% of the blood drains into different chambers of the heart via cordis minimi, Thracian veins, and anterior cardiac veins. Coronary sinus conveys the greater part of the blood from the right coronary artery, where the anterior cardiac veins drain most of the blood from the right coronary artery. The veins which do not join the coronary sinus are as follows. The veins which do not join the coronary sinus are as follows. Anterior cardiac vein. There are 3 or 4 in number, drain into the infundibulum of the right ventricle and end directly into the right atrium through foramina minimorum. Veni cordis minimi. These veins open in different chambers of the heart through the foramina minimi. The foramina are more numerous in arterial septum. Sometimes the right margin of the vein, instead of the joining of the small cardiac vein, opens directly to the right atrium. Coronary sinus. It's a wide venous channel about 2 to 3 centimeters long, situated on the posterior part of the ventricular groove. The coronary sinus receives about 60 cm of the venous blood from the heart. Blood grouping and blood typing, ABO and RH system. Student objectives. After completing this experiment, the student should be able to define the term blood grouping and blood types and name the various blood grouping systems. Describe the physiological basis of blood grouping and its clinical significance. Describe the Landsteiner's law and explain the basis of the term universal donor or universal recipient. Describe the RH factor and how it was discovered. What is the significance? Determine the blood grouping by using commercially available anesthesia and precautions to be observed. Explain how blood is stored in blood banks and it changes that occurs in the blood grouping during storage. List the indications of blood transfusion and the degree and the dangers associated with it. Explain why it is essential to match donor and recipient blood groups before giving a transfusion. Describe the effects of mismatched blood transfusion. Introduction. Estimation of HB, RBC, TLC. RBC indicates TLC blood grouping, BT and CT. The surface of human red cells contain a variety of genetically determined glycolipids. Genetically determined glycoproteins. Genetically determined glycoproteins that act as antigens. The plasma containing antibodies that can react with these antigens when the two are mixed in the presence of suitable antibodies. They are also called agglutinogens. The antibodies in the plasma are called agglutinogens, and the antibodies in the plasma are called agglutinins. Agglutinogens are agglutinins. Blood grouping system. ABH blood grouping classical. RH blood grouping research others, for example M or N. A group of related red cells antigens that show similar chemical, genetic and reactive properties constitutes a blood grouping system within a body group. For example, ABO system. There may be two or more different blood types. For example, A, B, O and AB. Therefore, the term blood grouping and blood type are often used synonymously. Therefore, the term blood grouping and blood types are often used synonymously. There are at least 30 commonly occurring antigens and hundreds of rare antigens have been found on the surfaces of human red cells of different blood grouping systems. Only two are of great clinical importance. These are the ABO system and the RH system. Other blood groups systems are Aman, Lutheran, Cal, Colton, Daffy, Ked, Diego, Lewis, Lee, YT, XZ, P, C, etc. These groups are of little importance because they are not antigenic, because they are of value in anthropological and genetic studies. Some of these functions as cell recognition molecules. Blood groups. Blood group, a blood type, is also called blood group, is a classification of blood based on the presence or absence of inherited gene substance or on the surface of red cells. Is the presence or absence of inherited antigenic substance on the blood cells. Classify blood groups. Major principal blood groups. The sieve blood groups, ABO blood group, is based on the presence or absence of group specific antigens in RBC member. Rh blood group based on the presence of Rh antigen in the RBC membrane. M and N blood groups used to determine paternity. M and N blood groups. Other Lutherian, Cal, Cade, Daffy, Diego, Lois, etc. Name the ABO and Rh blood group with the corresponding antigen and anticoagulant. Agglutinogen, agglutinin, genotype and prevalence. A. Antigen A. Agglutinin B. Genotype AAAO. ABBBO. B. AB and then OO. Actually 9 3 47. Rh negative blood with agglutinins, genotype and prevalence. Rh negative Caucasians 85%, Asian 99%. Rh positive Rh negative Caucasians 15%, Asians 0.1%. Name the classical blood groups. The ABO blood group system is called the classical blood group. It is the following key reasons. First, discovered by the blood group system. Landmark discovery. Karl Landsteiner identified the ABO system, making the first human blood group system ever discovered. This groundbreaking discovery led to the foundation of modern transfusion of medicine. Karl Landsteiner. Fundamental importance of transfusion medicine. ABO compatibility is crucial for safe blood transfusion due to natural occurring antibodies and severe hemolytic reactions of mismatch. Fundamentals. Universal presence of clinical relevance. ABO antigens are expressed on RBC, endothelial cells and secretions. Governs blood transfusion, organ transplant and forensic sciences. Simple genetic basis. Follow Mendelian inheritance. Only 3 alleles A, B, O and single locus. Follow Mendelian inheritance. Simple genetic basis. Basis for other blood group discovery. ABO discovery led to the identification of 40 other blood group systems. For example, Rh-Cal still remain the most clinically significant system in the worldwide. Why not the other system? The Rh system discovered later in 1936 is crucial but lacks natural antibodies. Other systems, DAF kit is less immunogenic. Conclusion: the ABO system is classical because it was first, most consequent, the simplest model for understanding blood group immunology. Its principles remain the cornerstone of transfusion science. Reference: Landsteiner original work. Landsteiner on agglutination phenomenon in normal human blood. Wiener Klins Uchencraft. Danish human blood group. Harmoning modern blood banking and transfusion practices. State the Landsteiner's law. The Landsteiner law, first part. An agglutinogen is present in the red blood cell. If an agglutinogen is present in the red blood cell, the corresponding agglutinin must be absent in the plasma. For example, in blood group A, agglutinogen A is present in the RBC membrane. The corresponding agglutinin alpha must be absent. Agglutinin alpha must be absent in plasma. If agglutinogen is absent on the red cells of the blood, the corresponding agglutinin must be present in the plasma. For example, if blood group is O, agglutinin A and B absent, then RBC membrane of the corresponding agglutinin alpha and beta are present in the plasma. Exception: the second part Langer's law is not obeyed by Rh and MNS blood groups. The Rh blood group antigen D is absent and anti-D is also absent in the plasma. To obey this second part, anti-D should be present in the plasma. Anti-D should be present in the plasma. In the amino system, absence of M or N substance is not accompanied by presence of anti-M or anti-N plasma. Inheritance of ABO agglutinins and agglutinogens and agglutinins. The blood group of person depends on the two genes inherited from each parent. Gene A and gene B are dominant by themselves and gene O is recessive. Agglutinogens appear during the 6 months of fetal life. Agglutinogens appear on the 6th month of fetal life and concentrate on the birth at 1/5 of the adult concentration. It arises to the adult level at puberty. Agglutinogens present not only in RBC but also present in many other organs like salivary glands, pancreas, kidney, liver, lung, etc. The A and the B agglutinins are inherited from the parents as Mendelian phenotypes. The agglutin alpha and beta produced fetal life appears 2 to 3 months after birth. Agglutinin is produced in response to AB with the body through respiratory system and digestive system along with bacteria. Agglutinins are gamma globulins, which are mainly immunoglobulin G and immunoglobulin M. Describe Rh factor. It is one of the major blood grouping systems based on the presence or absence of Rh antigen or Rh RBC membrane. Absence of Rh antigen or RBC membrane. Rh factor antigens. The Rh factor or rhesus factor is an antigen which was first found on red cells of the rhesus monkey named after the animal. Rh factor are also called Rh antigens. There are 6 common types of Rh antigens, each of which are called Rh factor. They include C, D, E, C, D, E. D is the commonest anti-Rh antigen. D is the commonest Rh antigen. Persons having antigen D are Rh positive. Persons having no antigen D are Rh negative. Inheritance Rh antigen. Gene for antigen D is called D gene and for antigen D is called D. Rh gene is inherited from both mother and father. When both sperm and ovum carry D, the resulting genotype of offspring is DD. When both of the carry D and D, the resulting is DD. When both of them carry D and D, then the result is DD. Both DD and DD are Rh positive and both small d are Rh negative. Rh antibody. There are several varieties of Rh antibodies: anti-D, anti-C, anti-E, anti-D, and anti-C and anti-E. Unlike ABO system, Rh negative persons do not have Rh antibodies. Rh negative persons do not have Rh antibodies. Very rarely, anti-E antibodies may be present in Rh negative persons. Practically, Rh antibodies are produced Rh negative as follows case: transfusion. Rh positive blood and Rh negative blood causes anti-D production of Rh negative person. Passage of Rh positive blood fetus and Rh negative mother causes anti-D production in mother. What do you mean by blood typing? Blood typing. Definition. Before giving a transfusion to a person, it is necessary to determine the blood type of the recipient and blood donor so that bloods can be appropriately matched. This process is called blood typing or blood matching. Principle of blood grouping. Blood type is formed by mixing an individual's red blood cells with antisera containing the various anti-agglutinins on the side, seeing whether agglutinins occur. Procedure. At first, blood is collected from the individuals whose blood group is to be determined. The red cells are separated from the plasma and diluted with normal saline. One portion of the diluted RBC is mixed with anti-A, other portion anti-B, and other with the portion of the anti-D agglutinins. After several minutes, the mixture observes the microscope agglutinins takes place between the antigen antibody. Blood cells are clamped. Blood types, and then plus means agglutination and minus means no agglutination. What are the importance of blood grouping? The indicates of blood grouping. Blood typing. Importance of blood typing and groups. Blood grouping is essential in various clinical and non-clinical scenarios. The main indications include blood transfusion to ensure compatibility between donor and recipient blood, to prevent transfusion reactions, hemolytic reactions, pregnancy and obstetric care, to access to Rh negative women to Rh status in pregnant women. The Rh negative mothers carry Rh positive fetus are at high risk of hemolytic disease of the newborn. Blood type is required to prevent HDN, to ensure compatibility between donor and recipient blood. Organ and tissue transplant, so blood transplant, organ and tissue transplant, pregnancy and other obstetric care, to ensure ABC compatibility of organ transplant, kidney, liver, heart. Preparative preparation before surgery, the potential blood loss, cardiac, orthopedic or obstetric procedures. Obstetric neonatal testing. If a mother is Rh negative or a history has HDN, the newborn's blood group is checked for hemolytic disease. Neonatal testing. Forensic and legal purpose. Criminal investigations. Parental testing and identification of mass disasters. Military and emergency preparations. Soldiers and emergency roles are typed for rapid blood availability if injured. Research and epidemiological studies. Research and epidemiological studies to study blood group distribution of popular and disease associated. Certain blood groups have higher risk of specific diseases. Conclusion: blood grouping is crucial for safe transfusion, pregnancy management, and surgeries, transplant, medical emergencies, and it prevents life-threatening complications like acute hemolysis and HDN. Clinical importance of Rh typing. Rh typing, especially Rh antigen detection, is crucial in a medical practice due to the following reasons: prevention of hemolytic transfusion reactions. Rh negative plus given to Rh positive with Rh negative but can trigger anti-D formation. Subsequent exposure to Rh positive blood may cause acute hemolytic transfusion reactions. Subsequent acute hemolytic transfusion of anti-reactions. Rh typing ensures compatibility before transfusion. Management of pregnancy, hemolytic disease of the newborn, HDN. If Rh negative mother plus Rh positive father, Rh positive fetus, risk of maternal anti-D antibody formation. These antibodies can cross the placenta in future pregnancies, causing fetal hemolysis or erythroblastosis fetalis. Prevention and measures. Rho immunoglobulin is given 21 weeks and within 21 weeks and within 21 hours of delivery to prevent sensitization. 21 weeks and 72 hours sensitization. Also given after miscarriage, abortion, ectopic pregnancy if the fetus was Rh positive. Neonatal care and monitoring. If HDN occurs, early detentions via combo tests and early treatment is life-saving. Organ and stem cell transplant. If Rh compatibility is considered, a bone marrow or a solid organ transplant is reduced immune complications. Forensic paternity testing. Rh antigen inheritance patterns can assist the paternity disputes and forensic in identifications. Conclusion: Rh typing is essential for safe blood transfusion, prevention of HDN and pregnancies, managing neonatal complications, transplant compatibility. Without Rh typing, life-threatening transfusion reactions and severe fetal newborn complications could occur. What is cross-matching and what is the importance of cross-matching? Explain why blood must be cross-matched before transfusion. Cross-matching definition: cross-matching is a pre-transfusion laboratory test that checks compatibility between a donor's red cells and a recipient's serum or plasma. It ensures safe transfusion by detecting antibody-mediated reactions. The cross-matching is a pre-transfusion laboratory test to check the compatibility of between the donor's red blood cells and recipient's serum plasma. Cross-matching: recipient's serum and donor's RBC. Cross matching is recipient's serum and donor RBC. Major types: major crossmatch and minor crossmatch. Test: donor RBC with patient's serum detects antibody in recipients could destroy donor RBCs. Minor RBCs rarely performed in the body. Donor serum and recipient's RBC checks for donor antibodies against recipient's RBC. Checks for antibodies in the recipient that could destroy the donor's RBC. Checks for donor antibodies. Procedure: blood is collected in both donors. The serum and red cells are separated from the other from the blood samples. The cells are diluted in normal saline. The donor of RBC are mixed with recipient serum. Then it is seen if agglutination takes place or not. Similarly, recipient's RBC are mixed with donor serum when it is seen if agglutination takes place or not. If there is no agglutination, step 4 or 5, two blood are perfectly combined and transfusion can be given. Importance of cross-matching: prevents life-threatening transfusion reactions, identifies ABO incompatibility, detects irregulation of antibodies, conformation compatibility beyond ABO RH via transfusion cross-matching, other antigen antibody mismatch, essential for patients and other prior transfusion pregnancies. Those patients may have alloantibodies against minor blood groups antigens. Reduce risk of delayed hemolytic reactions. Some antibodies cause slow RBC destruction. Crossmatch helps avoid delayed complications. Legal and safe requirements: mandatory in most countries before transfusion, except extreme emergencies with Rh negative blood. Why is cross matching is essential for blood transfusion? Cross matching is mandatory for before transfusion to confirm blood compatibility beyond just ABO-R type. ABO-R type detecting harmful antibodies. Beyond blood type detecting harmful antibodies. Prevent life-threatening reactions. Ensure patients safe transfusion and success. Without cross-matching, the risk of fatal complication makes sure transformation dangerous rather than life-saving. In cross-matching the blood transfusion donors RBC patient, why? If there is no mismatch transfusion, donors RBC may cross-react with the recipient's huge amount of plasma and clump will occur. Recipient's RBC will not cross-react with the donor's plasma, as the donor's a small amount of plasma will be diluted in huge amount of recipient's plasma. For this reason, RBC patient's serum is considered. Why serum agglutinations of the donor are usually ignored in the blood transfusion? Serum agglutination of the donors are usually ignored in the transfusion because the serum of the donor is sufficient diluted by the much larger plasma volume of the recipient, thus provides no ill effect. Agglutinins in the donor serum are neutralized by soluble agglutinins present in free from recipient's body fluid. Agglutinins in the donor's blood serum are neutralized in a soluble agglutinogen free from recipient's body. Present in free from agglutinins of the body, neutralized by soluble agglutinogens. Why does mismatched recipients RBC not agglutinate? Mismatched recipients RBC not agglutinate. Mismatched recipients RBC does not agglutinate because the serum of the donor is sufficiently diluted by which larger plasma of the placenta, thus provide no ill effect. Mismatched agglutinate recipients RBC. Agglutinins in the donor serum are neutralized by soluble agglutinogens present in the recipient's body fluid. How M and M system determination of paternity. Each person carries two sets of genes M and N group. M plus M equal to M, N plus N equal to M, M plus N equal to Mn. As a result, the baby's blood supplementary group is Mn, Mn. Parents must have given in MM, Mn. no matter which. So the mother's supplement group was no matter which, NM. NM. The father could not have been NM. It should always be remembered that blood grouping tests can never prove that any suspected person is an actual father. They can only show that he could not be possibly have been the father, or the likely other have been, or the likely others have been. Structured equations. Agglutinogens present in RBC membrane. A and B antigens are present in the RBC membrane. No Rh antigens are present in the red cell membrane. Agglutino antibodies and in the plasma. No antibodies are present in the plasma. Transfusion of AB negative blood and AB positive blood. No, it is not safe to transfuse AB negative blood into A positive blood. Reasons: the donors agree have both A and B antigens in the RBC. The Rh negative has both AB and antigens in their RBC. The recipient A has anti-B antibodies in their plasma. Has anti-B antibodies in their plasma. If AB negative blood is given in transfusion, the recipient anti-B antibodies will react with the donor antigen antibodies, causing acute hemolytic transfusion reaction. Blood transfusion and its hazards. Transfusion is the process of transferring the whole blood and blood products such as red cells, plasma, or platelets from one person to another through intravenous route of replace losses caused by the injury, surgery, and disease. Infusion is the process of therapeutic introduction of fluid other than blood into the vein, for example saline infusion. Write down the relation of RBC blood grouping and blood transfusion. Relation of RBC blood grouping to blood transfusion. The effects of RBC transfusion from one group to another were worked out by following groups. The serum agglutinins of the donor can usually be ignored as they diluted by much larger volume of the plasma if the recipient and the neutralization by soluble glutatins which are found free recipient's body fluid. So the donor's agglutinin can produce no ill effect. It shows that therefore the recipient's agglutinogens need not usually be considered. Therefore follows that the recipient's agglutinogens need not to be considered. Account only taken after a recipient's agglutinin and donor's agglutinations. The table shows that group A can simply transfer to group B on their own group. The classification terms universal donor and universal recipient there are no longer valid as they ignore the complications produced by Rh factors. So except the emergency intra-group transfusion should be used. It should be emphasized that only safe method of determining compatibility to test the direct of the term supply recipient against the body's red blood cell. What are the indications of blood transfusion? Blood transfusion, whole blood. What are indications? Whole blood. Surgical indications, severe hemorrhage during major operation. Severe burns and when it is excessive hemolysis and dehydration, postoperative and severe debilitated anemia patients during pre-operative operations in severe anemia as a prophylactic measure of pre-operative and hemorrhagic tendencies, for example ITP, hemophilia and obstructive jaundice. Fresh blood, thalassemia, DCP, TITP and hemophilia. Fresh blood, thalassemia. Heparinized blood, it is bypass surgery. Heparinized blood in bypass surgery. Exchange transfusion, hemolytic disease of newborn, acute renal failure, hepatic coma, gaseous poisoning, acute transfusion reaction, red cell concentrated surgery. If HB is low and elective surgery, medical anemia with CCF and ERF, hepatic failure, hepatic hemorrhage, white blood cell, overwhelming bacteria and fungus. Usually after chemotherapy, the bone marrow transplant and non-respective conditions to produce antibodies. Platelet concentration ITP, acute leukemia, plasma anemia, fibrinogen clotting factors, coagulation disorders, liver disease with oral anticoagulant, fresh antigen plasma, coagulation factors deficiency, and thrombocytic purpura. Transfusion reactions after due to ABO incompatibility. Transfusion reactions are the adverse reactions in the body which occur due to the transfusion error of the involved transfusion in incompatibility mismatch. In mismatch transfusion, the transfusion reaction occurs during RBC recipient's plasma. So, if the donor plasma contains agglutinins against recipient's RBC, agglutinin does not occur because antibodies are developed in recipient's body. If the recipient plasma contains agglutinins against donor's RBC, the immune system launches a response. Severe or transfusion reactions. Severe transfusion reactions vary from mild fever to chills to severe kidney failure, shock, and death. Severe depends on the amount of blood transfused, type of reaction, which generally health of the patient. Cause of transfusion reaction. Transfusion of incompatibility blood produces hemolytic reactions. The recipient antibodies, hemoglobin, hemoglobin, and adhere to the donor's RBC, which are agglutinated and destroyed. Large amount of free hemoglobin is liberated from the plasma. This leads to a transfusion reaction. Hazard is mismatched incompatible blood transfusion. Major transfusion disorders of RBC incompatibility, RH incompatibility. Immediate effect. The acute hemolytic reactions occur within a few minutes of mismatched transfusion. It develops because of rapid hemolysis of donor's RBC. Symptoms include shivering and restlessness, nausea and vomiting, chest and back pain, pulse and respiratory rate increased, blood pressure decreased, shock. Delayed effect of late hazards. Delayed hemolytic reactions occur due from 1 to 5 days of transfusion. The hemolytic RBC results in the large amount of hemoglobin in the plasma, which leads to the following complications: inapparent hemolysis post-transfusion and jaundice due to hemolysis, hemoglobin and hemoglobinuria, urea, and acute kidney shutdown. What are the complication hazards of blood transfusion? Fever and allergic reaction. Transfusion reactions due to RBC incompatibility or RH incompatibility. Shivering, restlessness, shock, post-transfusion, jaundice, hemolysis, hemoglobinemia, and hemoglobinuria, acute kidney shutdown. Reactions due to massive transfusion, circulation overload, circulatory shock. Hyperkalemia due to increased potassium concentration stored but an increased potassium concentration. Hypocalcemia leading to tetany due to the massive transfusion of saturated blood. Increased potassium concentration in the stored blood. Hypokalemia and hypokalemia and hypocalcemia, massive transfusion due to the massive transfusion of saturated blood. Hemosiderosis, increased deposit of the iron from hemosiderin in organs such as endocrine glands, heart, liver, due to iron overload of the repeated transfusions. Hemosiderosis, reactions due to faulty techniques during blood transfusion. Thrombophlebitis, air embolism, faulty techniques, reactions due to faulty techniques during blood transfusion. Thrombophlebitis, inflammation in veins. Thrombophlebitis and air embolism. Blood transfusion without precaution leads to transmission of blood infections. Hepatitis C, hepatitis B, C, AD, malaria, syphilis, malaria, and glandular fever infections molecules and caused by aspirin, barvirus, papyrus, simplex, and bacterial infections. Mention the chemical risks of blood transfusion. Chemical risks of blood transfusion: hyperkalemia due to excess potassium concentration in blood stores, hypocalcemia due to limited to massive transfusions of saturated blood. Hypocalcemia and hyperkalemia. Alkalosis, the patient with renal failure due to excess HCO3. Describe the mechanism of acute renal shutdown due to mismatched blood transfusion. Acute renal shutdown (ARS) is one of the most lethal effects of transfusion reactions. It can begin within a few hours and continue until the person dies in renal failure. Mechanism: the RS results from three causes: renal and vasoconstriction, circulatory shock, renal tubular blockage by excessive hemoglobin. Explanation: renal vasoconstriction, the antibody reaction of the transfusion reaction unless toxic substance from the renal vasoconstriction. Explanation: renal cause powerful renal vasoconstriction. Circulatory shock leads to loss of circulatory red cells along the product of toxic substance from the hemolyzed cell to produce immune reactions cause circulatory shock. The arterial blood pressure falls very low and the renal blood flow urine output decreases. Renal tubular blockage, the total amount of free hemoglobin in circulatory blood is greater than the quality of that can be haptoglobin. Plasma protein binds very small that can be haptoglobin. Much of the excess leaks into the globular membrane and block the kidney tubules. Renal tubular, the total amount of hemoglobin in the circulatory blood is greater than the quantity that can be bound with the haptoglobin. A plasma protein that binds a small amount of hemoglobin, haptoglobin, causes increased leaks through the glandular membranes of the blocked kidney tubules. Thus, renal vasoconstriction, circulatory shock, renal tubular blockage within together cause acute renal shutdown. If the shutdown is complete and fails to open up, the patient dies within a week or 12 days. Alternative answers: physiological mechanism of AKI, hemolytic transfusion reaction, immune mediated hemolysis. Donors RBC are destroyed in recipients RBC antibodies. Massive intravascular hemolysis, free hemoglobin in plasma. Hemoglobin toxicity of tubular damage. Free hemoglobin exceeds hepatoglobin binds capacity, unbinds FB filters into renal tubules. HB breakdown, release in iron, oxidative stress and formation of hemoglobin pass. Renal vasoconstriction and ischemia, hemolysis, release in endothelin, nitric oxide scavenging, and reduced RGF, glomerular fibrillation rate. Acute tubular necrosis, hypoxia, hemoglobin toxicity, necrosis and proximal tubes, sloughing of dead cells, backplate kidney filtrate, secondary DIC, disment intravascular coagulation, hemolysis active coagulation, microthrombotic renal vessels, ischemic injury. Summary of the pathology and physiology: mismatched transfusion, antibody mediated hemolysis, free HB, renal vasoconstriction, tubular obstruction, oxidative damage, ATM, AI, anuria and creatinine and hyperkalemia. Identified conditions: acute tubular necrosis, acute kidney injury causes mismatched blood transfusion, intravascular hemolysis, hemoglobin, renal vasoconstriction, and tubular damage. Key features: anuria, oliguria, 40 ml per day, more less than 40 ml per day, rising serum concentration, azotemia, hyperkalemia, metabolic acidosis, possible uremia. Physiological mechanism of KI in hemolytic transfusion reactions. Why do jaundice occur in mismatched blood transfusion? Why mechanisms of KIT hemolytic transfusion reaction? Jaundice and mismatched transfusion. If mismatched transfusion, there is an increased hemolytic red cells. This result decreases to increased bilirubin production and development jaundice. Increased bilirubin production, which causes jaundice. Bilirubin production mechanism: the mismatched transfusion, hemolysis, free RBC, heme, congenital bilirubin, liver overwhelmed, and jaundice. Yellow skin, yellow cells. Rh incompatibility. The most common blood groups in India: O positive 32.53%, O negative 2.03%, A positive 21.8%, A negative 1.63%, B positive 32.9%, AB 7.7%, AB negative 0.48%. Physiological basis of ABO system. The antigens A and B are complex oligosaccharides. The antigen A and B are complex oligosaccharides differing in their terminal sugars. These are found on red cells differing on their terminal sugars. Complex oligosaccharides differing in their terminal sugars. Those found on red cells are glycolipids, while those found in tissue and the bodies are soluble glycoproteins. The fructose containing H antigen is the basic antigen and is found in all individuals. In case of antigen A, transferase places an acetylgalactosamine. A transferase places an acetylgalactosamine as the terminal sugar on protein on antigen H. In antigen B, the terminal sugar is galactose. In case of antigen A, a transferase places an acetylgalactosamine as the terminal sugar on antigen H. On antigen A, the transfer places an acetylgalactosamine as sugar in galactase. In AB persons in acetylgalactosamine. In B, the terminal sugar is galactase. In AB persons, both transferases are present. While group O individuals have none of the enzymes. So the antigen H persists in them. Normally, H antigen has no antigenic activity, and as a result, it is identified by the capital O letter. H antigen is not antigenic. There is no corresponding antibodies. It seems that group O persons produce a protein that has no transferase activity. This results from single base deletion of corresponding gene. Genetic basis of ABO system. The blood group of a person is determined by two genes, one of each of two paired chromosomes. These genes can be one of three types, A, B, or O, but only one type is present on each of the two chromosomes. The O gene is functionless and does not produce O antigen in red cells, while A and B genes produce strong antigens in red cells. O gene is functionless and does not produce O antigen on red cells, while A and B genes produce strong antigens on red cells. O gene is functionless and does not produce O antigen red cells. Produce strong antigens on red cells. Thus, there are 6 possible combinations of genes: AA, BB, AB, OA, OB, and OO. Each person one is of these 6 genotypes. A person with genotype O has no antigen on red blood cells, so the blood group is O. A person with genotype AA or OA is group A, while BB or OB is blood group B. Agglutins of ABO system. The antibodies in the plasma are gamma globulins. The agglutins react with antigen A is called anti-A or alpha and reaction with B antigen is called anti-B or beta. Blood groups O has no antigens, both anti-A and anti-B antibodies in the plasma. Both group AB has both A and B antigens but no antibodies. Reacting with B beta. Blood group O has no antigens but both anti-A and anti-B antibodies in the plasma. Blood group AB has both A and B antigens but no antibodies. The immune system forms antibodies against whichever ABO blood group antigens are not formed in the individual's RBC. The immune system forms antibodies against whichever ABO group antigen is not found in the individual's RBC. Thus, group individual will have anti-B antibodies. And group B individual will have anti-A antibodies. These antibodies are present without any specific red cell antigen stimulus. For example, they are absent in a newborn. The AB antibodies start appearing in the plasma by the age of 3 to 4 months. AB antibodies, due to the cross-reactivity of ABO antigens present in naturally occurring bacteria. 3 months. Due to the cross-reaction of ABO antigens present in naturally occurring bacteria, viruses, pollen, etc. present in the environment. For example, they are absent newborn. ABO antibodies start appearing 3 to 4 months. Present in the environment. Rh factor. Rh factor. In addition to antigens in the ABO system, the red cell of 80 to 85% of humans also contain an additional antigen called Rh antigen or Rh factor. The factor is named so because its antigen was discovered in rhesus monkey by Landsteiner and Wiener in 1940. Rh rhesus monkey. They injected red blood cells of rhesus monkey in the very common Indian variety with red ischial callocytes into rabbits. The rabbit's immune system reacted by forming antibodies against the rhesus red cells, and when the rabbit plasma was tested against human red cell, and agglutination occurred in 80 to 85% of individuals. Person whose red cells contain the additional antigen is called Rh positive. Those who lack in this antigen are called Rh negative. There are several varieties of Rh antigen, C, D, E and CDE, but D antigen is the most common and antigenetically the most potent. Therefore, Rh plus positive persons are also called RHD positive, and negative are called RHD negative. The antibody D antigen is called anti-D antibody or Rh antibody. However, there are no natural occurring antibodies against RHD. The Rh antigen is not present in the body fluids and tissues but only on red cells. This antigen is a warm antigen and can cross the placenta easily. The difference between ABO and Rh antibodies are given in table 12. Principle. The surface of red cell membrane contains a variety of genetically determined antigens called isoantigens. The surface of red cells membrane contains a variety of genetically determined antigens called isoantigens. Or agglutinogens. While the plasma contains antibodies, agglutinin. To determine the blood group of a plasma, his or her red cells are made to react with commercially available antisera containing common agglutins. The slide is then and examined under microscope to detect the presence or absence of clumping and hemolysis, agglutination of red cells which occur as a result of antigen-antibody reaction. There is a reciprocal reaction between antigens and red blood cells and antibodies in the serum. It's known as the Landsteiner's law. The reciprocal relation is known as Landsteiner's law. The Landsteiner's law, 1900, which has two major component sites that if an agglutinogen is present on the red cell of an individual, the corresponding agglutinins must be absent in the plasma. Agglutinins, antibodies. If an agglutinogen is absent on red blood cells, the corresponding agglutinins must be present on plasma. Note: exception to the second components of the law is that absence of Rh antigens from the red cells in the Rh positive is not accompanied by the presence of anti-Rh agglutinins. Obviously, this component of the law was enunciated before the discovery of Rh factor in 1940 by Landsteiner and Weiner. Apparatus: microscope, glass dropper with a long nozzle, sterile blood lucent in needle, sterile cotton, gauze swabs, alcohol, 5 ml test tube and toothpicks. Clean dry microscope slides. A principal procylindrital with 12 depressions in the is available for this purpose and may be used in places of glass dishes. Normal saline, 0.9%. Anti-A serum. This contains monoclonal anti-A antibodies against human. Anti-A serum. This contains monoclonal anti-A antibodies against humans. Monoclonal anti-A antibodies. These antibodies are also called anti-A or alpha. These antibodies are also called anti-A or alpha agglutinins. The anti-A serum can also be obtained from a person of blood group B. Anti-B anti-serum. This contains monoclonal anti-B antibodies. Monoclonal anti-B antibodies against humans. These antibodies are also called anti-B or beta agglutinins. The anti-B serum can be obtained from a person with blood group A. Anti-D or serum. This contains monoclonal anti-Rhd antibodies against humans. These antibodies are also called the anti-D agglutinins. Note: the large volume requirements for high quality ABO and Rh typing reagents can now be supplied by selected monoclonal antibodies. Monoclonal antibodies are antibodies produced by a single clone of cell or cell line and consisting of identical antibody molecules. A monoclonal antibody is created by exposing a white blood cell to a particular antigen protein, which is then cloned to mass produce antibodies to target the antigen. The monoclonal antibodies are antibodies produced by a single clone of cell or cell line consisting of identical antibody molecules. The monoclonal antibody is created by exposing a white blood cell to a particular antigen protein, which is then cloned to mass produce antibodies to target the antigen. The antisera are available commercially for a quick identification. The anti-A is tint blue. This tint blue. Anti-B is yellow. Serum is yellow, while the anti-D serum is colorless. The antibody against the Rh factor do not occur naturally. Do not interchange the droppers provided with the Antisera bottles. Procedure: preparation of red cell suspension. A suspension of red cells in a saline should preferably be prepared and used instead of adding blood drops directly from the finger prick to the Antisera for the following reasons. Dilution of blood permits easy detection of anti-agglutinated and hemolysis if present. Red cells in undiluted blood tend to form a large rolex and masses. These may be difficult to disperse and may be mistaken for agglutination. Plasma factors likely to interfere with agglutinogens are eliminated. Method: put 2 ml of saline of a small 5 ml test tube. Put 2 ml of saline in a 5 ml test tube. Then get a finger prick and allow the blood dot to form. Now place the prickle fingertip on the top of the test tube and invert it. Mix the blood and the saline by inverting the tube with 2 or 3 times. The suspension of red cells is now ready. Determination of blood groups. Mark the wells in blood group inside slides as A, B, and D separately. Put 1 drop of antiserum B, antiserum, and D antiserum separately in 3 well marked as A, B, and D on blood cells. In addition, take 1 drop of normal saline as control in the separate well. Add a drop of cell suspension from the bottom of the test tube by a dropper on each anti-A, anti-B, and anti-D serum, and 1 drop of the normal saline taken on the control slide. The normal nozzle of daughter should not touch any of the antisera. In this way, the red cell saline mixture on the control side will act as a control to confirm agglutinations or no agglutinations on the corresponding test slide. Gently mix the antisera and red cell suspension using 3 separate application sticks. Wait for 8 to 10 minutes. There inspect the red and the antisera cell mixed and the control mixture. First with the naked eye to see either agglutinations, clumping of red cells have been taken place or not. Then confirm under a low magnification microscope, comparing the text mixture with the control mixture. If there is no agglutination, the RBC remain evenly distributed and separate from each other. If there is agglutination, the RBC are clumped together and lose their outline. Reaction of different blood groups with different antibodies given in table 11 and 13. Determination of blood groups showing agglutination, hemolysis, and clumping of red cells, and no agglutination cells remain uniformly distributed. The reaction between the anti-D serum and the red cells is not shown. In agglutination, RBC are meshed together in clumps and lose their outline. If there is no agglutination, RBC remains separated and evenly distributed. Determination of blood groups. Denote agglutination. Agglutination and antigen A serum, antigen B serum, group agglutinogens, agglutinins, and agglutinate RBC groups. Note: agglutinin against agglutinogen not present in red cell is present in the plasma each type. There are no naturally occurring antibodies against anti-D antigen. It is obviously from the above that if a known type A or B is available, it is possible to determine the red group of any unknown person. If blood type A or B is available, it is possible to determine the red blood group of any unknown person. False positive reaction. This can occur when the ancilla are contaminated with bacterial growth. False negative reaction. It may occur due to the improper storage of antisera, leading to loss of potency. So bacterial growth and loss of potency. Observation and results. It is essential that you should be able to distinguish between agglutination and no agglutination. The features of each are as follows. Agglutination. If agglutination occurs, it is usually visible to the naked eye. The hemolyzed red cells appear as isolated, separate, dark red masses, clumps on different sizes and shapes. There is brick red coloring of the serum by the hemoglobin released from ruptured red cells. Tilting or rocking the slide a few times or blowing on it does not break or disperse the clumps. Under the low power LP objective, the clumps are visible as dark masses and the outline of the red cells cannot be seen. No agglutination. In the control mixture, the red cells may form a branch on Rolex. These sediments red cells give a orange tinge of suspension of red cells rather than isolated dark red masses of ruptured red cells. The red cells will disappear or disperse if you genuinely blow on the slides or tilt them a few times. No attenuation. Red cells will gently blow on the slides. In the control mixture, the red cells may form a branch or a roulette. This is sedimented red cells given in an orange tinge of suspension rather than an isolated dark red mass. Orange tinge of suspension. Rolex. Formation occurs mechanically due to orderly linear stacks of RBC where the cells overlap each other. It is reversible on shaking, whereas RBC agglutination is an by grape-like RBC aggregates. In agglutination, the outline of the individual RBCs cannot be distinguished. Confirm all these features of no agglutination under the microscope. Since similar agglutinogens and agglutinins, i.e. A and anti-B, B and anti-B, D and anti-D, would react with each other, they cannot exist in the same individual. Therefore, the presence or absence of these agglutinations will indicate your blood group, as shown in table 13. Where the sign denotes the agglutination, the sign indicates no agglutination. Physiological significance of blood grouping. Physiological significance of blood grouping. Blood grouping or typing is important in blood transfusion for treatment purpose. Blood transfusion is a life-saving procedure in all cases for severe loss of blood and in life-threatening anemias. However, blood can only be given after blood grouping, which is an essential requirement before blood is given to any individual. Determination of RH incompatibility between the mother and the child. Paternity disputes. The ABO, RH and MNS blood grouping is used to settle cases of disputed paternity. Antigens A and B are denoted, whereas O is recessive. It is possible to produce the proof that a person could not have been the father, but not that he was or is the father. DNA fingerprinting is now a recognized procedure for settling much disputed, as it can prove paternityhood with 100% accuracy. To prove a person is not only father, selection of donor issue or organ transplant. Three main blood tests that will determine if a patient or a potential blood donor is compatible matches for organ transplant are blood typing, tissue typing, and cross-matching. Blood typing, tissue typing, and cross-matching. Genetic studies, molecular type of blood grouping, genes to provide to be a powerful tool. Genetic studies, molecular typing of blood group genes is provided to be a powerful tool, preventing alloantibodies formation with potential advantages for identifying rare blood types and finding better antigen matches for chronically transferred patients. This has allowed the development of a plethora of DNA tests to predict blood group antigens. Everybody has an ABO blood type, A, B, AB, and O, and an Rh factor, positive or negative, just like hair or eye color. Everybody has an ABO blood type, A, B, or O, or Rh factor negative, just like eye or hair color. The blood type is inherited from the parents. Each biological parent donates one of two pair of ABO genes in the child. The A and the B genes are dominated, and the O gene is recessive. Dominated and the O gene is recessive. For example, if an B gene is paired with an A gene, the blood type will be A. For example, if an O gene is paired with A, because scientists often use methods to identify blood types, blood typing, because an individual blood type isn't affected by disease, drugs, climate, occupation, or living condition, or any other physiological circumstance. Additionally, scientists use blood type to determine paternity. It is obvious that if a known type A or B is available, it is possible to determine the blood group of any unknown person. Blood group is available. Medico-legal use. Any red stain or clothing may be claimed to be blood by a supposed victim. Therefore, it is called confirmed that it is really human blood. Blood grouping of that extracted sample can be proved or disproved the claim of a victim. In doubtful cases, the DNA fingerprinting can decide the claim on a way that no other. Medical use. Medicological use. Susceptibility to disease. The people of blood type O are more susceptible to peptic ulcer. Blood type A is more commonly seen in sarcoma of stomach and to some extent diabetes mellitus. A positive correlation has been shown between blood group A with chronic hepatitis B infections and peptic cancer, and blood group B with ovarian cancer. Infected with A is hepatic B, infected with pancreatic cancer, hepatitis B and blood group B with ovarian cancer. Protection against falciparum malaria can be achieved in blood group O people. Blood group O also increase the severity of infection in certain Vibrio cholera strains. In blood people against falciparum malaria can be achieved protection in blood group O people. Blood group O also increases the severity of infection Vibrio collagen strains. Physiological significance of Rh factor. Although there are no natural anti-Rh antibodies and they never develop spontaneously, they can be produced only in Rh negative persons. This can happen in either two ways. One, when Rh negative person is given Rh positive blood, and two, when Rh negative mother carries an Rh positive fetus. In transfusions, when an Rh negative person receives Rh positive blood, there is no immediate reaction since there are no antibodies. But during the next few weeks or months, he or she may produce Rh negative antibodies that will remain in the blood. Even 0.5 milli Rh blood is enough to produce an immune response. However, if within a few weeks or even a years later a second Rh factor is injected, the newly donated heart cells will be agglutinated and hemorrhaged, thus resulting in a serous transfusion reaction. In pregnancy, the most common problem due to Rh incompatibility may arise when Rh negative mother phenotype DD carries an Rh positive fetus phenotype DD or DD. Normally, no direct contact occurs between maternal and fetal bloods. However, if a small amount of positive blood, leaks from the fetus through the placenta into the mother's body, the mother's immune system will start making anti-Rh antibodies. This happens at the time of delivery when a small amount of fetal blood leaks into the mother as the placenta separates from the uterine wall. As a result, some mothers develop high concentrations of anti-Rh antibodies during the period following delivery. However, if a small amount of Rh positive blood leaks from the fetus into the placenta into the mother's birth, the mother's immune system will start making anti-Rh antibodies. This happens at the time of delivery when a small amount of fetal blood leaks in. As a result, some mothers develop high concentration anti-Rh bodies during period of following delivery. Therefore, the firstborn baby will not be affected unless the mother has previously received Rh positive blood transfusion. There are cases of fatal placental bleeding during pregnancy itself, and fetal blood may enter the mother's circulation. During the first pregnancy, however, the anti-Rh level do not reach enough level to cause complications. However, during the second subsequent pregnancies, the mother's Rh antibodies across the placental membrane into the fetus where they cause agglutinations. Rh antibodies across the placental membrane to the fetus. The clinical condition that develops in the fetus is called hemolytic disease of the newborn, hemolytic HDN, or erythroblastosis fetalis. Clinical form of ADN. The chief clinical form of ADN syndrome of ADN are hydrops fetalis and icterus gravis, neonatorum, and carciterus. Hydrops fetalis. If the hemolysis in the fetus is severe, it may die in the uterus, or the fetus may develop anemia, severe jaundice, or gross edema. Icterus gravis, neonatorum, grave jaundice of the newborn. Though the infant is born at term, there is jaundice, hemolytic jaundice, or becomes so within a day or so. Anemia may be absent for a few days, though reticulocyte count is high, and may nucleated red cells, erythroblasts, are present, hence the term erythroblastosis fetalis. And many nucleated red cells, erythroblasts, are present, hence the term erythroblastosis fetalis. Physiological significance of Rh factor. Although there are no natural anti-Rh antibodies, they never develop spontaneously. They can be produced only in Rh persons. Carnectaneous. In adults, the bile pigment cannot cross the blood-brain barrier (BBB). In adults, the bile pigment cannot cross the blood-brain barrier. In infants, the BBB is not fully developed, so that these pigments may pass into the brain and get disposed in the basal ganglia, giving them a bright yellow color. Blood-brain barrier. Note: Neurological syndromes of Carnicterus is rarely a complication of physiological jaundice of the newborn because there is no hypotoxin stimulus. Also, the bilirubin level is not as high as in HDN. The physiological jaundice is due to immaturity of the liver and disappears in a few days. Precautions: the slide should be dry, dust free, and grease free. Identify the marks all slides, containers, and test tubes clearly and legibly. Double check every step of the procedure. The drops supplied with the Antisera bottles should not be interchanged. Examine the slides with the naked eye, then under the microscope after 8 to 10 minutes before sera blood mixture dried up. Do not add undiluted blood from the finger prick directly to the Ancera for two reasons. One, the sera may get intermixed, and two, false positive undiluted blood directly to the Antisera may get intermixed and the two false positive may develop. Sometimes it is not possible to say what certainty whether agglutination has occurred or not. In such cases, the grouping must be repeated with diluted blood. A control should always be used to exclude false positive results. What are the dangers of blood transfusion? The following are the dangers associated with blood transfusion. Transfusion of disease. The donated blood has the potential of transmitting some serious diseases. It is therefore mandatory to test the blood from human immunodeficiency virus, HIV antibodies for AIDS, hepatitis B surface antigen, hepatitis C virus antibodies, syphilis, venereal disease research laboratory test, and malaria parasite. Incompatibility due to mismatched transfusion. This is the most serious and potentially fatal competition, where the transfusion reactions are immediate or delayed, as well as their severity is determined by the speed and extent of hemolysis and denoted red cells. Body aches and pain joints. Within a short time of starting the transfusion, the patient complains of severe pain in the back, limbs, and chest, and a severe suffocation and tightness in the chest. Symptoms are due to the blockage of capillaries by clamps of agglutinin cells, chills, and fever during accompanying pains. Renal failure. Acute renal kidney shutdown appears to result the three main causes. Substance of immune reaction. The toxic substance releases the hemolyzing blood cause powerful renal vasoconstrictions. These substances and diseases in circulating red cells frequently cause circulatory shock. The arterial blood pressure falls in very low levels and renal blood flow and urine output decreases. The amount of free Hb or hemoglobin is present in a small deno whenever the filter is reabsorbed. If this amount is large, it gets precipitated and blocks many tubules. The result of these factors is acute renal failure, and death may occur within 8 to 10 days if the shutdown is not resolved or treated within dialysis. Faulty technique of transfusion. Cardiac arrhythmias, cardiac arrest, or circulatory overload may occur especially in elderly patients. Cardiac anemia, heart or kidney disease. If repeated transfusion are given, say 24 hours, thrombophlebitis may occur. If the intravenous needle remains in the same site for many hours, air embolism i.e., enter into the air into the blood via the transvenous needle is much less likely to occur because of use of the plastic bags instead of glass bottles, which collapse down as they empty out of blood. Allergic reactions. Reactions such as skin rashes and asthma may occur if the donor blood contains substance to which the patient is sensitive. Pyrogenic reactions. Reactions such as chills and fevers are probably due to the presence of antibodies in leukocytes and platelets. Pyrogenic reactions. Chills and fevers probably due to the presence of antibodies and leukocytes platelets. Tetany. With massive transfusions, the normal conversion of citrate to bicarbonate in the fusion may be delayed. This will result in fall in plasma ionic calcium and hence tetany. Iron overload. Repeated transfusion in the absence of blood loss may lead to hemochromatitis. Repeated transfusion in the absence of blood loss. What precautions are taken while selecting a blood donor? The following precautions are observed. The donor should be healthy and aged between 18 years and 60 years. Pregnant and lactating women are excluded. Any healthy adult, both male and female, can donate every 3 months. The donor between 18 and 60. The donor should be screened for communicable disease such as AIDS, hepatitis, syphilis, malaria, etc. The malaria parasite can survive at 4 degrees Celsius for 3 weeks. 4 degrees Celsius for 3 weeks. The donor's hemoglobin should be within the normal range, usually above 12.5 gram percent, plus between 12.5% plus between 50 to 100 minutes with no irregulations. Between 50 to 100 minutes, no regulation. Blood pressure systolic 100 to 180, hemoglobin, and diastolic 50 to 100, 50 to 100 to 180, and 50 to 100. Body weight not less than 45 kg. Professional donors must be discharged. Professional donors must be discouraged. What are the blood banks? How is donated blood stored? What is the fate of transfused citrate in the body? With the modern surgical and medical procedure, the demand for blood has greatly increased. It is for this reason that blood banks were started where blood from voluntary donors could be stored so that it was always available on demand. Most blood banks have a list of would-be donors so that they may be contacted when required. Storage of blood. After a donor has been screened for donation, 1 unit of whole blood, 500 ml, is collected under aseptic conditions from the antecubital vein directly into the special plastic bag containing 63 ml of CPDA, citrate phosphate detoxated adenine. Citrate phosphate detroxadenine mixture. Citrate phosphate detroxadenine mixture. The blood bag is suitably sealed, labeled, and stored in 4 degrees Celsius, where it can be kept for 20 days. Faulty storage, i.e., overheating or freezing, can lead to gross infection or hemolysis. The citrate prevents clotting of blood. Sodium disulfate acts as a buffer to control disease in pH. The dextrose supports adenosine triphosphate ATP generation via glycolytic pathways and also provides energy for sodium potassium pump that maintains the size and the shape of red cells and increases the survival time. The adenine provides substrate for the synthesis of ATP, thus improving post-donation valuably to red cells. Blood stored at low temperature for two reasons. One, it decreases bacterial growth, and two, it decreases the rate of glycolysis and thus prevents a quick fall in pH. Component, volume and contents. Whole blood 1 unit 500 ml. Red blood cell, platelets and plasma RBC in inactive solution 1 unit 350 ml. FPP of other plasma products. All soluble plasma proteins and clotting factors. Cryopreset called cryo platelets. Changes in cells, red cells during storage. Changes occur due to the decreased metabolism and include increase in their and sodium and potassium concentration due to the reduced sodium potassium pump activity. The result being net increase in total base and water content for the cells that swell and become more spherocytic. The ATP content decreased and inorganic phosphate content increases. Changes after transfusion. These changes occur within a day or so, or red cells lose sodium, gain potassium, with the volume shape and fragility returning to normal. Their survival time increases of blood is given within a week or donation. Fate of transfused citrate. The citrate in the form of dry sodium citrate that is used in the stored blood can safely be injected intravenously. Oxalate or toxic within a few minutes. The liver removes the citrate from blood and polymerizes it into glucose. A metabolite is directly for energy, but if the liver is damaged or large amount of citrate are injected too quickly, the citrate may lower the calcium level in the blood to result in tetany or even death from convulsions. Blood substitutes. Separate components of blood, packed RBC, whole blood, fresh frozen plasma, PFFP, fresh frozen plasma, to provide clotting factors, platelets and leukocyte plasma and plasma expanders are now available. What is the only certain way to avoid blood incompatibility? The slides and contents must be properly labeled. Cross-matching is the only certain way to avoiding the dangers of mismatching. Rh plus positive blood never be given to Rh negative female or any age before menopause. Name the precautions you will observe before during blood transfusion. Blood should be transfused only when absolutely required. It should be confirmed that cross-matching has been done. Exclude mismatching of groups such as ABO system. The blood bag should be checked of the blood group indicated on it. It should be confirmed that the block has packed infections, especially AIDS. Rh positive blood should never be given to Rh negative person. Blood should never be transfused at a fast rate, usually not more than 20 to 25 drops per minute, unless otherwise indicated, as in acute or severe loss of blood where the blood may have to be pumped into the patient. A rapid transfusion under normal condition may cause chelation of calcium ions and tetany. The condition of the recipient should be checked carefully for the first 10 to 15 minutes of starting transfusion, and from the time of later on the transfusion must be stopped if there is a rapid rise of temperature 40 degrees Celsius or any other reaction. What are the earliest effects of mismatched transfusion? Within a short time of starting the transfusion, i.e., when a few milliliters of blood have entered the recipient's body, there may be severe pain anywhere in the body, as a sense of suffocation or tightness in the chest. There may be chills and shivering, fever, etc. Which blood substitute may be used in restored blood volumes? It should be donor blood is not available. The blood volume may be reduced due hemorrhage. Plasma volume may be reduced due burns, vomiting, or diarrhea. Therefore, the ideal treatment would be depend on which body fluid needs in replacement. Whole blood. Though it is the ideal medium to replace lost blood because it replaces cell and plasma in physiological ratio, it may not be available in such situations. An intravenous drip of crystalloid or colloid solution is immediately started. This role is also available for any drug that may be needed and of course for later transfusions. Crystalloid solutions. A commonly used intravenous fluid is glucose saline. Intravenous fluid is glucose saline. 6% glucose in 9% sodium chloride. 6% glucose. Sodium chloride, 0.5 sodium chloride. However, since crystalloids leave the circulation within a short time, this solution resolves the blood volume temporarily. Colloid solutions. Various colloid substitutes are available to increase the plasma volume. They are called plasma expanders and include human albumin, dextrose in NaCl, and a polymer of degraded gelatin. Plasma separated from donated blood can be stored in the liquid from FFP, fresh frozen plasma, for many months or for a year. It is dried, it is constituted with distilled water just before use. Blood substitutes. Under certain conditions, instead of whole blood of plasma, a blood component may be required, say, to avoid circulatory overload, lower risk of transfusion, etc. These blood substitutes include packed red cell, white red cell concentrations, immunoglobulins, and clotting factor concentrate. What is Bombay blood group? The Bombay blood group is a rare blood group. Phenotypes of this blood lacking H antigen on the red cell membrane and have anti-H serum. It fails to express any A, B, or H antigen, and their red cells are other tissue. H antigen deficiency known as Bombay phenotype. As known as OH is found in 1 in 10,000 individuals in India and 1 in 1 million in Europe. Since there is no H antigen, there is no antigen A or antigen B in the red cells. However, the plasma contains anti-A and anti-B and anti-H antibodies. As a result, such a person can receive blood only from a person having Bombay blood type. What is forward and reverse blood typing? ABO testing is a two-part process involving testing a person's red blood cell for A and B antibodies as well as testing a person's serum and plasma for ABO antibodies. The first step is blood grouping or typing procedure, i.e. the blood cells are mixed with antibodies against type A and B, and the sample is checked to see whether the blood cells stick together. The blood cells stick together, it means the red cells react with one of the antibodies. For example, if the blood cells agglutinate when mixed with antibodies matched against type B, anti-B agglutinates, it is type B blood. The procedure is called blood typing or forward blood typing. The second step is called back typing or a reverse blood typing or serum typing. The liquid part of the blood without the red cells plasma is mixed with the red blood cells containing known antigens, i.e. red blood cells persons with blood type A, B, and AB and O. If agglutination occurs with a AB red cell, the blood type is B. If agglutination occurs with the B and AB red cells, the blood type is A. If agglutination occurs in AB and AB red cells, the blood type is O. And if there is no agglutination in any RBC, the blood type is AB. In AB blood type, there are no antibodies in plasma. Reverse typing can also be performed with commercial preparations of type A and type B erythrocytes. Thus, the forward grouping typing suggests that presence or absence of A and B antigens in RBC, where reverse grouping antibodies with antigen or absence of antigen A and B antigen serum. Unlike abiotyping, a reverse test is not performed for Rh system because the person does not have performed antibody to D antigen unless they have previously allowed allogenized D antigen transfusion or pregnancy. The serum is alone the blood typing as precaution because leukocyte BC may antigens. Blood typing is the process of determining the Rh factors and supply of blood. Cross-matching involves feeding into donor patients. If antibodies exist in recipient pledged antigens or red cells in the donor, transfusions can occur. Hemoglobin. Definition: hemoglobin is a chromatoprotein consisting of protein globins united with pigment heme. Normal values: male 14 থেকে 18, 12 থেকে 15.5, 14 থেকে 18, 12 থেকে 15.5, 15.5 in average, 14 point in average. At birth, neonate 23, at the first year 12. Functions of hemoglobin: transports O2 from the lung to the tissue and CO2 from the tissue to the lungs. It is an important blood buffer, maintains acid-base balance of blood. Formations of various pigments and bilirubin, stercobilin, urobilin, etc. It preserves the iron and protein. Give the chemical structure of hemoglobin. Hemoglobin is a globular protein made out of 4 subunits. Each subunit contains a heme moiety conjugated to a peptide. Figure: structures of hemoglobin and reaction of heme O2. The abbreviation MPV stands for the group showing the molecular on the left. MCH3, VCH2, NN, PCH2CH2CH. Heme. Heme is an iron containing porphyrin, known as iron porphyrin FIX. The porphyrin nucleus consists of 4 pyrrole rings joined together by 4 methane bridges. The porphyrs are thus tetraroles. The pyrrole rings are numbered 1, 2, 3, 4. There are 8 side chains attached to 4 pyrroles. The position to which the side chemicals are attached are numbered 1 to 8. The side chains 1, 3, 5 and 8 are methyl, and 2 and 4 vinylene, and 6 and 7 are proanoic acid, CH3, CH2, CH2, COOH. The iron in heme is the ferrous Fe2+ from Fe2+ is attached to the N to form pyrrole and to the N. The iron is in heme is the ferrous from 2 of the imidazole group of the globin. There are 2 pairs of polypeptide in each hemoglobin molecule. The polypeptides are referred to collectively as the globin protein of the hemoglobin molecule. The iron, the normal adult human hemoglobin A. The 2 types of polypeptide are called alpha chains, which contain 141 amino acids. 141 amino acids, and the beta chain, which contain 146 amino acids. Thus, hemoglobin A is is distinct as alpha 2 and beta 2. Describe briefly the steps of hemoglobin. Hemoglobin synthesis. Hemoglobin synthesis begins in the basophil erythroblast stage and is completed in the orthochromatic hematoblastic stage. Begins at the basophil stage, basophil erythroblastic stage, and is completed in the orthochromatic erythroblastic stage. Heme synthesis. Raw materials: succinyl-CoA, glycine and iron. Side cell synthesis in mitochondria. Heme synthesis happens on mitochondria. Raw materials are succinyl-CoA and glycine and iron. Steps: the acetic acid is converted to succinyl-CoA in Krebs cycle. Acetic acid converted to succinyl-CoA, 2 succinyl-CoA, 2 glycine pyrrole, 4 pyrrole porphyrin and protoporphyrin IX, ferrum plus heme. Globin synthesis, raw materials, amino acids, side cell synthesis, iron in ribosome. Steps: DNA transcription, mRNA translation, polypeptide, globin. Finally, heme plus polypeptide equal to hemoglobin chain alpha from beta. 2 alpha chains plus 2 beta chains equal to hemoglobin A. Enumerate factors of hemoglobin synthesis. Name the dietary factors of hemoglobin synthesis. The factors of HB hemoglobin synthesis. The dietary factors. First class protein required for globin synthesis. Metals: iron, copper, cobalt and manganese are essential for heme synthesis. Copper, cobalt and manganese act as catalysts. So is constitute of vitamin B12. Vitamins B12, C, folic acid, riboflavin and nicotinic acid are necessary. Endocrine gland. The thyroid, adrenal cortex, pituitary gland contribute indirectly to the formation of hemoglobin. Porphyrins, they are readily synthesized in the body. Bowel salt, necessary for absorption of metals such as iron, copper, cobalt and manganese. Genetic control. Structured assay questions. Hemoglobin. Why abnormal hemoglobin leads to anemia? Abnormal hemoglobin leads to anemia through 3 primary mechanisms. Impaired red blood cell. Impaired red cell production. Defective globin chains. Thalassemia, hemoglobinopathy, sickle cell disease. Increased RBC destruction, hemolysis. Unstable hemoglobins. Sickle cell disease. Sickle RBC block microvasculate. Ischemia, intravascular hemolysis. Reduced oxygen delivery in tissue, hypoxia. Methemoglobinemia. Why is hemoglobin enclosed inside the RBC? Hemoglobin is enclosed in the RBC. Free would pass quickly through the globular membrane and is excreted through urine. Globular membrane. Free hemoglobin would increase the viscosity of blood. Free hemoglobin would increase colloidal osmotic pressure of blood. If the hemoglobin is outside the RBC, it will be unable to carry CO2 from tissue due to the absence of carbonic anhydrase in plasma. For these reasons, hemoglobin remains enclosed inside the RBC. What are the functions of RBC in hemoglobin molecules? Functions of globule in hemoglobin molecules. Globin helps in iron to remain in ferrous form. Globin helps to combine O2 loosely and reversely. How can hemoglobin molecule carry four molecules? Hemoglobin molecules carry four O2 molecules. The iron in the heme is in ferrous state. Fe2 plus is attached to the end of the H pyrrole ring to the end of the amibodiazole group in hemoglobin. One bond is available for loose union of O2. Thus, one ferrous ion can bind only one O2 molecule. As one molecule helps in Hb containing four ferrous ions, it can carry four O2 molecules. Hemoglobin structural formula. Adult. Hemoglobin A, alpha 2 beta 2. Hemoglobin A2, alpha 2 delta 2. Fetal. Hemoglobin F, alpha 2 gamma 2. Hemoglobin by bats, hemoglobin grower. Gamma 2 delta 2 E2. Hemoglobin F, hemoglobin A. Hemoglobin grower 1, hemoglobin grower 2, embryonic. Hemoglobin Portland. Pathological abnormals. Hemoglobin S, hemoglobin C, hemoglobin E. Hemoglobin D, Punjab. Hemoglobin O, Arabia. Hemoglobin M, Suston. Hemoglobin G, Sandoz. Was first found in Punjab. New Library Hospital. Differentiate between hemoglobin A and hemoglobin F. Hemoglobin A, alpha 2 beta 2. In adult, alpha 2 delta gamma 2. In adult, in fetus, also in adults. Some adults. O2 affinity less, O2 affinity more. Building effects of PDB more, less. The comparison about 95% of the hemoglobin of adult cells. The greater affinity for O2 facilitates movement of O2 from material to the fetal circulation. Short note: hemoglobin F. Hemoglobin F or fetal hemoglobin is normally present on the blood of human fetus. It is normally replaced by adult hemoglobin A as soon as after birth. In certain individuals, it fails to disappear and persists after a life. The structures consists of two alpha 2 gamma chains. Importance: hemoglobin F has greater affinity for O2 than HBO because it binds easily avidly with 3,2-DPG. This facilitates movement of CO2 from the maternal to the fetal circulation. However, it is more resistant to alkali than hemoglobin A. Resistant to alkali. Classify hemoglobin, their functional differences. Globulin types and their functional differences. Adult hemoglobin A. Functional hemoglobin A2, hemoglobin F, hemoglobin H, hemoglobin HB bars, hemoglobin S, hemoglobin C, and hemoglobin E, metheoglobin, and carboxyhemoglobin. The key functional differences: oxygen affinity, hemoglobin F, hemoglobin A, hemoglobin A2, hemoglobin S, C. Pathological effects: hemoglobin S, sickling; hemoglobin B, hemoglobin S, Barts, thalassemia. Developmental role: age hemoglobin F, developmental role HbA adult, fate of RBC and catabolism of hemoglobin. briefly discuss about the fate of RBC with mention lifespan and etc. Define and classify jaundice. Jaundice. Fate of RBC. The lifespan of RBC is about 120 days. As the age of RBC increases, the enzymes which protect the RBC from the damaging effects of oxygen begin to lose their efficiency. On the other hand, RBC cannot synthesize new enzymes due to absence of nucleus. So the oxidative damage begins to appear and RBC becomes rigid, less deformable, spheroid or fragile. During circulation, the RBC has to pass through a separate space in the red pulp of the spleen. The spaces have a diameter of 3 micrometer in comparison to 8 micrometer of the RBC. So extreme alternations in shape and are required for the red cells to pass through the space successfully. Reduced deformity makes the passage difficult and leads to the sequential RBC within them in the red pulp. Then mononuclear phagocytic cells in the spleen phagocytose the RBC and destroy them. The nuclei are pathogenic cells then breaks the hemoglobin and releases the iron and amino acids from hemoglobin to the blood and convert hemoglobin of the hemoglobin into bilirubin, which is also released in the blood. What is the graveyard of RBC? Graveyard of RBC. The RBC became older and fragile. They are destroyed while trying to squeeze through the capillaries. The destruction occurs mainly in the capillaries of the red pulp of the spleen because the diameter of the spleen capillary is very small. So the spleen is called the graveyard of RBC. Bone. What is bone? Bone is a specialized, living, highly vascular, mineralized, constantly changing, rigid connective tissue which forms the framework of the body. Bone is specialized connective tissue composed of calcified intracellular material. Bone matrix, the bone cell. Mention the features of the bone. A specialized rigid form of connective tissue composed of calcified intracellular material and bone cells form the rigid framework of the body and serves as a level for muscles. Forms consist of outer compact part and inner spongy part. The compact part is ivory-like and extremely porous. The spongy part is also known as the cancellous bone. Is composed of a network of trabecules covered by the periosteum except the articular surface. Sesamoid bone is devoid of periosteum. Anterior of the adult long bone presents a cylindrical medullary cavity which is filled with bone marrow which manufactures blood cells. Medullary cavity and is lined by vascular membrane known as the endosteum. Highly vascular and nerve supply acts as the storehouse of calcium and phosphate. Mention the physical properties of bone. Bone is rigid. Bone is radio opaque. Enumerate the functions of bone. Mechanical functions: gives protection, gives shape by forming rigid framework of the body, supports fleshy structures, acts as a lever for the muscles and helps in movement, transmits weights, acts as shock absorbers. Metabolic functions: material storage, growth factor storage, fat storage, acid-base balance, detoxification. Synthesis of functions: bone contains bone marrow which manufactures blood cells. Mention how bones differ from cartilage. Different between bone and cartilage. Bone, cartilage. Organic, more fiber, less ground substance. Inorganic, crystallic, present. And cartilage, organic, more ground substance, less fiber. More fiber, less ground substance. এখানে হচ্ছে less fiber, less ground substance, আর এখানে less fiber, cartilage. Few crystals are present irregularly. Calcification of matrix. Intercellular matrix calcified, intercellular matrix is uncalcified. Stability, more permanent, alive even in classified stage. Classified cartilage is dead tissue. Calcified cartilage is dead tissue. Vascularity, vascular, avascular. Bones get nerve supply, they have no nerve supply. Arrangement, arranged in a definite pattern termed Haversian system. Arranged in a definite pattern termed Haversian system. Such arrangement is not present. Haversian system. Canonical mechanism present, absent. Haversian mechanism present, canonical mechanism present. Covering periosteum and perichondrium. Growth only by oppositional method, are both oppositional and interstitial method. Classify bone according to shape and size. Long bone, short bone, flat bone, irregular bone, pneumatic bone, sesamoid bone, accessory bone. According to position and location. Axial bone, skull bone, vertebra, ribs, sternum. Appendical bone, upper limb. Pectoral girdle, free bones, lower limb. Pelvic girdle, free bones. According to ossification. Membranous bone, parietal bone, clavicle. Cartilaginous bone, limb bones. Membranocartilaginous bone, occipital bone, and mandible. Macroscopic classification. Compact bone and spongy cancellous bones. Cross section shows dense area. Spongy bone. Microscopic histological classification. Microscopic classification. Macroscopic classification. Woven primary immature bone composed of randomly arranged collagen fibers. Lamina and secondary mature bone composed of regular parallel bands of collagen fibers. Vertically all bones in healthy lamina. Woven or immature bone composed of randomly arranged collagen fibers. Randomly arranged collagen fibers composed of regular panels of bands of collagen fibers. Virtually all bones of the in a health adult lamina composed of regular parallel bands of collagen. Regular parallel bones of collagen fiber. Randomly arranged collagen fiber. Regular parallel bands of collagen fiber. Regular parallel bands of collagen fiber. Regular parallel bands of collagen fiber. Randomly arranged collagen fiber. Woven and laminar. So write down the morphological classification of the bone example. Morphological classification of bone. Long bone, typical. Humerus, femurs, radius, ulna, tibia, fibula, etc. Typical. Miniature, short, long bone. Metacarpal, miniature, short bone. Typical and atypical. Typical, humerus, femur, radius, ulna, fibula, tibia. Atypical, short bone and modified. Miniature, short bone and modified long bone. Metacarpal, metatarsal, phalangeal bone of the fingers and the toes. Modified long bone, clavicle, body of the vertebra. Modified long bone. Short bones, carpal and dorsal bones. Flat bones, ribs, sternum bones and the wall of the skull. Irregular bones, hip joint, scapula, vertebra, mandible. And pneumatic bones, maxilla, sphenoid and ethmoid bones, etc. Sesamoid bone, patella, pisiform and fibula, rider's bone. Accessory and supranumerical bone. These bones are regularly present. They are commonest of the skull. Sutural and omodian bone, accessory bone and interpartial bone, etc. Sesamoid bone, patella, pisiform, patellar rider's bone, sesamoid bone. Accessory and supranumerate bones. Sesamoid bones. Define long bone. Long bone is a bone that has two shafts and two ends and is longer than it's wide. Long bones is thick outside compact bone and inner medullary cavity. Compact bone and medullary cavity consist of bone marrow. Ends. Long bones are spongy bone The ends of the long bone are a spongy bone and epiphyseal line. All of the bones of the arms and the leg, except for patella, bones of the wrist, ankle, and long bone, except for the patella, the bones of the wrist and long bone. Characteristic features of long bone: length is greater than width, has two ends, shaft of the body. Typically, the shaft presents three surfaces separated by three borders. Transmits body weight. Characteristic features of long bone: rise vertically. Ossification from one primary and at least two secondary centers. One primary and two secondary centers. There will be at least two epiphyseal plates, one for each. Undergoes intracartilaginous ossification. Two epiphyseal plates and one for each end. At least two epiphyseal lines. What are the parts of a typical adult bone? The typical adult bone of the humerus consists of the following parts. Shaft. It is a long middle part of the adult human bone. It is almost totally composed of packed bone and thin, compact, spongy bone on the inner surface around the bone cavity, and two ends. The ends of the long bone are actually not compact but as the shaft. They are made out of spongy cancellous bones, a line of the compact bone. Classify bone morphological and type of long bone. Typical long bone. A typical long bone presents a diaphysis, at least two epiphyses. One at each end. Most of the long bones of the limbs of typical humerus, etc. Characteristics: rise vertically, ossifies from the one primary to secondary centers. There will be at least two epiphyseal plates on each end. Undergoes intercartilaginous ossification. Miniature long bone or short long bone. The long bone presents a single epiphysis at the end, one only. A single vase at the end, one only. Metacarpals, metatarsals, and phalangeal bones and megatoes are examples of this type. Characteristics: small in-field ossification from one primary and one secondary center. Will be one epiphyseal plate. Undergoes intracartilaginous ossification. Epiphyses of all metacarpal cattle bones are directed towards the head, except for the first, where they ate directly towards the base. Long bones derived from the typical features. A clavicle may lie horizontally, may have more than one primary ossification center. Undergoes intramembranous ossification. Why clavicle and the body of the vertebra are modified long bone? The clavicle is a modified long bone in spite of the fact that it divides a medullary cavity. Undergoes intramembranous ossification. This is due to the fact that functionally clavicle is weight bearing and transmits the weight of the upper limbs axial like a long bone. Because of the similar reason, body of the vertebra is modified long bone. Explained from an knowledge of anatomical how metacarpal bone is miniature long bone. Metacarpal was miniature long, characteristic small size, ossification from one primary one secondary center, possesses a single epiphysis at one end only. There will be one epiphyseal plate. Intracartilaginous ossification. Metacarpal bones fulfill all the above criteria. They possess only one epiphysis at its distal end, except the first, which has its epiphysis at the coaxial end only. That's why it is a miniature long bone. Tell about the short bones of the flat bone. Short bones, they are cubical in shape. Possess 6 surfaces, 4 surfaces in the articular and the remaining 2 surfaces are non-articular. Short bones, for example, carpal and tarsal bones. Flat bones, they are plate-like. They consist of 2 plates of compact bone of intervening spongy bone marrow. The intervening spongy tissue of the bone is a vault of the skull known as the diplo. Consists of numerous veins. Example: rich sternum bones, vault of the skull. Functions: flat bones form the boundaries of some of the bony cavities and gives protection to important organs. The sternum and gives protection to the heart and the lungs. Skull bones gives protection to the brain. What is the macroscopical characteristic of the bone. The macroscopical of a flat bone is characterized by an outer table of compact bone and an inner table of compact bone. Between there is dipole or a spongy bone. What are the characteristics of irregular bone? Give example. Irregular bone: vertebra, scapula, mandible. Most of the bones of the skull, non-definite shape, have multiple processes. Define the pneumatic bones of the example and function. Pneumo means air. Pneumo. Their bones contain air-filled spaces which are limited by mucous membrane. Characteristics. Features of pneumatic bone. Pneumatic bones are confined to close proximity of the nasal cavity. The spongy bone, these bones absorb and form a cavity which is filled with air, called air spaces, air sinuses. Example of pneumatic bone: maxilla, sphenoid, ethmoid, frontal and temporal bone, etc. Functions and pneumatic bone: lighten and weight of the skull, add resonance to the voice, give help in development of craniofacial region, help in dentition, help in dentition, air conductor of adding humidity and temperature inside air. The word sesamoid is an Arabic origin. Sesamoid means seed. Sesam means seed. Bones develop from seeds in the tendons of the some muscles. These tendons are subject to friction and during the movement of the joints. Sesamoid bones act as pulleys for muscular contraction. Patella pisiform. They include patella pisiform, fibula and rider's bone. Patella pisiform, fibula and rider's bone. And the adductor longus, the gastrocnemius, flexor carpi. Preculatories of the sesamoid bone. Sesamoid shape develops from the tendon of the muscles. Ossifies after birth, devoid of periosteum. Absence reproduction system. Write down the complications of the bone. Write down the composition of the bone. The bone is considered cell and intracellular subcellular matrix. The bone cell constitutes of 2% of bone weight. 4 types of cells are recognized in the bone tissue: osteoprogenitor cells, osteoblasts, osteoclasts, and osteocytes. Bone matrix. Matrix consists of 2 parts: organic and inorganic. Organic matrix 35%. Organic matrix or osteo tissue consists of type 1 collagen fiber, ground substance, glycoproteins, proteoglycans. Glycoproteins include osteocalcin, osteopontine, and osteonectin. Osteocalcin, osteopontine, and osteonectin. Osteopontine and osteonectin. Osteocalcin. Proteoglycans. Glycoproteins, proteoglycans containing glycosaminoglycans and chondroitin sulfate. Proteoglycans and caratins, proteoglycans. Inorganic matrix: calcium, phosphate, magnesium. The most common salt in the calcium phosphate is remains of hydroxypate crystal. Explain anatomically, histologically. Bone is a rigid form of connective tissue. Bone is rigid due to 3 features: presence of calcium and PO4 from crystalline salt called hydroxypate. These crystals form plaques. Regular parallel arrangement of the collagen fibers associated of plate hydroxyapate crystals with collagen fibers. Enumerate the bone cells. 4 types of bones are recognized in the bone tissue: osteoprogenitor cell, osteoblasts, osteoclasts, and osteocytes. Osteoprogenitor cells, they are undifferentiated. Pluripotent stem cells derived from the connective tissue of the mesenchyme and lie with the inner cellular layer of the periosteum. Main function: during bone development, osteoprogenitor cells proliferate during mitosis and differentiate between osteoblasts, which secrete collagen fibers, and bony matrix, osteoblasts are precursor cells of osteocytes, and they are developed from osteoprogenitor cells. They are located exclusively on the surface of the bone. Osteoblasts A mature but active cell synthesizes the organic compound of the matrix. Osteocytes are mature from osteoblasts and principal cells of the bone. They are trapped by surrounding bone matrix produced by osteoblasts. They occupy cave-like spaces known as lacunae of the matrix. They are only osteocytes found in each lacuna. Main function: mature inactive cells that are responsible for maintaining the bone matrix. Osteoclast. The osteoclasts are large multinucleated cells derived from mononuclear phagocytic system (MPS). Osteoclast. NPS, mononuclear system. The bone marrow do not belong to the osteoprogenitor cell. They do not belong to osteoprogenitor. They are found in bone surface where or reabsorption, removal of the bone and repair take place. House shapes lacunae or resorption lacunae. Main function: they are phagocytic cell of bone. They are involved in the respiration and remodeling of the bone tissue. Osteoclast. Give the structure of the individual bone cells. Osteoprogenitor cells. They are pluripotent stem cells derived from mesenchyme. Osteoblast. Shape cuboidal. Cytoplasm. Each osteoblast cell presents an abundant cytoplasm with some cytoplasmic process. The cytoplasm is strongly basophilic due to the ribosomes granular to the endoplasmic reticulum in addition to RER, rough endoplasmic reticulum. Cytoplasm by Golgi apparatus, mitochondria, other granules. Nucleus large, rounded, and eccentric in position. Osteocyte shape, spindle shaped cell process. Cytoplasm less basophilic. Osteocytes. Osteoblast. Basophilic. Osteocytes are less basophilic, poor in organelles with scanty of rough endoplasmic reticulum. The cells have numerous cytoplasmic process. A osteocyte and a process occupy the lacuna and the canaliculi radiating from it. Process of the adjacent cells makes connective via gap junction. A nucleus flattened, centrally placed with more condensed nuclear chromatin. Osteoclast shape and size very large, rounded grand cell cytoplasm and eosinophilic and devoid of any process. The cytoplasm contains Golgi apparatus and numerous lysosomes. Osteoclast. Nucleomatid giant cell nucleus. Multinucleated giant. Multinucleated 10 to 20 giant cell. Osteoclast. Special features: the cells are provided with brush border which are produced by numerous microvilli. Brush border: the cells are provided with brush border with numerous microvilli. In active osteoclast, the surface against the bone matrix is folded into irregular projections. In active osteoclast, the surface against the bone matrix is folded into irregular projections which form a rough border. Surrounded by the rough border, the clear cytoplasm zone is rich in actin fiber. Surrounded the clear zone, there is a basal zone conducting nuclei, mitochondria, and Golgi apparatus. Mention the fate of osteoblasts. Some of the cells are converted into the osteocytes. Both osteoblasts and osteocytes do not divide by mitosis. Some are converted beneath periosteum as differentiated osteoprogenitor cells. Enumerate the functions of individual bone cells. Progenitor cells. During bone development, they proliferate by mitosis and differentiate into osteoblasts, which then secrete collagen fibers into bony matrix. Osteoblasts. They are immature but active cells and are responsible for the synthesis of organic components of bone matrix consisting of type 1 collagen fibers, proteoglycan, several glycoproteins, and including osteonectin, and also may be involved in bone mineralization. The osteoblasts increase the surface area of the bone by a positional method. Osteocytes. The mature but inactive cells are responsible for maintenance of bone matrix. They are phagocytic cells of the bone, are involved in the reabsorption of the modeling of the bone. They are participate in the long bone maintenance of blood circulation homeostasis by the response of parathyroid hormone and calcium. Parathyroid hormone and calcitonin, osteonectin, glycoproteins including osteoblasts, Enumerate the difference between osteoblast and osteoclast. Osteoblast develop in osteoprogenitor cells developed from osteoprogenitor cells do not belong to osteoprogenitor cell. They are derived from the mononuclear phagocytic system of the bone marrow, exclusively on the bone surface. At the surface of the bone within the Haversians, house ships lacunae, cuboidal, large rounded shape, multinucleated, strong basophilic, eosinophilic. Osteoclasts are eosinophilic, whereas are strongly basophilic. Cytoplasmic process absent, present. Cytoplasmic absent. Fate. Some of the cells converted into the osteoclast, some of the converted beneath periosteum and as undifferentiated osteoprogenitor cells. They dissociate with mononuclear cells of bone marrow. Function. Synthesize organic compounds of the bone matrix. Reabsorption of remodeling of the bone. Enumerate different parts of the growing, developing long bone. Parts of the young growing bone. This diaphysis is a part of the growing long bone which ossifies from the primary center of ossification. It forms the main part of the adult bone. Epiphysis. Diaphysis is the part of the growing long bone ossifies from the primary center. Epiphysis is the part of the growing long bone which ossifies from the secondary center of ossification from accessory part of the adult bone. Epiphyseal plate. The plate of harding cartilage separates epiphysis from the diaphysis growing long bone of the epiphyseal plate. Metaphysis. The part of diaphysis close to the epiphyseal plate is called metaphysis. A bone grows in length at the expense of metaphysis. Epiphysis. Epiphysis is the part of the growing long bone which ossifies from the secondary center. Epiphysis ossifies from the secondary center of ossification and forms the accessory part of the adult bone. Types of epiphysis. Pressure epiphysis. Epiphysis develops due to the weight transmission of called pressure epiphysis. It transmits the body weight and protects the epiphyseal cartilage. Examples: head of the humerus, femurous matters, upper end of the ulna, condyles and tibia and humerus, lower end of the fibula. Traction epiphysis. Epiphysis which develops due to muscle pull. Example: upper end of the fibula, trapezoidal femur, trabecular humerus. Example: traction epiphysis, pressure epiphysis. Epiphysis develops from the weight transmission of the pressure epiphysis, and which epiphysis develops from the due to muscle pull. Upper part of the fibula, femur, and humerus. Apparent epiphysis. The epiphysis is unusual located called apparent epiphysis. Example: head of the first metacarpal and the metatarsal bone. Apparent epiphysis. Atavistic epiphysis. The epiphysis which is fused in other bone for better nutrition is called atavistic epiphysis. It grows like a parasite. Fused with which is fused with other bone for better nutrition. Example: the two classification in human body: coracoid pressure of the scapula and posterior tubercle of the tarsus. Scapulocoracoid process and posterior tubercle of the talus. Metacarpal bones possess only one epiphysis at the distal end except for the first, which is epiphysis at the proximal end only. Define metaphysis. Metaphysis, epiphyseal end of the diaphysis is known as metaphysis. Clinical importance of metaphysis. It is the most active growing area of the long bone, has profuse blood supply from nutrients and prosteal and juxta metaphyseal arteries, where nutrient arteries from hairpin-like capillary loops. Microorganisms circulate the blood. Many settle of the loops, hence infection of the long bones occurs primarily at the metaphysis. Nutrients of hairpin-like capillary loops. Hence, infection was primarily the metaphysis. metaphysis. Here, nutrition arteries from hairpin-like capillaries, microorganisms circulating the bone may settle at these loops, hence malfunction loops, and profuse with the blood supply. Actively growing area, muscles, ligaments, joint capsules are attached to the metaphysis. As a result, the area is likely to be damaged by shearing stains of muscles. Some of the parts of the metaphysis may lie within the scapula of the joint. In such circumstances, the distress of the metaphysis may affect the joint and vice versa. Any disturbance of the metaphysis can growth retardation. Right above the epiphyseal cartilage, the plate of hyaline cartilage separated epiphysis from the diaphysis of a growing long bone is called epiphyseal cartilage, also called epiphyseal plate of the growth plate. A plate of epiphyseal cartilage divided into 5 zones, starting from the epiphyseal side of the cartilage: resting zone, proliferating zone, hypertrophic cartilage zone, classified cartilage zone, and ossification zone. Functions and general features. The epiphyseal cartilage is responsible for the growth to the length of the different adults. With the bone increases in adulthood, elimination of the epiphyseal plate. Epiphyseal eclosion occurs in different times with different bones and is complete in all bones over the age of 20. In forensic, through the X-ray examination of the growing skeleton, it is possible to determine bone age. Of a young person, noticing the epiphyses are open and which are closed. Which epiphyses are open and which are closed. Epiphysis occurs. Elimination of the different epiphyseal plates occurs at different times with different bones, including all bones by the age of 20. Bones are open and closed. Open or closed. Epiphyseal cartilage is responsible for the growth of the length of adults, which is the growth of adulthood. Once the epiphyses have close growth in length of the bones become impossible, although bone widening may still occur. As long bones grow in length, epiphyseal cartilage persists. When the length is reached, usually after puberty, the epiphyseal cartilage is replaced by bone. More than one epiphysis may be present in one or both ends of the long bone. In such cases, when the individual epiphysis unites with the diaphysis by separate epiphyseal plate, this is known as the simple epiphysis. This type found is upper out of the femurs. If the multiple epiphyses unite first with another, later fuse with the epiphyseal plate, then these are known as the compound epiphysis. Upper and lower end of the humerus will belong to this type. A typical long bone presents epiphyseal at the each end. The epiphyseal union does not take place simultaneously at both ends. One epiphysis fuses with the apparatus either than the other one. The epiphysis which unites last with the diaphysis grows for a long period before union, hence is known as the growing end of the bone. What is growing end? What is a growing end? It is the end of the bone where the secondary center appears first and the unites last with the diaphysis. It is the end of the long bone where the secondary center appears first and unites last with the diaphysis. Direction. The bone root direction is situated against the direction of the nutrient artery, nutrient foramen of the bone. The bone of the end situated against the direction of the nutrient artery. The direction of the nutrient foramen of the long bone can be remembered by using the following dictum: to the elbow I go, from the knee I flee. To the elbow I go, to the knee I flee. Therefore, the upper extremity, the shoulder, and the waist end of the bones are growing ends, and the lower extremity knees ends of the femur tibia are growing ends. Clinical importance. The knowledge of the growing ends is important in clinical practice. An injury or infection of this end in young age makes the bone structured in growth. Bone end of the radius. The growing end is situated against the direction of the nutrient foramen of the bone. The direction of the nutrient foramen of the long bone can be remembered by using the following dictum: to the elbow I go, to the knee I flee. Therefore, upper extremity, shoulder, waist, and the verse are grow ends. So wrist and lower end radius is the growing end. For some reasons, shoulder ends, upper end, humerus is the growing end. Growth of the bone. It may be defined. It may be defined as the process by which the size of the bone increases. Type of bone growth. Interstitial growth. Growth in the length which occurs to 25 years of age. This occurs in the growth zone of the bone. Appositional growth. Growth in the width which occurs throughout the life. This occurs by the help of periosteum. Write in brief about the growth of the long bone. Growth of the long bone, both in length and in width. Growing length. Epiphyseal fluid or cartilage is responsible for the growing in length of long bone. It is accomplished by the proliferation and the interstellar growth of cartilage of epiphyseal plates. Growth in width. Bone growth in width by appositional growth. It is accomplished growth subperiosteal deposition of the bone by osteoprogenital cells of inner cellular layer of periosteum. A long bone grows in length by intracartilaginous ossification of breadth by intramembranous ossification. Once the epiphyses have closed around the age of 20, longitudinal growth of the bones become impossible. Epiphyses, but widening may still occur. Name the factors acting bone growth. Factors affecting the bone growth. Genetic factors. Hormonal factors. Thyroid hormone, growth hormone, thyroid hormone, parathyroid hormone, calcium hormone, sex steroid hormone. And nutritional factors. Adequate blood supply, adequate amount of minerals and vitamin D, C, A. Mechanical factors. Tensile force. What is the genetic? What is the decalcification of bone? Decalcification of the bone means the loss of calcium from bone. Because of concentration of calcium tissue in the blood must be kept constant. Nutrition deficiency of the calcium results in decalcification of the bone. Severe decalcification of the bone are more likely to fracture. Decalcification of the bone may also be caused by excessive production of PTH. Hyperthyroidism that can cause increased osteoclast activity produced by PTH. Hyperthyroidism and inabsorption of blood. Intestinal reabsorption of blood elevate the bone levels. Increased osteoclast activity and levels abnormal deposits calcium in kidneys and arterial walls. Blood supply of the bone. Mention the blood supply of the developing bone. Blood supply of the developing bone. A developing bone is supplied by 4 sets of blood supplies. Nutritional supply, metaphyseal or juxtaphyseal arteries, epiphyseal arteries and periosteal arteries. Epiphyseal and periosteal. Epiphyseal and periosteal. Metaphyseal and nutrient arteries. Artery and course and area supplied by it. Nutritional artery. Some bones such as femurs have several nutritional arteries where the other bones tibia have only one. Some tibia have several nutritional arteries where other bones like tibia have only one. Course. The nutrition artery enters in the middle of the shaft of the bone through the nutrient foramen, runs obliquely, reaches the medullary cavity where it divides into ascending and descending branches. Each branch subdivides into a number of small parallel vessels which terminate with the adult epiphysis by the it supplies the bone marrow and inner 2/3, the compact part of the diaphysis. It supplies the bone marrow and the inner 2/3, the compact bone of the diaphysis. Area of the pipeline. Inner 2/3 of the compact diaphysis. Juxta epiphyseal or arises from the anastomosic joints. It supplies the metaphyseal arteries. Epiphyseal artery arises from the anastomosing around the joints. It supplies the epiphysis. Periosteal arteries. They ramify beneath the periosteum, enter the bone through the Volkmann's canal. The supplies of outer 3/3 of the compact part of the bony bone. Inner 2/3, the nutrient artery, inner 2/3 of the compact for nutrient artery, and periosteal artery 1/2 and 1/3 of the compact bone. 1/3 of the cartilage bone. nutritional artery, metaphyseal artery, epiphyseal artery, and periosteal arteries. Give the source of blood supply by movement of flat bones. Short, nutrient short, long bones. Nutrient artery, metaphyseal artery, epiphyseal artery, and periosteal artery. Flat bones, nutritional artery, and periosteal artery. Nutritional artery and periosteal artery. Mention the artery supply of the typical adult long bone. Artery supply of the typical adult long bone. Shaft of a typical adult long bone is supplied by nutrient and periosteal artery. Ends of typical adult bone were supplied by the artery extremity of the around the joint. Draw and label the parts of the typical adult long bone showing its source of blood supply. Parts of the typical adult bone with the source of blood supply: upper and epiphyseal artery, periosteal artery, epiphyseal artery, periosteal artery, nutrient artery, periosteal artery, nutrient artery. Nutrient artery, metaphyseal artery, and metaphyseal artery. Arterial supply of the long bone. Macroscopic anatomy of the bones. Macroscopic. From exterior to the anterior bone consists of periosteum, bone tissue, periosteum. Dead bone loses periosteum during preparation. Bone tissue proper: outer compact bone, inner spongy bone, medullary bone in case of adult bone, and endostem in case of adult bone. Odd question: Viva, the adult long bone, periosteum, bone tissue proper, medullary, and endostem. Periosteum, endostem, mediastinum, the marrow. Periosteum, bone tissue proper, medullary, and endostem. Write down the structure. The functions of periosteum. The outer surface of the bone marrow is covered by a layer of dense, irregular connective tissue called periosteum. Periosteum is the outer covering of the bone. The fibrovascular cellular layer covering by the external surface of the living bone, except the arterial surface, is called periosteum. Living bones are covered by the periosteum everywhere except the arterial surface. It is also absent from the sesamoid bone. Periosteum, the outer surface of the bone covered by the dense, irregular connective tissue. Dense, irregular connective tissue is called periosteum. But fibrovascular cellular bones is called periosteum. Periosteum is attached to the bone through some collagen fibers called Sharper's fibers. Structural layers of periosteum. The function: periosteum consists of 3 layers. Outer fibrous layer, it consists of collagen fibers. Function: it protects the bone, maintains the shape of the bone, helps in maintaining muscular functions. Medial vascular fiber, it contains the periosteal bone vessels which supply the 1/3 of the compact part of the bone. Medial periosteal functions. Medial vascular supply and inner cellular supply. Inner cellular osteogenic layer. The innermost cellular layer of the osteoid contains of mesenchymal cells and osteoprogenitor cells that are divided by osteoblast different osteoblast function. It helps in growth in bone in width by positional method. Osteoprogenitor cells are the main cells in a process that helps in repair of the bone when fractured. Functions of the periosteum: protect the bone, receives the attachment of the muscle, tendon, and shape of the bone, gives nutrition to the outer part of the backbone by periosteal vessels, helps in increase the weight of the bone, repair the bone when fractured. Explain from your knowledge of anatomy how periosteum is attached to the bone. The way periosteum is attached to the bone through some collagen fibers called perforating Sharper's fibers. Mention the periosteum. Bone growth. Bone growth is generally associated with parietal reabsorption of the reformed tissue and simultaneous laying down of the new bone. The process permits the shape of the bone to be maintained as it grows. The innermost cellular layer of the periosteum contains mesenchymal cells during osteoprogenitor cells with the potential divide into meiosis and differentiate into osteoblasts. Thus, osteoprogenitor cells play a prominent role in bone growth repair. Explain anatomically. Developmentally, periosteum takes part in repair of the bone fracture. Periosteum. Because bone contains osteoprogenitor cells throughout the endostenium and periosteum and has an extensive blood supply, it has a excellent capacity for repair and regeneration. Periosteum is important in the repair of function when it is absent. For example, within the joint capsule of the femoral neck, fractures are slow to heal process. When a bone is fractured, blood vessels are disrupted and bone cells are adjoining other features die. The damaged vessels produced a localized hemorrhage to form a blood clot. Soon the blood clot is removed by macrophages and adjacent matrix of the bones absorbed by the osteoclast. The periosteal of the endothelium at the site of the fracture responds to the intense proliferation, producing a soft cellulose fibrous like tissue that surrounds the fractures and covers the extremities of the fractured bone. Primary bone is then found by combination of the endochondral and intermediate ossification. Further repair produces irregular bones and bones incompatible with the extremities of the fractured bone, forming a hard bone cellus. The primary bone in the cellus is gradually reabsorbed and replaced by secondary bone, remodeling the restoring the original bone structure. If the structure of a compact bone, this is the structure of a compact bone. Endochondrial periosteum takes. Explain how the anatomy how periosteum is attached to the bone. Periosteum is attached to the bone through some collagen fibers of the periosteum surface fibers. Through collagen fibers. Protect bone. Role of periosteum in the bone growth. Bone growth is generally associated with periosteal reabsorption performed by tissue and stemleus laying down of the new bone. This process permeates the shape of the bone and maintains the growth. The innermost cellular layer of the periosteum contains mesenchymal cells, that osteoprogenitor cells, with the potential of divide into mitosis, differentiate into osteoblasts. Explain anatomically, developmentally, histological. Periosteum takes part in the repair of the bone fracture. Periosteum takes part in repair of bone fracture because bone contains osteoprogenitor stem cells throughout the endostenium and periosteum and has a extensive blood supply. It has excellent capacity for repair and regeneration. Periosteum is important for the repair of fractures, where it is absent. Within the joint capsule reformed neck, fractures are slow to heal. When a bone is fractured, blood vessels are distributed and bone cells adjoining the fractures die. The damaged blood vessels produced a localized hemorrhage from a localized hemorrhage. Soon the blood clot is removed by the macrophages and the adjacent matrix bone is restored by osteoclasts. The periosteum and the endostenium at the site of the fracture produce response to the intense proliferation produced by soft cellus of membranous cartilage like tissue that surrounds the fracture, covers the extremities of the fractured bone. Primary bone is then formed by a combination of endochondral and intermediate ossification. Primary bone is then formed by a combination of endochondral and intermediate ossification. Further repair proceeds to irregular bone formation in primary bone that temporarily unite the extremities of the fractured bone, forming a hard bone cellus. The primary bone of the cellus is generally reabsorbed by the replaced by the secondary bone, remodeling and restoring the original bone structure. Histological structure of the compact bone. Histological of the bone are arranged in the number of cylindrical units known as Haversian system and second osteo. The Haversian system runs longitudinally in long bone branch and anastomose. Definition of Haversian system. The cylindrical structure unit the compact bone is Haversian system. Components of Haversian system. Haversian canal. Each system has a central channel called Haversian canal, which runs parallel to the long edges of the bone. Each canal contains blood vessels, slings, and nerves, lamellae. Each Haversian channel is surrounded by concentric bony layers called concentric lamellae. They are made out of layers of bony matrix. Haversian channel lamellae. Lacunae. These are the small spaces of innervated between the lamellae. Canaliculi. These are fine radiating channels which connect the lacunae with its channels, Haversian channels. And Volkmann's channel, Haversian channel, which are the medullary radiating channels which connect the lacunae with each other with the central Haversian channel. They contain cytoplasmic process of osteocytes. Volkmann's perforating channels. The Haversian channels communicate with each other with medullary cavity and with the surface of the bone of numerous oblique channels termed the Volkmann's channel. They contain blood vessels and nerves which permit the bones from the periosteum or endosteum. Volkmann's channel. Each Haversian system is about 150 micrometer in diameter. 150 micrometer in diameter, and central channel is about 20 micrometer in width. Each Haversian channel is about 50 micrometer in diameter and 20 micrometer in width. 150 micrometer in diameter and 20 micrometer in width. Surrounds each Haversian channel is the deposit of amorphous material called the cementary substance, which consists of materialized matrix and collagen fibers. Eponymous anatomy related bone. Howship's lacuna. Space of the surface of the bone containing osteoclast. Howship's lacuna. Space on the surface of the bone containing osteoclast. Sharp's fiber. Collagen fiber. These connect with periosteum bone. Collagen fibers. Haversian system with the base of the bone. Haversian system. Cylindrical structure unit of compact bone. Cylindrical structure units of compact bone. Central channel with Haversian system. Haversian canal. Central canal with Haversian system. Cylindrical structure units of compact bone. Haversian system. Volkmann's channel. Transverse channels that connect two Haversian channels or Haversian canals to Haversian canals to either periosteum or endosteum. Volkmann's extra bone pieces that occur within a Borman's bone. Extra bone pieces occur within a suture of the cranium. What are the types of lamina in long bone? Haversian lamina surrounded by Haversian channel. Outer circumferential lamina immediately beneath the periosteum. Inner circumference laminae surrounded the medullary cavity. Interstitial laminae between the Haversian channels. Tell the morphology of the compact spongy bone. Morphology of the compact bone, ivory-like, extremely porous, consists of number of cylindrical in the Haversian system. Morphology system of the bone, honeycombed, calamated without Haversian system, possess large marrow space containing red bone marrow. What is the difference between the compact bone and spongy bone? Development of Haversian system. No development of Haversian system. Cross section of the compact exchange. It is generally without cavities. Cross section of the spongy bone will numerous connections with trabeculae. It contains bone marrow with the medullary cavity before to the 20 red bone marrow after the yellow bone marrow. It contains numerous red bone marrow. Nice to know long bones of bulbous and either epiphysis of the spongy bone. The cylindrical part of the diaphragm almost fully composed of a bone with a thin layer of cavity. Short bone, or a spongy bone, flat bone. Short note: bone marrow. Bone marrow is a flat vascular system which is present in medullary cavity. Long bones, spongy bone types. It presents with two forms: red bone marrow. In newborn, entire skeleton occupy the red bone marrow. By the stage of 20, red bone marrow found in the scalp only. And so it consists of interlacing network of reticular fibers into which reticular cells are attached. Hematopoietic stem cells are located in this network. Yellow bone marrow. At the age of 20, almost all the bone changes into yellow bone marrow, which consists mainly of adipose cells in the admixture of macrophages and differentiated mesenchymal reticular cells functions. Hematopoiesis. It is the chief blood forming organ of the adult body. Storage of iron in the form of ferritin and hemodioxide. Destruction of RBC. Initiation of the toxic substance. Storage of fat. Short note endosternum. Long bone are lined internally in the endosternum. Medullary cavity or adult bone is lined by a skull membrane known as the endosternum. Endosternum is a single layer of osteogenic cells that line large intermedially narrow cavities of adult long bone. It is thinner and more cellular than periosteum. Composition. It is formed by the flattened osteoblast precursor cells of reticular tertiary collagen fibers and lies all the intermediate cavities of bone, including the Haversian channels functions. In resulting adult bone, osteoblast osteochondral cells are present chiefly on the endosteal surface, which act as a principal reservoir of bone marrow forming cell growth and repair. The endosternum provides a surface approximately 7.5 meters square endosternum through important calcium homeostasis. Describes the vertebral column. The vertebral column is a central axis obstructing the dorsal semioids. Vertebral cervical, thoracic, lumbar, sacral, and coccygeal. Length male 70 cm and female 60 cm. Vovial or true. Groups movable or true, fixed or false. Cervical through the lumbar thoracic. These include the sacrum and the coccyx. Curvature of the vertebral column. The sagittal plane. Primary curvature. Present at birth. Primary curvature. The primary curvatures of the thoracic cervical both are concave forwards. Secondary curvature develops after birth. Secondary curvature of the lumbar, which are concave forwards, which are convex forward. Concave forward and convex forward. In congenital plane, there is a slightly lateral curve in the thoracic region with concavity towards the left, due to the greater use of the right upper limb and the pressure of the aorta. There is a slightly lateral curve to the thoracic region due to the greater use of the right upper limb and the pressure of the aorta. Vertebral column contains a spinal cord of containing membranes. Intervertebral movement of the intervertebral column. Flexion. A forward bending of the vertebral column. Extension. Forward bending of the vertebral. Backward bending of the vertebral column. Slide during the vertebral rotation. Twisting of the trunk. Circumduction. Combination of movements. Deformities. Kyphosis. Forward bending of the vertebral column. Lordosis. Backward movement of vertebral column. Kyphosis, lordosis, and scoliosis. Large bending of the vertebral column. Explain anatomically why cervical vertebral column is called secondary curvature. The cervical vertebral column is called secondary curvature because it develops after birth when the child lifts the head during 3rd or 4th months of postnatal life. Next to axial bone. Axial bone is the portion of humerus that consists of bone of the head and of the organism. It consists of 80 bones and composed of 8 pairs. The skull bone, classical bones, and the hyoid bone, the rib bone, and the skull bone. Pectoral girdles. Head, thighs, feet, and ankle.